That's the best.
Good morning, everyone.
Good morning.
Good morning.
Right. Hi, Willie.
Morning.
Gaurav, Ajay, Rajalingam, Yoginder Singh. Yoginder Singh? Oh,
Yes,
Yoginder.
sir, driving at the moment, driving.
Driving? Oh my God.
Concentrate.
Akanksha, are you there to start the proceedings in a while?
Yes, ma'am.
Okay. We have a few minutes here.
उमा उसका भेजना लियान का कहाँ पर है वो भी भेजना जरा मुझे.
हाँ जी सर, हम भेज रहे हैं.
So today Ranjana is not able to come for the session 11.
Yeah.
योगिंदर का पार्टनर भाग गया. तो इसको पकड़े. अरे अनिल हांडू तू कहाँ है?
वो सर वो पोस्टर सेशन करेंगे मॉडरेट आज.
आई नो, मुझे पता पोस्टर सेशन में की बात हुई उसके साथ अभी.
Rajalingam, would you stay at the end of the meeting?
Of course. Whatever Friday evening.
Rajalingam,
So maybe
Every day. 2 a.m. I stay until 2 a.m. Then come to 4 o'clock in the morning.
so,
yeah,
But I feel tired today.
Since Rajalingam has sleepless genes, so Rajalingam, why don't you moderate this session at 11 with Yoginder?
Oh, sure. 11, your time more than my time.
Ranjana is not
It's like a 1 o'clock.
So Ranjana is not able to come, so I think you do that. Since
Okay, I'll pour more coffee, that's it.
Since you have these sleepless genes.
I think that Raj, you could, because the discussion on the technology can be interesting.
Oh, yes, the NGS talk. Yeah,
The
I'll
NGS
stay
talks.
until then.
Ryan's going to talk on Oxford Nanopore, so you can compare the KDX with.
Yeah, so you are the final speaker. I will stay.
So for the 18th workshop, Eric had said that he will come. Maybe a reminder डाल दो जरा एक email.
Okay, and also, sir, he has sent a video which Ben has recorded for the 18th workshop data submission. So that video is already with Manas. So that they want us to play, and also there's a promotion video of the FE 2022. So these two, Eric has requested to play those two.
Eric का?
Eric ने भेजा है कल, हाँ जी. एक FE का भेजा हुआ है FE 2022 और एक भेजा हुआ है जो 18th workshop का data management के बारे में.
चला दो उस वक्त, हाँ ठीक है.
That Manas has already got. I have told Manas in that session, and we will again email Eric so that he also joins live. Then we can.
Live भी आ जाता उसके पहले इसको चला दे.
हाँ. So I'll send an email him right now. It must be early morning for him. I will send one.
Absolutely.
Willie, Narendra Mehra was my PhD PI.
What did you say?
Did you get it? So no, I was telling Willie worked with Peter Parham, same place where I went.
Yeah. We were debating at that time when Rajalingam had to go to Peter Parham, where to go. He said, I want to go for a year, and then he stayed back.
So I was telling Willie, yeah, so that Narendra was my PhD mentor, right? I did PhD under that.
I didn't know that.
After 3-4
on
years,
HLA.
Peter Parham was asking me after 3-4 years, why don't you ask him now to move somewhere else? The best was when Elaine Reed met me. I think we were in dinner, some US conference somewhere. And she spotted me. She said, "Oh, I want to sit with you. I want to talk to you about a candidate that we want to hire. And he has been your former student. Can you tell me about him?" I said, "Look, we are enjoying our dinner. I can only say one statement, that this fellow can work 26 hours a day. And I would say they get his signatures on a blank sheet. Just hire him."
Good advice.
Peter was helping me a lot. By fourth year, he was pushing me, "Hey, go and talk to him." He was literally pushing me. I don't think he ever did for other postdocs. So he was very kind enough.
So Willem was also there in Stanford?
Oh yeah.
Oh, yeah? With you?
No, he's,
I mean, you
he's
guys
little
were
senior,
together?
little senior.
Ah.
Yeah.
Great.
I think 2 or 3 years he left when I joined.
Oh,
So 95 probably you left.
I left in 93.
ah.
Yeah, so it's a long time. It's like
Yeah, but
4 years.
you went there in
Nine-
97, I think.
97. Yeah.
Yeah. Because we were doing the International Congress of Immunology in 98, and that was a huge task in front of us. I was the vice chair of the Congress, and I wanted a person like Rajalingam to be around. As I said, that I wanted somebody who could work 26 hours a day, but then he left and he said, "I will come back prior to the Congress, don't worry."
Yeah.
But then he wanted to VP over there, so that's fine.
So and Margaret, then Alejandro Madrigal is all
Oh, yeah.
similar
Alejandro
staff,
was-
similar. Yeah,
Alejandro
group.
was there, I know that. Yeah.
Yes, I was there with Ann Margaret, Alejandro.
Yeah, that group.
Oh, yeah.
William wanted to congratulate the Ishi meeting was really good.
I'm glad people enjoyed it.
Yeah, it was very nice. Nice talks.
It was a lot of work.
Yeah, a lot of
Lot
work.
of work. 2 meetings.
2 meetings.
So, okay, we are about 5 minutes more than the allotted time. We should at least start.
Akanksha?
Yeah.
Do you want me to just pull up my presentation at this time?
Somebody will introduce you, William, and the moderators will take care of that.
Is the Jonathan around?
Yep, I'm here.
Oh, okay. Hello. How
Hi.
are you?
I'm good, thanks, Sasha. I've not seen William's bio yet. Is it going to be on the screen for me
It
to?
will be on the screen, yeah.
Great. I can do reading. That's good.
Arey, Akanksha, kahan
I
hai?
think
Yes,
Akanksha,
I'm here.
Shuru
start.
kar.
A very good morning to everyone. Today is the last day of this wonderful conference, and I trust all the delegates have enjoyed this scientific feast for last 3 days. We had a galaxy of speakers with so much expertise in the histocompatibility and immunogenetic specialty. Today, the organizers have set up yet another day of learning via this virtual mode. With this, on the behalf of the organizing committee, we would like to welcome our moderators, Dr. Jonathan Downing and Dr. Varsha Trivedi. Dr. Downing is a scientist in charge in the Department of Clinical Immunology and Pathways, Stanley Hospital, Perth, Western Australia. Dr. Varsha is a senior scientist in Institute of Kidney Disease and Research Center, Dr. H.L. Trivedi Institute of Transplant Services, Ahmedabad, Gujarat, India. There will be a joint discussion at the end of this session, and we encourage the participant to kindly post their queries in the chat box. I now request the moderators to kindly take over the stage. Thank you. Thank you,
Thank you.
Akanksha. Yeah, Jonathan, go ahead.
Yeah, thanks, Sasha. Thanks for that introduction. I'd like to thank the organizing committee for asking me to participate in this meeting. On behalf, just quickly, of my fellow Aphia councillors, I'd like to express our gratitude for providing this joint platform and continuing the long history of collaboration in the Asia-Pacific region. So it's my pleasure to introduce the first speaker in this session on our last day, Dr. William Hildebrand. Professor Hildebrand's in the Department of Microbiology and in Immunology at the Oklahoma Health Sciences Center. and holds honorable awards for the Presbyterian Health Foundation Endowed Professor, scientific founder of the Pure Protein Company, and is on the board of directors for the AMA Society of that should be American Society for Histocompatibility and Immunogenetics. So I'm very happy to hand over to Professor Hildebrand. Thanks.
Thank you very much for that introduction, and I really appreciate the opportunity to present at your meeting. That's a great honor. Let me see if I can get my presentation pulled up here. And I hope that the moderators will let me know if that looks okay.
Yep, we can see that.
Okay, so what I'm going to tell you about today, my talk today is going to pertain to HLA and cellular therapies. Let's make sure this is working properly. I have two conflicts to disclose. I'm the chief scientist. I have equity in consulting at both Pure Protein and Pure MHC. That really doesn't impact my presentation today, but I need to disclose those conflicts. So the HLA class 1 molecules, the goal of my laboratory and also those companies for which I'm chief scientist is to determine the peptide complexes that are bound by class 1 HLA molecules on cancerous and virus infected cells. So the hypothesis of our group is that if we can accurately assess and determine the peptide HLA class 1 complexes on cancerous and virus infected cells, we can then develop immune therapies that'll use T cells or T cell receptors to destroy the cancerous and virus infected cell. That's really the whole hypothesis, the whole goal of our work is to identify peptide HLA complexes on cancerous and virus infected cells. So how I'm going to give that presentation, how we do that, first I'll start talking about viral class 1 neoantigens. So viral peptides that are presented by the class 1 HLA of virus infected cells. And I'll use West Nile virus as an example. I'll then, let me get my laser pointer going here. See if I can get that. Then I'll shift from discussing West Nile virus class 1 HLA peptide complexes to talking about melanoma or cancer class 1 HLA peptide complexes. I'll give you an example of our work in that area. And then I'll finish up with some HLA-based cancer immune therapies that are being used. And I'll have a little bit at the end about HLA-based companion diagnostics for this area of research and this area of clinical immune therapies. So I start with West Nile virus because I want to show that our group was able to find HLA peptide complexes distinct to West Nile virus infected cells and develop an immune therapy with that. And the work I show with West Nile virus will be tied into what we do with melanoma and then the third part of the conversation. So West Nile virus is a flavivirus. We find that in North America, these flavivirus infections rise every 5 to 10 years. So every 5 to 10 years, we see West Nile virus, St. Louis encephalitis virus, other flaviviruses have an outbreak. Where there's no vaccine for West Nile virus, there's no immunotherapy, and we know that T cell immunity is key. And if T cell immunity is key, then HLA molecules are key. So two PhD students in my lab really helped develop the system that I'm going to use as our experimental foundation. One is Kylie Prilliman. She's now a senior medical writer at Verista. She helps get protocols through the US FDA. And then the other PhD student in my lab is Heather Hickman. She's now the chief of viral immunity and pathogenesis at NIH, NIAID. And the system they designed a system whereby we could secrete HLA molecules. So rather than having HLA molecules stop at the surface, we designed a system with Heather and Kylie where HLA molecules are secreted from a cell line. So we take a human cell line and we secrete any desired HLA. In this instance, I'm going to talk about HLA-A2. Then we take West Nile virus and we infect this cell. So these are supposed to represent virus particles in the cell. So we can gather HLA from virus infected cells. We gather that HLA and then we take the peptide complexes out of the HLA molecule. Only the peptides come through the 3KD filter and we get tens to hundreds of thousands of peptides. So we fractionate those peptides on a preparative HPLC. So these lines each denote an HPLC fraction, and each fraction contains hundreds, if not thousands, of peptides. Each one of those peptide fractions then goes on to an electrospray time-of-flight mass spectrometer. So we further separate the peptides in each one of these fractions. And then we look at the peptide size or mass on this piece of equipment, the mass spectrometer. And we're also able to sequence the peptide fragment. So here you can see that we sequence the peptide from these HLA molecules that we gathered from the cell line that was infected with West Nile virus. In some of the instances, we detect a peptide sequence that we believe is from the virus. So we make a synthetic peptide to make sure that the peptide sequence is correct. We confirm that this peptide binds with a binding assay to HLA-A2. And then we test to see if West Nile virus infected people make an immune response to these peptides. So in this instance, we've gathered HLA from West Nile virus infected cells, identified the peptides presented by the HLA, identified a viral peptide, and shown that people make an immune response to that viral peptide HLA complex. So let's take a look at West Nile virus. West Nile virus, much like we were having a little conversation about COVID-19 a few minutes ago, West Nile virus is a positive sense RNA virus, much like COVID-19. It's smaller. This is a 3,000 amino acid polyprotein that West Nile virus encodes. COVID-19 is about 3 times the size. So COVID-19 is a larger virus. So the question is, when this viral polyprotein is inside your infected cell, how many peptides does the HLA molecule sample? And what Curtis McMurtry, who led this work, he was a PhD student in my lab, what Curtis McMurtry found is that he found 6 peptides from West Nile virus are sampled by HLA class 1. So it's interesting to see if this larger piece of the virus is inside your cell, what does the class 1 HLA molecule make available at the cell surface? And we found 6 peptides. So we thought, well, that's interesting. The HLA molecule is reducing a large virus down to 6 peptides and showing that at the cell surface. So our next question was, of these 6 viral peptides that the HLA-A2 makes available at the cell surface, how many of these peptides do you make an immune response to? What do T cells respond to? What we saw is that this peptide SVG9 here was the immune dominant. Almost all the A2 people made a strong immune response to this peptide. And I don't have time to fully characterize that response today, but it was a strong response. The T cells were polyfunctional. They made many different cytokines to this peptide. Then we saw that the second peptide, SLF9, here, infected individuals made a weaker immune response to this peptide, and also the T cells secreted fewer cytokines to this peptide. So we call this a subdominant response. And then the other peptides, like this YTM9 peptide, we referred to those as immunorecessive. There was very little immunity in infected individuals to this peptide. So we saw there was a hierarchy of one peptide was dominant. You made a strong immune response to this. A second peptide like SLF9 that was subdominant, there was some immunity to that. And then the other peptides resembled YTM9 in that they were weakly immunogenic. So that was interesting. You take a large virus, you reduce it down to 6 peptides, and you focus your immune response on one peptide. And most people fight West Nile virus quite well. So this is a successful immune response to this virus. And so that's an example of how HLA molecules elicit a successful immune response to a viral pathogen. Now, we were interested to know, when do these HLA peptide complexes from the virus come to the surface of the cell? Does this peptide get to the surface of the infected cell first? And is it much more common at the surface than these? Is that why it's immunodominant? So what we did was, here's your T cell receptor, here's your T cell receptor recognizing the piece of West Nile virus in an HLA molecule. We generated antibodies, and these are challenging to make. We generated antibodies to the HLA peptide complex, and we call these antibodies T cell receptor mimic. They mimic the T cell receptor. T cell receptor mimic monoclonal antibodies. So we were able to make T cell receptor mimic monoclonal antibodies to SVG9, the immunodominant peptide, SLF9, the subdominant, and YTM9, the recessive. So here's your peptides. And what we saw is when we infected cells, either primary cells or cell lines, the peptide that was recessive, YTM9, reached the surface of the infected cell earlier and at much higher concentrations than did these more immunogenic peptides. So what we learned is that the most immunodominant peptide, the peptide that the immune response targeted, actually reached the surface later and at lower numbers than peptides that the immune response ignored or didn't attack. So that was, we didn't expect that. We thought that this peptide would be up here and that these others would be down below. So that showed us that the timing of the peptide getting to the surface and the amount at the surface is not what the immune system is using to target the West Nile virus infected cells. Now, so this was our first thing we learned, that the immunodominant complexes are 5 times lower than subdominant recessive. It's difficult to predict what makes the peptide immunodominant, and factors other than the levels of peptide at the surface influence T cell recognition. We also wanted to know if that immunodominant SVG9 peptide could be used to make an immune therapy, a vaccine, that would protect animals from a lethal challenge with West Nile virus. So we built the SVG9 into a covalent construct that had the peptide, beta-2M light chain, and the HLA-A2 heavy chain. And we gave this into transgenic mice that were A0201 transgenic mice. We delivered this DNA vaccine in 4 mg DNA doses of 3 days, 3 times at 3-day intervals. And then we challenged the mice with a lethal dose of lethal infection with West Nile virus at 5 or 45 days. And what we saw is when we had a helper T cell epitope in the vaccine, we were able to protect these animals from a lethal challenge with the virus. So what we saw with our West Nile virus example is we were able to gather HLA from virus-infected cells, get the peptides out, identify which viral peptides are presented to the immune system, find out which peptide the immune system would focus on if you were to get a West Nile virus infection, and then we built a vaccine that protected animals from a lethal challenge with the virus. So we really took a full loop around the whole circle as we found targets, tested the targets, built the targets into a vaccine, and then used the vaccine to successfully protect animals. And along the way, we made those antibodies to peptide HLA complexes. So that's the example of a precision immune therapy for a viral neoantigen, viral peptide. Now, what Sagar did in the lab is she basically said she would like to work on melanoma. And so Sagar had also used those antibodies to stain West Nile virus infected cells. So Sagar had done the staining with the West Nile virus antigens, and she began working on melanoma. Now, with cancer, we know that there are tumor samples, and people do exome sequencing. They sequence the DNA from the tumor samples, and they identify mutations. They then try and target the immune system to those mutations in a vaccine. And melanoma is a great place if you're going to make a vaccine that targets mutations, because if you look at the mutational burden of different tumor indications, AML, breast cancer, colon cancer, et cetera, melanoma has the largest number of mutated proteins in a tumor. So if you're going to test vaccines for cancer and you want to target mutations in the cancer, melanoma is a good place to start your experiments. And so what the team we worked with at Washington University, now at University of Penn, working with Carl June, what this team did was they would identify mutations in the melanoma patient, up to 25 mutations that they thought were presented by HLA at the cell surface. So they thought any one of these 25 mutations would encode a peptide that would be presented at the surface of the melanoma cell. And what they did in this DNA construct here is they had up to 25 mutations. They also had our West Nile virus peptide SVG9 that they put into their little test vaccine. And they would use our antibody to show that this DNA construct, when they put it into a melanoma cell line, was expressed. And so what they wanted to know is, of these 25 mutations, which mutations were reaching the surface? And they could show that this peptide they knew would reach the surface, and they could stain with an antibody, or this T cell receptor mimic, to the West Nile virus peptide, and show that it was expressed. So what we did was they had selected a series of mutations from the melanoma patient, and they wanted to know which of those mutations led to a peptide, a mutated peptide that was presented. And so what we were able to do was use our mass spectrometer expertise to begin to show them, of the 25 they selected, how many of those peptides were reaching the surface. So there were 3 patients here enrolled in the study, MEL21, melanoma 38, MEL218, 3 patients that had melanoma. And we'll take a look at this patient here, MEL21. So we selected 25 different possible mutated peptides. We know that the SVG9 peptide reached the surface because we had our antibody. We could do flow to show that the construct was getting inside the cell and that this peptide was reaching the surface in HLA-A2. And what we typically found of the 25 possible peptides in this construct, we typically found 2 or 3 that were reaching the surface. For this patient, MEL21, we found a TKT was reaching the surface. We found a TMEM was reaching the surface. And we found that CDK was reaching the surface. So these other peptides, which predictive algorithms had suggested might be presented on the surface of this MEL21's tumor cells, were not getting to the surface. So what we were able to tell our collaborators was just get an immune response to these peptides that are likely found on the tumor. Don't worry about getting an immune response to these other peptides. Get an immune response to these peptides because they're found on the tumor. And so that's what they did. And the resulting thing was we could see pre-vaccine, we saw that the patient had 3 areas where the tumors were found. Post-vaccine, we saw a strong immune response to this TKT peptide here. This peptide here, we're tracking that peptide. That turned out to be the immunodominant peptide. Much like with melanoma, we had that SVG9 peptide that was immunodominant. TKT R438W mutation was immunodominant here. When we got a strong immune response to that, we actually resolved the tumors. And this was a study published in 2015 in Science, and these patients are still alive today. So this patient was cured of melanoma by making an immune response to this peptide HLA complex. So with the summary of the West Nile virus and melanoma data, we see that HLA molecules distinguish cancerous and infected cells for immune recognition via peptide presentation. HLA peptide complexes specific to disease cells can be identified with our proteomics approach. So by gathering HLA molecules from either infected cells or from tumor cells or tumor cell lines, we're able to find the peptides that are presented, and those peptides then can be used to guide immune therapies to target and kill those infected cells and those melanoma cells. So that's a pretty good success to show that we can find the peptides on the HLA molecules, test their immunogenicity, and then develop an immune therapy for cancer or virus infections. Excuse me for a second. So the last thing I wanted to talk about is these data were published in 2015, the melanoma data. The last thing I wanted to say is there's really been an explosion in HLA-based cancer immune therapies. And I just thought it's interesting now to look and see what companies and how are they being used. One therapy is to take a T cell receptor and to make a bispecific molecule. So this is the T cell receptor that recognizes a specific HLA peptide complex. Here's another molecule that recognizes CD3. And they make this into a bispecific molecule. It's called an ImmTAC. And so what these ImmTACs do is they recognize an HLA peptide complex on the tumor cell. And then this portion of the bispecific molecule recognizes CD3 on the T cell, and it brings the T cell in close proximity to the tumor. And what this company is advertising here is they're saying, look, if you're targeting a cell surface protein on the tumor, very few of the proteins inside the cell, what they say 10%, actually reach the cell surface. But HLA molecules are beautiful in that they sample peptides from virtually every protein inside the cell. So almost 100% of the proteins within the tumor cell can be targeted if you use HLA peptide complexes as your targeting moiety. And they make a T cell receptor with an anti-CD3 that brings T cells to the tumor to kill the tumor. So that's one way that people are using HLA peptide complexes to target tumors. The second way is I talked about these antibodies that we made to a peptide, SVG9 West Nile virus peptide and HLA. You can also make these to cancer peptides. And once you've done that, you can find a peptide that's on tumor cells. Here's a tumor cell. Here's a peptide from a tumor antigen that's on a tumor cell. You can make a T cell receptor mimic antibody that recognizes this. And then the other portion of this molecule, you can again make a bispecific molecule that pulls T cells to the tumor so that these T cells can better kill the tumor. Another mechanism is to take your T cell receptors that recognize a particular HLA peptide complex and transfect them or transduce them into T cells. So you can take this T cell and transduce a T cell receptor in, and people have talked about CAR Ts one of the emerging concepts in immune therapies for cancer now is called TCR-Ts, where we, rather than putting an antibody in, we put a T cell receptor in. And again, that works through HLA peptide complexes to target the cancerous cell. So you can see a host or a series of HLA, of immune therapies that target HLA peptide complexes on tumors are really starting to emerge for immune therapies. And so I'm going to conclude my talk then with, as you look at these immune therapies, and in the United States, they go through the FDA, we're beginning to see that these immune therapies require the HLA type of the patient. So our HLA typing lab has installed an FDA-approved method so that if you're enrolling patients in your clinical study, we HLA type the patient on an FDA-approved method, and that's a companion diagnostic for the immune therapy when you deliver the peptides. Another interesting thing is to consider is tumors are thought to downregulate HLA. And what we're starting to see is that people want to test the HLA expression on the tumors before they give them the immune therapy. Two other interesting things are HLA peptide targets on the patient's tumors. You saw us in the melanoma study use our proteomics methods to show that the peptides were present on the patient's tumor. That's a companion diagnostic that proteomics companies are starting to develop, in that if you want to treat a patient, you can use the mass spec and your HLA knowledge of pulling HLA off the tumor cells to make sure that the target peptide is on the tumor before you give the immune therapy. So you can see that that would be a companion diagnostic. And a final companion diagnostic is if you find that a peptide HLA target is on the tumor and you want to give that patient the immune therapy, you also can check the healthy tissues in the patient to make sure that that target isn't on their brain, their heart, or their kidney, so you have an off-target effect. So you can see now that that HLA typing for transplantation is starting now to move into other fields where you HLA type the patients for immune therapies. You can check HLA expression on tumors. And then you're starting to see these proteomics applications that I showed you here, where we find HLA peptide complexes on tumors and viruses, is also starting to emerge as possible companion diagnostics for immune therapies for cancer and virus infections. So in summary, HLA molecules present peptides that distinguish cancerous and infected cells. You can use proteomics methods to identify those peptides on disease cells. These peptide targets transition into T cell eliciting immune therapies. And I gave you an example of some of the ways that different groups, companies, and university labs are developing these things. There's a whole suite of methods now arising that people are using to go after the HLA peptide complexes on tumors. And finally, I gave you an example of a companion diagnostics that are going along with these immune therapies for cancer. And then there's been a number of people in my lab. Curtis has been a real leader in the mass spec. Sagar has done a great job with bringing HLA on the tumors. Just a number of people. Ted Hansen and Mike Diamond made the West Nile virus vaccine. John Wydance made the T cell receptor mimic antibodies. This team did the West Nile virus work, and Beatrice Carino and Jerry Lynette did all the work. They're geniuses, and the work they do on targeting melanoma. And that's really my entire talk. Thank you for your attention.
Great. Thank you very much, William. That's a fantastic talk. My understanding is that we're going to have questions and discussion at the end of the session. So everyone can get their questions into the chat area. And I'll hand over to Varsha now to introduce the next speaker.
Everyone. Hello, I will take an opportunity to, this is the last day of our conference. So I would like to thank all the speakers and delegates and moderators to just make this conference successful. As a joint organizing secretary, I would like to thank everyone. And my best wishes to Dr. Jaiswal and team for the next meeting. Now moving to the next speaker is, can I have a slide, please? The topic is non-HLA antibody panel discovery, and development of validation from bench to bedside. So this lecture will be delivered by the Dr. Carrie Butler. Is an assistant director at the UCLA Immunogenetics Center and an assistant professor in the Department of the Pathology and Laboratory Medicine. Dr. Butler's current research interest includes the identification and validation of the novel non-HLA antibodies involved in solid organ transplant rejection. The rejection in HLA matched donor and the discovery that antibodies directed against the protein expressed by the endothelial cells are associated with an increased risk of the transplant rejection. Emphasize the importance of the further characterization of the non-HLA antibodies in allograft rejection. She completed her PhD from the South Florida University and did her postdoctoral study at UCLA. And she, in her postdoctoral time period, she did work on the non-HLA antibody in the solid organ allograft rejection. Over to you, Dr. Carey.
Hi, I'm Carrie Beller. I'm from UCLA. I'm an assistant director and assistant professor at UCLA. And today I'm going to tell you about our project, non-HLA antibody panel discovery, development, and validation from bench to bedside. And I want to take a moment to thank Ishi and Aphia Con for giving me this opportunity to share our research with you. So the first slide is just kind of an overall study design. So this study was done in renal transplant cohorts. You can see here there are 4 main categories for the study design. And the overall aim was to identify and validate novel non-HLA antibodies that may be a marker of or contribute to renal allograft rejection. So in the first far left panel, you can see the first goal was to screen patients for endothelial cell antibodies. And we did this by looking at 69 renal allograft recipients with rejection. And we used an endothelial cell crossmatch, or ECXM, to screen these patients to identify those patients that expressed antibodies that bound to the endothelial cells. And with this, we identified 16 patients that had both rejection and were positive by this ECXM, indicating that they had these non-HLA antibodies that bound endothelial cells. So from this, 16 patients, we ran them on protein microarrays to identify the targets of the antibodies. We ran these both at a neat and eluted off of these endothelial cells in order to concentrate the antibodies that were binding to the endothelium cell. And then we also ran 3 no rejection technical controls. And these were from the patients that had, 3 of the patients out of the 16 that had a pre-transplant sample that was available. So from that, we ran these on 35 protein microarrays. They contain over 9,000 human proteins, and this allowed us to identify the targets of the non-HLA antibodies. We used prospector analysis, looking for those antigens that were increased at 1.5-fold change with a p-value less than p.05. And we identified 386 using this approach. So from this, we went on to further prioritize. And so we prioritized both by frequency. We wanted to find non-HLA antibodies that were coming up in more than one of our patients, expression in the kidney and endothelial cells and on the membrane of the endothelial cells. And with this kind of prioritization strategy, we identified 12 novel non-HLA antibodies. With these 12 novel non-HLA antibodies that we identified, we combined them with 67 previously described in the literature to work with Aphia Con and develop this panel of 79 non-HLA antibodies. And then from here, we went on and tested it in 2 independent single center study cohorts, an adult cohort of 160 patients and a pediatric cohort of 62 patients. So here you can see this is the discovery cohort demographics. It includes all 16 patients have rejection. And they also have endothelial cell antibodies identified by a positive endothelial cell cross-match. You can see it included both pediatric and adult renal transplant patients. The majority were male, Caucasian, and the majority of them were primary transplant patients, although a few of them were retransplants. And you can see the other demographics here. So again, with these discovery cohort, we screened protein microarrays with 9,000 human antigens. And you can see with the 16 neat over on the left in blue, we identified 1,252 targets. And with the eluted serum, we identified 388. Overlapping, there were 386 antigens that overlapped in both the neat and eluted. And then we excluded 391 non-HLA antibodies that were identified in the pre-transplant serums. So this identified 135 non-HLA antibodies, and from that we prioritized the 12 antigens. And then at the very bottom, you can see we added in the 67 antigens identified by literature search with ImmuCore's help. And there are 79 antigens to be verified in downstream analysis. So the gene ontology of the 135 that were identified, you can see in purple are the biological processes. Primarily these were indicating a response to stimulus or signal transduction. And in blue, you can see the molecular function, primarily signal transducer activity. So with the prioritization of the non-HLA antibodies, we went by frequency. We looked for ones that occurred in more than one patient. We looked for ones that were expressed in the kidney, known expression in endothelial cells, and prioritized those on the membrane. And then we identified 12 non-HLA antibodies to be added to the ImmuCore Luminix panel. On the bottom left, you can see the frequency and the different eluted sera out of the 16, and the number of protein microarrays there on the y-axis. In blue, you can see there were 2 growth factors. The primarily in purple, there were 5 of them that were involved with signaling. There was also cell adhesion and cytokine factors out of the 12. And then on the bottom right, you can see the mRNA expression. All 12 of them are expressed in the kidney and expressed in endothelial cells using publicly available data. So this is just a longitudinal pediatric renal cohort, and to kind of go into the demographics of the cohort. So again, there were 62 patients, 34 were classified as no rejection, and 28 were classified as having a rejection. And there's no significant difference between the two. So the strength of this cohort was that it is a longitudinal. It had both for-cause and protocol biopsies and sampling. So we went with every sample that we had a paired serum with a paired biopsy, as well as like a pre-transplant serum. So for these, we mainly have a pre-transplant, a 6-month, a 1-year time point, a 2-year time point, and then anytime there was a biopsy for suspected rejection. So with this, there were 233 blood samples, 129 blood samples with matching biopsies. You can look at them broken down by the different time points and no rejection or rejection. And so these were screened for HLA and non-HLA antibodies. So with this approach, looking at this pediatric cohort, we identified 15 non-HLA antibodies that were associated with renal allograft rejection. They're shown here. In blue are the ones that are newly described, either in our previously published cardiac paper or in this renal paper first, and then all of them here were identified in the pediatric renal cohort. And you can see the hazard ratio is down on the x-axis. It's a forest plot, and the bars indicate a 95% confidence interval. Here we're looking at the pediatric patients without rejection and those with rejection. And this is a correlation matrix of these non-HLA antibodies and how they correlate with each other. And so you can see between those patients that have no rejection and those patients that do have rejection, that the antibodies and how they're associating with each other are different. Here is a classification and regression tree analysis. This is used to further classify those patients with rejection and no rejection. And it's just using, we put in all 15 non-HLA antibodies that were associated with rejection and use this classification and regression tree analysis in order to further classify. You can see in the further right-hand corner, bottom corner, those all in black are those patients with rejections that are classified, and the decision tree that got them to be, completely classified as rejection. And then in the all white boxes on the far left, and then 4 boxes over from the far left, those would be patients that have no rejection, and the decision tree analysis in order to identify those patients with no rejection. And then you have some patients that are kind of falling in between. So here's kind of a longitudinal analysis. Like I was saying, this cohort has a strength of having these longitudinal samples. And so one thing we looked at that I'm not showing here is, the non-HLA antibodies in both the non-rejectors and the rejectors over time at all the different time points. But then we also wanted to look at how do these change when you go from a biopsy that's negative for rejection and it goes to a positive for rejection, and then vice versa. Those patients that had a positive biopsy for rejection that went to negative. And so here on the x-axis, you can see the 15 non-HLA antibodies associated with rejection. Then you can see for each one of these, it is the patients, there's 7 of them that went from having a positive biopsy to a negative, and then 8 who went from having a negative biopsy to a positive. And you can see those patients that had went from negative to a positive, they had an increased fold change in their MFI levels of these non-HLA antibodies. And conversely, when they went from a positive to a negative, they had a negative fold change, indicating that the antibodies are going down. Next, we wanted to look at the association with the graft pathology. Here, out of the 15, what we did is we did an analysis removing any HLA-DSA as a confounder, and then associated these non-HLA antibodies that were associated with rejection in this cohort with other scores of graft pathology. So here you can see these non-HLA antibodies are associated with arteriolar hyalinosis thickening, or AH. And then more excitingly, you can see that 4 out of the 15 non-HLA antibodies associated with rejection were independently associated with IFTA and A. And even more exciting, CSF2, one of the non-HLA antibodies that was identified, was associated with both acute peritubular capillaritis, or PTC, and acute intimal arteritis after removing samples with confounding HLA-DSA. And these 2 are known to be involved with rejection and indicators of the pathology. So this might be able to help hint at what kind of mechanisms are involved, or indicate non-HLA antibodies that are more involved in maybe a potentially mechanism leading to the graft pathology versus those that are maybe like bystanders. Next, we went on to look at this these non-HLA antibody panel and an independent cohort of adult renal transplant recipients. And so here you can see there was 160 patients. These include 90 patients with no rejection and 70 patients with rejection. Again, no significant difference between the 2. And in this independent cohort, we identified these 3 non-HLA antibodies in the top A panel that were significantly associated with rejection. And then the blue is indicating the novel SHIC3 identified. And then in B, you can see here is another classification and regression tree analysis, much smaller here because there was only the 3 that were identified in the adult renal cohort. But you can see here that this classification and regression tree analysis is using 2 of the non-HLA antibodies identified out of the 3 in order to further classify. So in summary, the post-transplant neat and endothelial cell eluded sera from 16 EC cross-match positive, rejection positive patients, as well as 3 available pre-transplant sera were analyzed by protein microarray and identified EC antigens bound by patient sera. The protein microanalysis of these EC cross-match positive and rejection positive sera identified 391 proteins in the pre-transplant sera, 1,252 proteins in the neat sera, and 388 proteins in the EC eluded sera. And 135 out of these, out of the 388 eluded sera of the EC cross-match positive, rejection positive samples were detected in post-transplant sera, and indicating that they may be de novo non-HLA antibodies associated with rejection. We prioritized 12 novel antibodies to select for proteins with EC membrane localization, expression in the kidneys, and antibodies that were seen in more than 1 patient. And with this approach, we identified 15 non-HLA antibodies significantly associated with renal allograft rejection in a pediatric renal single center cohort, and 3 non-HLA antibodies significantly associated with renal allograft rejection in an adult renal single center cohort. So with this, we concluded that a multiplex bead array assessment of non-HLA antibodies may identify renal transplant recipients at risk of rejection. This concludes my talk, and I look forward to the live discussion later. Thank you.
Thank you, Dr. Carey. It was a very interesting talk and great talk. Now session is open for the questions. You can send your question in the chat box. There are a few questions I can see.
Can I just begin by asking a question from William? William, I enjoyed your talk, and my question is related to the immune hierarchy of the West Nile virus ligands. And you said the immunodominance and the surface expression, and I saw that the more immunodominant peptide had a lower surface expression than the other. You know, immunologically speaking, we would think that the immunodominant peptide would have higher expression and therefore would generate. So what is the thinking behind this? Is it the peptide size that we are talking about, or the location of the peptide?
Well, I'm glad it's late on a Friday afternoon because I can speculate. I'm late on a Friday afternoon, late on a Friday evening. We never resolved the question. There are several lines of thought. One is that the self-peptides that select for T cells provided T cell precursors that had a prevalence, or no, that had a predilection to go after that peptide. What I think it is, based on what we do, is when we search these peptides that we find, these viral peptides, and we search them, and we search them against human proteins, it's as if the peptide has to be similar enough to a host protein that the T cell recognizes it, with a few key differences. And there's a scientist, Brian Baker at the University of Notre Dame, that studies this. And he got his PhD in Don Wiley's group. And it seems as though there's a couple of positions in the peptide, 4, 5, and 6, that if you have the right amino acids, where the CDR3 region of the T cell receptor interacts with the peptide, those peptides seem to be key. So it's as if the peptide has enough, have to have, much like when antibodies recognize HLA proteins, that the peptide has to have enough similarity to other peptides that the T cell receptor recognizes it. But a couple of key differences seem to be stimulate a strong immune response. If the peptide is too dissimilar, it's as if the T cell receptors don't go after it. And if it doesn't have the right differences in the right places, it doesn't seem like the T cell receptors acknowledge those differences. That's my interpretation of the data. And my interpretation's incomplete at this point.
So I mean, HLA molecule or the HLA phenotype of the individual, we know for a long time, HLA phenotype of the individual would actually determine the avidity of the peptide. And therefore, many of us are exposed to the MTB, for example, the antigen, but only a few of us go on to develop the disease there. And that would have something to do with the HLA phenotype of the individual. So it's the peptide on one hand, is the phenotype of the HLA phenotype of the individual that is more important.
I don't know if I'm answering your question directly. I'm probably trying to twist your question around so I can answer it. But what I would say is that if you have B35, there's peptides presented by West Nile virus peptides presented by B35 that are, then you will ignore the A2 peptide and you will chase the B35 peptide. So there are, if you have the right haplotype of HLA molecules, if you have the right grouping linkage disequilibrium of HLA molecules, you will chase other HLA peptide complexes such as B35 will present a dominant peptide. I don't think that answers your question. That's probably the closest I can get to answering your question.
Okay.
Yeah. I'll just read a couple of questions out from the chat box from the audience, if I can. From Gaurav Sharma, we got, can you please share your views, William, on the cross-reactivity of the YTM9 TCR specific antibodies? And do you think the observed heightened immunodominant response to this peptide could partially involve cross-reactivity?
These antibodies are very challenging to elicit and to make. And so we don't get very many of them. So the few that we do get are well quality controlled. So we run in numerous assays, like we stain uninfected cells and we see no staining. We pulse with synthetic peptides from all the West Nile virus peptides, and we test to see if the TCR, these antibodies, recognize those, and they seem specific. One final thing we've actually done is we make a column with the antibodies. So we take the antibodies and we stick them to beads or a column support, and then we run our soluble HLA. We produce, remember how we secrete that soluble HLA, we run it over and we look to see if it's specific, and if it's cross-reactive, we can identify what peptides it cross-reacts with. And so with these three antibodies, we don't get too many of the antibodies, but these three antibodies seem to be highly specific for the peptide HLA complex. So I don't think that it's cross-reactivity that's giving us those data.
And one from Rajalingam for you, William. What drives the low expression of immunodominant peptide SVG9 laden HLA-A2 expression on the cell surface?
Yeah, Rajalingam always asks me a good question. So many people, when they select an antigen that's going to provide peptides, they'll use RNA expression. And what Tony Purcell and other groups have seen is the level of RNA and the level of protein does not indicate how well a peptide is going to be excised from a protein so that HLA can bind and present it. All of these peptides bind with similar affinity. We did a binding assay and tested their binding. They all bind well. It just seems to be, there's an immunoproteasome, there's trimming by ERAPs. There's a number of things that contribute that we don't fully understand. And so level of protein, the level of RNA, those don't correlate with how well a peptide is created and made available to HLA. And so that's what we do, is we just get HLA and look to see what's presented. We don't try and anticipate, because it remains a bit of it's exciting, but it remains questions as to, if you have equal amounts of a protein, why is one peptide created better than another? It might be cleavage, processing, transport. Rajalingam, I just don't know the answer to that. I think once we do, I'll be put out of business, because we'll be able to predict. But fortunately, before I retire, hopefully we don't answer your question.
Okay, one more question from Raj to Kerry. Is there any correlation between endothelial cell crossmatch and non-HLA antibodies by LIMNEX? So Dr. Carey, is she around?
Not sure Carrie's online, is she?
No, she's not online.
No.
No? Okay.
Does anyone else know if there's a correlation between endothelial cell cross-match and non-HLA antibodies?
I think she didn't log in. They only played the video that she sent.
Sure.
Okay.
We do have another one for William from Dr. Monica. Any comment on the HLA locus stroke antigen preferentially expressed on the tumor tissue?
I'm not sure I fully understand the question. We selected, many researchers select A2 because of its high frequency. And so we know that with tumors will upregulate and downregulate different HLA. As far as the tumors go, why one antigen is preferentially expressed and another isn't, I think that goes back to Rajalingam's question, in that we just don't, people try, many people, and we're familiar with this, everybody on this call, everybody at this meeting is familiar with it. If people can get away with dealing with the HLA community, if they can avoid working with us, they would love to avoid working with us. HLA is complex, hard to understand. It's not, it's not something you dabble with. You have to be serious about it. If people could predict based on how much antigen is there, the nature of the mutation, how well a peptide binds, they wouldn't need HLA people to do the work. But we just at this point are unable to ascertain what peptides are going to be made available by HLA, probably because the intracellular complexities of the tumor cell in this question are just not fully understood. We'll see a peptide from an antigen presented on a tumor cell. That same antigen is present in healthy cells, but that peptide is not there. So we could see a protein in a healthy cell and a protein in a tumor cell, and only in the tumor cells do we see a peptide from that protein. So we just don't understand the complexities, especially of what changes when a cell becomes neoplastic.
William, can I ask you on your component that you talked about, the HLA companion diagnostics for immuno-oncology? And we know that a lot of solid tumors and even melanoma, they lead to downregulation of the HLA molecule on their surface. So are you suggesting that the HLA phenotype of the individual would actually determine the success of immuno-oncology? Immunotherapy, I mean?
Yeah, I'm suggesting two things. One thing is that the number of possible targets available to someone developing an immune therapy is increasing. So we're starting to learn about more and more peptide targets on more and more HLA that might be used to target a tumor. And at the same time, we realize what you said is it's possible that perhaps an HLA molecule A24 on tumors is downregulated, or on particular tumors is downregulated. So if you have peptides that could work in A24, A2, and B7, and you see that a patient's A24 is downregulated, you might want to use the A2 or the B7 therapy. So we're starting to see more and more tools and more peptides and more receptors emerge. And I think if you want to see an immune therapy work, but the HLA is missing, that might preclude using that immune therapy. We're certainly using HLA typing to say, does this patient have HLA-A2 so we can provide an HLA-A2 specific immune therapy? And I'm just starting to wonder is, do we want to study whether or not a tumor has downregulated HLA expression, usually at a locus? Usually you don't see whole haplotypes gone, or we don't. But sometimes you see A2 go up and down, and sometimes you see other HLA go up and down on tumors. Is this something that can be used as a guide? And this was a question. I really just put this out. I thought it would be interesting to discuss, and I'm glad you asked.
In continuation of that, I just wanted a very small, now that there are a lot of trials going to be done or being done now, they have gone to phase 1 trials for CAR T cells or CAR NK cells for immunotherapy of various kinds of solid tumors, melanoma, and glioblastoma, and things of that sort. And you think that there again the HLA phenotype of the individual would determine the success of this CAR T cell or CAR NK cell therapy?
Well, as I raised that question, I don't know if I have the answer for that, but if you want these immune therapies, as the tools become more and more numerous, if you know that a tumor has downregulated HLA-A2, would you attempt to use an A2 specific immune therapy on that tumor? Especially if you had in your toolkit something that might target another HLA. So I told someone I'm going to put that into my presentation this evening just to see what kind of response I get from this audience to see if it's something that they would consider. That one was really one I just don't know about. I know that the proteomics, I'm talking to companies and they're asking me about, is the target on the healthy tissue, is the target on the tumor, is the right HLA there? So I know that those companion diagnostics are things that people are asking me about. But I just started to wonder when I was preparing this presentation, I wonder if people would ever want to check the levels of HLA expression as a companion diagnostic. Would that guide your immune therapy? And I really didn't know the answer, but I'm kind of glad you asked because it shows that, it stimulates thought in the area.
Well, sounds very interesting to me anyway. Yeah.
I think we've got a question from Uma, and then after that, Rajalingam also has a question for William. I think Uma's on mute, I think.
Okay. So in cancers, there is loss of expression. So I was also wanting to, you already discussed a bit on that. So when we lose the expression on the cancer cells of the HLA, so then how these interactions are impacted. We discussed.
Well, that's an interesting comment. And this is something I would encourage people to go back to the literature and read the manuscripts where they see a loss of HLA on tumors. Because many people on this call are serologists. They study HLA serology. If you look at how the people ascertained the loss of HLA, very few to no studies actually took antibodies to HLA and stained the tumor cells. So there's very little work done. Our data, when we extract HLA off of tumors, it's always higher than the corresponding healthy tissue. So if we take a healthy colon and a cancerous colon, we always find more HLA on the cancerous colon. If we look at a healthy lung and lung cancer, we find more HLA on the cancerous lung. If we look at ovarian cancer, we find more HLA on the cancerous on ovarian cancer. So our data is suggesting that it's not as common as the literature indicates. And I believe that if this community here, especially, were to go back and read the literature, you would start to ask questions. That being said, we do see it decrease, but not nearly as frequently as what the literature suggests. So we see HLA drop, but I'd say on 1 of every 20 or 30 tumors, not nearly as frequently as we would have thought we were going to see it. So when we do see it drop, we don't see all the HLA drop. If we use W632 and we pull out all class 1 HLA, we'll see a particular molecule drop, but we seldom see a whole haplotype or both alleles of the locus drop. So I think it happens. I don't think it happens as frequently as the literature suggests. And I would say that if you went and looked at those manuscripts, you might, as a group that does HLA serology, start to question those data a little bit more. And it happens at a lower frequency, I think, than what people think.
Thanks, William. Thanks. Raj has a question, I think.
Willie, thank you. I mean, it's always your talk is very stimulating. So the low expression that triggers more immune response is absolutely interesting. So in transplant, we know the de novo DSA is always DQ, which is considered to be the low expression allele. So it's just a comment, it's not a question. So it's also the CW7 seems to be low expressed. and it triggers more antibodies. So I think the expression is very important. I don't know how to explain that, why the low expression allele triggers more immune response, not only the T cell, but also the antibody response.
Yeah, I agree with you on that. You know, the T cell receptors and the antibodies are so exquisitely sensitive that you don't need a lot of peptide or HLA or HLA peptide complex for a T cell or an antibody to recognize something. So it's probably more the differences in similarity to self, because B cell receptors and T cell receptors are so sensitive. You need one or two peptides on a tumor for a T cell to be able to see it. You need one or two peptides on a virus infected cell for a T cell to be able to mount a response. I mean, Mark Davis and many others have done so much work on that to show that how exquisitely sensitive those receptors are. So the levels of expression, while they are lower, I agree with you. Boy, they sure fall within the range of what a B cell and T cell can see.
Very quickly, you know, when you say the HLA class 1 expression is low, immediately I'm thinking about the NK cells. You know, so you talked about the A2s, maybe the B or C locus loaded peptides, you know, when expressed low levels, the NK cell probably is playing complementary role.
Yeah,
Thanks,
that's a
Billy.
good point. That's a great point.
There is always a surprise in immunology. If I wear the immunologist hat, you know, if a low expression peptide, I would say it should generate a lower immune response, but here we see a higher immune response. So it's not only the peptide expression, but also the LEL and the HEL that Rajalingam is talking about that brings about the final immune response. Yeah.
Okay, so there is no more question. Sorry about we were not able to discuss about the non-HLA antibody because non-availability of speaker. And now we close this session and move on to the next session. Thank you very much.
A very informative session indeed. On the behalf of the organizing committee, I would like to thank all the moderators and speakers for conducting this session very smoothly. So there will be a short break for now for 10 minutes, and we will meet after the break for virtual poster presentations. Thank you.
Are you still there, William? No.
Good morning. Can you hear me? Good morning, everyone. Can you hear me?
Yes, ma'am. You're audible.
Can you hear me correctly and
Yeah, yeah,
clearly?
yeah. Yes, ma'am. Yes, ma'am.
Okay. Thank you very much.
Thank you.
Hello. I see Sally, you're online. We have all the moderators here.
Yes, yes, Uma. I'm there. Good morning.
Too
How are
early
you?
for you. Thank you for joining.
Preparing my pen and everything for the scoring sheet.
That's really nice of you. You have
And
all
then
the abstracts already? You got all the abstracts?
yes, I got the abstracts and I was talking to Dr. Anil and Handu, and he's telling that we divide the posters into 5, 5, and 4. So I'll be moderating the first 5.
Okay.
And
so the last poster is not going to be presented. The delegate is not available.
Just a minute. Just a minute. Can everybody please silence just a minute? Dr. Sally?
Okay.
Dr. Sally?
There. Can you hear me?
Dr. Sally, please close down your platform, please. Only keep the Zoom. You have login from two platforms.
Really? No.
So close the browser one and keep the Zoom one.
Okay, wait a second. Like that? Did this do the idea? Did this work?
Sally, there are two platforms. One is on Zoom, where you can appear on the screen like you are now. And Manas thinks that you have also joined from the other one as well. And when you join from two, there could be some kind of an echo. And that's what he's referring.
I can't find the other one. I can't find the other one. On my laptop, there is no other one. And
so that means you're from Zoom. So Manas, she's already from Zoom only. That's okay.
She sounds fine.
Yeah.
Can you is hear an echo from my side?
Now there's
Not
no
much.
echo. Now it is okay.
Yeah.
Okay.
So how many posters are there today?
So there are 14.
14.
It will be 13. I think one candidate is not presenting the last one.
How are you going to do it? Are you going to give them?
So, sir, I thought we will divide between three of us because I, Dr. Sally and Dr. Monica are there. So Sally starts first for first 5, next 5 Dr. Monica, and last 4 I. That's how we'll moderate between us. And we are expecting to give everybody about 2 to 3 minutes or so to explain what they have done and maybe a question or two.
Oh, they should only, I mean, we have already seen their title. They should only give us a
Brief gist.
rationale of their study, the objectives and the outcome. That's all.
Absolutely, sir.
Yeah.
Dr. Handu, there are 13, so you can do.
So we can go 4, 4, 4, 4, maybe 4, 4, 5 or whatever. 5, 4, 4. 5, 4, 4 would be right. I think Sally can take first 5 and then Dr. Monica can take next 4 and I'll take the last 4.
No problem.
Thank you.
Glad to see all of you. It's really early here in the morning.
Anil Hando is technically right. He wants to prove ladies first.
Morning. Good morning, all.
Good morning, ma'am.
Good morning, sir. Good morning, Dr. Uma.
Morning. Sally, it's 5 o'clock for you or 6 o'clock?
Yes.
Very early. Thank you for joining.
Good morning, Sally.
Good morning, everyone. Dr. Mehra, Dr. Anil, Dr. Monica.
What part of the world is Sally from?
Egypt, sir.
Egypt.
Oh, Cairo. Are you in Cairo?
Yes.
Oh, the famous, what do you call the dunes or what?
Pyramids.
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 Pyramids. And I've been there, and there was a restaurant very nearby, and there was a lot of dancing going on.
Yes.
Fantastic.
I
was returning from London many years ago, and there was a problem in the flight, and they had to land in Cairo. So that gave us a full day and one evening to spend in Cairo. So we were on the camel and going through the pyramids. Yeah.
Can we ask Akansha to begin?
Similar pyramids are in Mexico.
Yes, but not that great like ours.
Oh, of course. Not that great, yes.
They are not that old.
Yes. Absolutely.
Akansha, are you there?
Yes, ma'am.
So shall we begin, sir?
हाँ हो गया ना 10 मिनट का टाइम
10 minutes हो गए Akansha, I think. Another 1 or 2 minutes maybe, just.
कर दो जी.
Technical team, can you check all the presenters are available?
So actually, Anil, we have more time. हमने 9:15 से 11 दिया हुआ है. So even if 9:30 बजे, तो भी हमारे पास 1.5 घंटा रहेगा. 90 minutes.
Yeah.
We will have at least 90 minutes. So these are 14 people, and even if we say 2 minutes for discussion each, so we have good time. That's okay. If you finish,
So we have roughly about 5 minutes per head. So if we give 3 minutes for they explaining the whole thing, and then 2 minutes for question answer, that should be good.
That's what I was thinking. So even if it is total of 5 minutes, so 14 plus 5, 70 minutes, maybe 10 minutes here and there. So that's fine. It's okay.
Yes, absolutely.
Yeah, but make sure that you don't go beyond 5 minutes for any of these.
Absolutely.
So will the technical team put the timer for the moderators?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
Thank you. So Dr. Hando and Dr. Sally and Monica, there will be a timer. So you can.
Oh, sure.
Okay.
Akansha?
Yes, ma'am. Welcome back, everyone. Now there will be a poster presentation and discussion. As it's in virtual mode, the organizers have arranged an opportunity for brief presentation for all the participants. The session will be moderated by Dr. Anil Handu, Dr. Sari Elfishavi, and Dr. Monica Jain. Dr. Handu is a Senior Director of Hospital Lab Services and Academic Affairs, Research and Continuing Education, Max Healthcare Institute Limited. Dr. Sally is an Associate Professor of Clinical Pathology, BMT Lab Unit, National Cancer Institute, Cairo University, Egypt. She is also the Vice President of Arabian Society of Histocompatibility and Immunogenomics. Dr. Monica Jain is a Senior Consultant in Institute of Medical Genetics and Genomics, Sir Gangaram Hospital, India. I'm sure all the delegates will enjoy the opportunity. Best wishes to all. Thank you.
Thank you.
Thank you.
Thank you. Thank you, Akanksha. I request Dr. Sally to take it forward for the first 5 posters.
Good day, everyone. It's glad to be here. I want to present our first poster for this session. The frequency of HLA alleles by next generation sequencing method among lung transplant recipients with interstitial lung disease and post-COVID lung fibrosis, single center report in South India. Would you please, Dr. Wawet Ayapu, sorry for the pronunciation, go ahead with your poster presentation.
Yeah,
please
good morning,
don't exceed
everyone.
3 minutes, as much as possible. Go ahead, sir.
Share screen. Share screen. Oh, good morning, everyone. So to start with my topic, so lung transplantation has now become a suitable option for people suffering with end-stage lung disease to retain their optimal lung functioning and to improve their quality of life and survival, as a result of which lung transplantations are continuously increasing across the globe, including in countries like India. So the aim of this study is to determine the prevalence of HLA alleles by high-resolution HLA typing by next-generation sequencing among lung transplant recipients. So coming to the materials and methods, a total of 50 lung transplant recipients were selected, comprising of 34 patients with interstitial lung disease and 16 patients with post-COVID-19 lung fibrosis. So as mentioned earlier, NGS remains to be the cornerstone of this study. And briefly, next generation sequencing consists of 3 steps, namely nucleic acid extraction, library preparation, followed by sequencing and analysis. The data was analyzed by using various bioinformatics, and the data was looked for multiple quality metrics like read depth, noise, allele balance, Q30 score, etc. At the end of the procedure, the results that were obtained showed some clear trends. Firstly, there was a clear male preponderance, and the most affected age group happened to be between 51 and 70 years, and the frequency seemed to be increasing with age. To further break down the results, the most predominant HLA class 1 allele frequencies in 34 ILD patients were A11, B40, C07 with frequencies ranging between 13.2 to 25%. Similarly, the most frequent class 1 alleles in post-COVID lung fibrosis were A02 with 15.6% and B15 and C03 with 12.5% each. Now coming to the class 2 HLA alleles, the most prevalent HLA alleles in ILD patients were DRB107, DQB102, DPB102 with frequencies ranging between 13.2 to 26%. And finally, the most prevalent HLA alleles in post-COVID lung fibrosis patients were DRB107, DQB103, and DPB102 with frequencies ranging between 12.5 to 31.2%, with 31.2% being the highest value of all. So to finally summarize the discussion and results, the most frequent HLA allele in post-COVID-19 lung fibrosis patients was DQB1030201 with a staggering 31.2%. And the most frequent HLA allele in the 34 ILD patients was DPB1020101 with a 26%. At this juncture, I'd also like to mention that not many studies have been conducted in India on frequency of HLA alleles, and this study happens to be the first of its kind. So in conclusion, the class 2 HLA alleles like DPB1020101 was found to be more prevalent among ILD patients, and DQB1030201 among post-COVID-19 lung fibrosis patients, suggesting that increased frequencies of HLA2 alleles may contribute to identify potential markers of susceptibility to ILD and post-COVID-19 lung fibrosis. thank you. I thank you all, and I thank Ishi for providing me with this opportunity. Thank you.
Thank you, Dr. Rowett. Any questions or discussions to Dr. Rowett for this poster?
So can I just make a comment? The claim that there are no other studies at all which have looked at the gene frequencies or the HLA frequencies may not really be true, because there are studies from Hyderabad itself, kidney transplant patients, and of course some studies from Ahmedabad and Delhi as well. Just a passing comment on this.
I also wanted to say, young man, it is very nice to hear you, but there are any number of studies done on NGS base. They may not be in print as yet. Anyway, that's a separate point. So, I want you to be careful anytime. A lot of us are very keen to say, I'm the first. Anyway, I wanted to know, you're saying DQ beta 1 302 was with a very high frequency. What does this DQ 302 encode in the DRB1?
Can you please repeat the question?
302 encodes a few DRB1 alleles. Which are those, and what were their frequency in your study?
DR alleles were 070101 and
No. You are saying 302. So 302 would be with DR4 or other. What was the frequency of DR4?
DR4.
Yeah. DRB1.
DR4 was DRB1 04 was about 6.2%, sir.
And what was the frequency of 302? DQB1 302?
It was 31.2%. It happens to be the most frequent, highest of all
Where does this
among
come
the lung transplant recipients.
Yeah, but where does this come from? 302, the DQ molecules are very close to DR. So they will be encoding some DR molecule. So you have to justify the 31% 302 with the similar frequency of some DRB1 alleles that are encoded in this.
Okay,
Do you follow my point?
DR was 12.5%.
The highest, the closest value was 12.5% to DQB. That was the closest value.
No, Dr. Rohit, what he's asking is the DQB or 302, what is the DRB1 linkage? Which allele express the DRB1, DQB or 302?
I was using the word encoding, but you can even talk about linkage disequilibrium. There's a very strong linkage disequilibrium between DRB1 alleles and DQB1. Anyway, that's fine. Okay, your time is up. So please go back and have a look at it. Okay?
Yes, sir. Yes, sir. Thank you.
Thank you.
Thank you for your poster. We will now proceed to the next poster presenter, DRB1, DQB1, DQ alpha 1 haplotype analysis in random population from Mumbai. Suchita, Dr. Suchita Jugail from Mumbai, India. Would you please present your poster?
Good morning, everyone.
We can see you and hear you. You can share your screen now.
Yeah. Good morning. I'm presenting a data for haplotype analysis of DR beta 1, DQ beta 1, and DQ alpha 1 antigens in random population from tertiary care hospital of Mumbai. So, human leukocyte antigens are cell surface glycoproteins encoded by a major histocompatibility complex, and it is located on the short arm of chromosome 6. It is known to be the most polymorphic genetic system in humans. And this is clinically important as transplantation antigens, as well as in population genetic studies and disease association studies. These are closely linked genes and and the entire MHC is inherited as an HLA haplotype. And certain haplotypes are found more frequent in some populations than expected by chance. So we have analyzed the DR beta 1, DQ beta 1, and DQ alpha 1 haplotype frequencies in our population. This is a study carried out between the period of 2017 to 2020. Total of 321 individuals were analyzed. The HLA typing was performed by PCR SSOP method. In 321 individuals, total of 642 haplotypes were analyzed. Total of 57 different haplotypes were identified in the 321 individuals. And these are the most common haplotypes which were observed. The most common one was the DR1502, DQ beta 10601, and DQ alpha 0103, followed by DR beta 10701, DQ beta 10202, and DQ alpha 10201. So to conclude, our analysis indicates certain alleles of the MHC class 2 region exhibit strong positive linkage disequilibrium. And this information can be useful in the population-based genetic studies, in disease association studies, and even in the identification or selection strategy. It can be used as a tool for selection strategies for hematopoietic stem cell transplantation donors. In presence of anti-HLA antibodies and unavailability of that specific donor antigen status, this tool can be used to assess whether it is donor-specific antibodies or not. Thank you very much. These are some references. Thanks.
Thank you very much. Is there any questions for Dr. Sushita?
Can I ask one?
Yes, go ahead.
Suchita, when you thank you for your presentation. When you mentioned random population from Mumbai, so what do you mean by random population? Usually
it's a patient population coming to Mumbai for organ transplantation testing. So it is a mixed population, like some from north, most are from Western India. So it's a mixed population. So that's why we have used the word random population.
What she's asking is, have you only, this data is based on only the healthy donors or also of the patients?
Patients also, so yes.
Patients
from
also.
Patient and healthy donors also.
Both of them. So if there
both
is
are included, yeah, both are included in the study.
Listen to my question. If it's a brother, sister, or a father, son as the patient and the donor,
yeah.
Did you take them both?
Yes.
Yeah. Okay. I mean, that's okay to derive a haplotype, but the other thing that you need to do when you say that these are the most common, you have to use another statistical method to calculate the delta value.
Yeah. Oh, yes.
Are you aware of that? Are you aware
no.
of the delta?
No, sir.
I think you should look into delta, because that will tell you which is the most common haplotype.
Okay, sure.
And that gives the information on the linkage disequilibrium.
Yeah.
And may I add something, if you please? For a sample to call it random, you have to have either a software or a randomization tool to pick your sample size. So there is a simple tool like flipping the coin, or just there is a more sophisticated sheet-like computer-based randomization programs to select the random sample out of your either donors or patients. So I would recommend using something like that before using your sample and calling it random, because it really randomizes the sample collection and avoids any bias in the statistics forwards.
Yeah.
And they should be unrelated, I think. They should be unrelated when you're saying population.
Of course.
Yeah.
Not family members.
Wait.
There's one question by Dr. Rajalingam. Can you take that, Dr. Sally, please? It's in the chat box.
Sure. Yeah, Justin, I know you didn't test for the DR beta 3. So whether this DR7 and DQ9 haplotypes is carrying this DR53 null, or maybe Uma and Dr. Mehra can answer. This seems to be very common in India.
Yeah, no, she showed the 302, the DQ302. That's the most common we have all over DQ3 or DQ8s, very few DQ9.
This is this is the DR7 and DQ9. So the DR701 and the DQ beta 0303.
0303.
But 701 would be mostly Rajalingam on DQ2.
No, sir, 0303 also we are seeing, but very
3 is also,
Yeah, I think the table it is simply one of the common.
DQ beta 103.
Mostly will be DQ2, and some you can also encounter. And we have seen that as well. We used to see that many years ago also, when we called it a DQ7, 8, and 9. Because the classical thinking at the time was 3 and 7 are encoded by the DQ2, 4 and 5 are encoded by DQ3. Because at that time we had only DQ3 split into 7 and 8.
Suchita, have you included only Indian samples or you have a mix of samples?
No, Indian samples. No, ma'am, Indian samples.
Okay.
Okay, thank you, Sushita. We'll go to the next poster presenter, utility of multiple platforms to detect anti-HLA donor-specific antibodies and improve renal transplant outcome. Rashwari Basvana, Mumbai, India. Would you please present your poster?
Can you see the poster?
You have to share.
We cannot see it now, but we see you clearly. We can see it now.
No, please keep it zoomed so that it's more viewable. Yeah.
Yes.
Better, ma'am?
Yes.
Good morning, everybody. So our study is on utility of multiple platforms to identify donor-specific antibodies, mainly to improve the outcome of renal transplant. This is a study from Hinduja Hospital. It's from Mumbai. So we all know that presence of donor-specific anti-HLA antibodies is considered an absolute contraindication for renal transplant. And there are many tests, cross-matches like CDC cross-match, Luminex cross-match, Flows cross-match is routinely used for pre-transplant testing as well as on the post-transplant monitoring. So since we are routinely using, we need to know what is the sensitivity for specificity, what are the drawbacks, and what is the best algorithm or what are the best tests we can put in place in a day-to-day routine use. So CDC being many economical, easy to implement, and being functional assay, many modifications has been done in spite of low sensitivity to increase this platform sensitivity. And from the last couple of decades, Luminex cross-match as well as Flow cross-match has been routinely used with variable sensitivity and specificity. So with this background, we wanted to analyze these 3 platforms and 5 tests to know what exactly the sensitivity of the specificity of these tests and how do we provide a best treatment care for the patients. So it's a prospective study done between 2018 and 2020. Ethics committee permission has been taken. During this study, 105 patients were enrolled in the study. Those were either going for the transplant or for the post-transplant monitoring patients. So all these tests had HLA cross-match done by 5 methods or 3 platforms. So CDC routine, where the incubation period, first and second incubation period was 30 and 60 minutes. So we increased the incubation period to 60 and 90 minutes, and that was a CDC extended. And CDC AHG method, where anti-human globulin was added to increase the sensitivity. So other 2 platforms were the flow cytometric cross-match and solid phase, that is Luminex cross-match. So what did we have? Coming to the results, we had 105 patients during this study period. 82 of them had negative results and 3 were positive in all 5 methods, and there were 20 discordant results. So this is the table which shows what was the positive rate in various test assays. CDC being the lowest, it was only 9, and Luminex cross-match identified 14 positives. So all these positive patients, we did a single antigen bead assay and donor molecular typing, and to identify true positives and false positives. So when we analyze further, so CD with this data, I'm sorry. Yeah. Hello. So when we analyzed with this, sorry, I just need to.
That's okay. We can see it.
Okay. So the true positive was only 4 in CDC routine, and for false positives were, sorry, just fine. Okay, so the true positives are only 4, and then when we extended the CDC, the true positives almost doubled up to 8, and the false negatives are decreased from 5 to 1. And in Luminex, as we can see, the false positives were the highest with 9, and in flow cytometry had the best sensitivity with the true positivity being 9 and false negatives were none. So when we analyze this data again in terms of sensitivity and specificity, 44% only the CDC routine had only 44% sensitivity. However, just with increasing the incubation period and CDC EHG, the sensitivity is doubled up. So Luminex cross-match had sensitivity of 55% and flow cytometry with the best with 100% sensitivity. This is the specificity and positive predictive value and negative predictive value of this test. So with this background, what we concluded was changing just from CDC to CDC extended itself improves the sensitivity almost by double fold. So why we are just saying CDC extended, considering the it's being more economical even compared to CDC EHG also, and also it decreases the washing steps in the CDC. And flow cytometry definitely had the highest sensitivity and good specificity, and this has to be implemented in routine practice, which has not been implemented so far in all the labs which we are doing the cross match. So definitely we would like to implement in our center with this data the CDC extended and flow cytometry in our routine practice in these patients. So CDC has an advantage. So thank you.
Thank you, Dr. Rashwari. Any question for Dr. Rashwari?
Rajeshwari, I'm sorry, I don't think I agree with you any of your conclusions.
Okay, sir. That's absolutely fine, sir. It's a learning point.
Word over, people are abandoning CDC, and what you are showing us is that the Luminex is not as sensitive or as specific. I think you are at variance with everybody around the world. My problem is that you may not be doing it right in determining false positivity and false negativity.
I'm sorry, sir?
You may not be determining in the right way the false positivity and false negativity. I want to understand from you, and I want to know what is your understanding of Luminex-based false positive reaction.
Sir, actually, since it is a denatured proteins are used as antigens, there is a non-specificity. So what they did was, whenever they said false positive, without the lysate, we did the cross-match, sir. So once we did without the lysate, the MFI has increased even without the antigen. So in that way, we confirmed that it was false positive. And also all the 9...
What do you say of a situation in a patient where the CDC is negative, flow is negative, and Luminex shows you a very high MFI? Do you think it is a false positive reaction? Because you made a statement, flow cross-match has to be used. Has to be used.
It has a
Everybody must follow Rajeshwari and have to use flow cytometry. So I'm asking you a situation where a patient shows a negative result with CDC, negative with flow, but high MFI on Luminex. Do you think that is a false positive reaction?
Sir, we did confirm with the single antigen bead assay and donor molecular typing if both were negative, and only Luminex cross-match, especially what we come across, all 9 patients were class 2 positive. And we did without the lysate also, the cross-match. If the MFI has increased without the just with the lysate, without the lysate, then definitely it is. And also there are case studies which definitely identified false positivity in Luminex platforms, sir.
Okay, I don't want to take a lot of the time here. Maybe
just one question can I ask?
Yes, yeah.
Yes,
You are the chairman. You're the chairman.
One question, Dr. Rajeshwari. I mean, while I like flow cytometry a lot myself, but I know that the question will always be contentious when we are trying to equate it with the Luminex and the stuff like this, especially the SAB. Now, my question was when you said that, how did you establish your cutoffs, for your MFIs and even for the flow cytometry? And how did you what was your benchmark against which you compared the whole thing?
I mean, $1,000,000 questions. Yeah, yes, the Luminex MFI, which validated, and it is 1,500. The inter-laboratory validation was also done. And see, there is no definite cutoff, as you know. We have to set up our own cutoff for the Luminex. And flow cytometry, again, we validated the true positive, false positive. We ran 100 samples, and then we took the cutoff, made a cutoff, and 50 and 80 plus from the negative control. That's what we are using, and we validated with the interlaboratory quality as well.
Thank you.
Thank you.
I think there are questions in the chat box, if the moderators can take that.
Yes, I think we have some questions. Such a high sensitivity and specificity with flow cytometry is baffling. Did you cross-check it with, did you cross-check it?
Yes, we did. And those who are all positive with the flow cytometry, all 9 cases had a donor-specific antibody, which was identified by the single antigen bead assay. These were mainly class 2, majority of them. And they did have donor-specific antibody in the single antigen bead assay.
No,
all these 20 patients. Yes,
with
sir.
donor-specific antibodies, did you cross-check it with donor-specific antibodies?
I'm sorry?
Did you cross-check it with donor-specific antibodies?
Yes, we did.
Yeah, Rajeshwari, Ajay Barnwal, these are the people who have used these technologies, on thousands and thousands of samples. His first statement, I want to again read for you, and you should go back and check it. He says, such a high sensitivity and specificity with flow cross-match is baffling. And that should correlate with your statement that flow cytometry cross-match has to be used. So please take care of both of these statements, and then it might help you.
Even Rajeshwari, we have also done quite a lot of Luminex cross matches, and we have compared, it has not been published, but we have also compared with flow, and I found somehow Luminex cross match to be more sensitive. I don't know about your center and how you have done. Almost in all the cases, it was comparable with flow. And in fact, we were able to pick it up a few of the cases, in Luminex cross match rather than flow.
Ma'am, can I make a comment? Actually, Luminex cross-match doesn't have DQ and DP.
I understand.
So we end up mixing most of these antibodies because it is not, the beads are not coated with this antigen at all. And we do know that DQ is playing a major role in chronic rejection. So we do miss these antibodies.
Can I just request Dr. Rajalingam to make his comment?
Yes,
Because he's done a
Dr.
lot of work on this area.
Wants to make a comment.
Yeah, so quick comment. I mean, seems to be you did the single antigen based antibody testing. So if you have the data, you don't need any cross match, period. So we stopped doing any kind of cross match. It's all confusing. CDC, no one in America does it. We just not at all useful. So stay with, I mean, yeah, you mentioned about that. So it's a learning point. So I think you should, once you have enough data, you should set.
You're changing her statement from flow cytometry cross-match has to be used to SAB cross-match has to be used.
I mean, if
that's a language.
Thank you.
Dr. Raja, I just had one comment to make. This is from your experience, which you've had at Pittsburgh, where you have clearly demonstrated in your papers also that, in addition to using SAB, we must have a cell-based assay. And that's where you had said that, we should be doing flow cytometric cross-match. In fact, in one of your posters in the Pittsburgh conference, which I happened to attend, it was prominently mentioned, and you also presented that data. What's your comment with respect to that? Should the flow cross-match still have its place, considering the SAB is far more important in your settings?
That's important. Yes, that's a good point, because when we use the single antigen based assays, it's all synthetic based. We are just looking at the MFI and binding reactivity. We have no clue how this antibody binds to the cells. If you may see 20,000 MFI, it doesn't bind to the cells by flow cytometry, doesn't cause a rejection. So we have C locus antibodies, DP antibodies, 20,000 MFI, it's completely negative on flow cross-match. So you should have this kind of data, understand which antibodies can bind to the cells. If it doesn't bind to the cells, it's not going to harm the graft.
So then maybe a combination of flow with the SAB would be a preferable methodology compared to the rest of it, right?
It's kind of it's like alternative methods, so you need to have the flow cytometry method as a secondary methods when you have any deal with these cases like this.
I agree with that the flow is a secondary method because the question is in SAB also, you're looking at the antigens on the surface of the beads, which may not be the same in the natural phenomenon. After all, you're doing a test to look at the antigens on the surface of the donor kidney.
Maybe helpful in the sensitized patients.
No, flow does
not all the patients, yeah. Not all the patients. Those who have a clean antibody, there is no HLA antibody, then you need to do the flow cross-match?
No, there are situations where you need to do flow as well, but not every time. I'm saying all that the discussion is centering around flow would be a secondary test. The most important is the single antigen bead.
Fair, sir. I think we'll move on. Dr. Sally, can you take it further because we are short on time?
Thank you, Dr. Ashwari. We will take it to the next poster. We will having genetic association of interleukin 18 single nucleotide polymorphism with the type 1 diabetes in North India. Akanachi Patak from New Delhi, India. Would you please present your poster?
Yes, ma'am.
Go ahead, dear. Share your screen.
आप कहाँ से हो बेटा?
Sir, I am from ICMR NIP.
अच्छा, मेरा नाम भी उसमें है. अरे वाह.
Yes, sir. Is my screen visible?
ये इनको कहते हैं academic great grandchildren.
Go ahead, ma'am.
Can I start?
Zoom
Yes, ma'am. Yes, ma'am.
It's not readable otherwise on the screen. Zoom it, please. No, you can, yeah.
You can pull it out later, yeah.
So good morning, everyone. This is Akanksha Pathak, currently working as a project trainee from ICMR NIP, and I'm going to give presentation on genetic association of IL-18 SNP with type 1 diabetic patients in North India. So we all know that there are a lot of factors which are responsible for development of type 1 diabetes. IL-18 is also one of them. IL-18 was originally named as interferon gamma inducing factor with a correlation between genetic SNPs at promoter site of IL-18 and interferon gamma production in type 1 diabetes development. So if we look at this figure, so it clearly shows that pathogenesis of type 1 diabetes. So firstly, there will be the activation of TH cells, then it will lead to the release of the cytokines such as IL-2, interferon gamma, IL-18, which will lead to the inflammation of the beta cell, which is also known as insulitis, and then it will lead to the death of the beta cell. So when there will be the complete loss of the beta cell from the body, then there will be no or less production of the insulin, which will lead to the accumulation of a glucose in the body, which will lead to a condition that is called as a hyperglycemia or type 1 diabetes. So coming to the objective part, so this study was basically planned to determine the genetic association of IL-18 SNP -607CA and -137GC with type 1 diabetic patients in North India. So for this experiment, two single nucleotide polymorphisms, that is -607CA and -137GC, were analyzed in patients and healthy controls. All the study participants, they were belonging to the North Indian regions. These two SNPs were analyzed using amplification refractory mutation system polymerase chain reaction, in short, which is termed as ARMS PCR. This method works efficiently because this method works on the principle that a specific primer will bind with the DNA and mismatch pair will not. So after obtaining our PCR product, we have obtained a result on the basis of visualization by running the PCR product on 2% agar shell, and we have obtained this result accordingly. So if we look at this data, so this is for IL-18 607CA. So for IL-18 607CA allele, we can clearly see
Did it happen to everybody or only to me?
I think it was snapped, sir.
Yes, I
It was
think.
disconnected.
Yes. I think
Happened
there
to
was
me
a
also.
disconnection.
Yeah, that was
Yes.
So where is Akanksha Pathak?
I think we.
Still joining.
Yes, we waited just a couple of minutes so everyone can join again.
Yeah, sure. It is like what happens when the power is off. You know, suddenly there was everything that was off.
Yeah, we are back.
Yes. But I don't think Akansha still isn't there.
No, she will come. I think our tech team is very, very smart. I'm sure they're trying to sort out.
Yes.
Manas is on the work. Yeah.
Ma'am, I'm here only, ma'am.
Yeah. We are waiting for you, Manas, to handle it.
अरे मानस एक आकांक्षा तो दिखती है मुझे यहाँ आकांक्षा शर्मा ये पाठक वाली कहाँ गई?
Avinash, can you please call?
Yeah, I'm calling you. I'm on call with you.
हालांकि उसने कर तो लिया था खत्म तो हो गया था करीब. We have seen the work that she has done.
आ गए मैम आ गए मैम आकांक्षा पाठक आ गए.
तो ये पोस्टर में टेक्निकल टीम प्रेजेंट कर लगा दे तो अच्छा रहेगा थोड़ा बड़ा.
I extremely apologize for the inconvenience.
Oh my God.
No, dear, go ahead.
Actually, sorry, it was because of the net connection. I'm extremely sorry. I'm just trying, just one second.
Don't be sorry for anything. Please go ahead with your presentation.
Yes, ma'am. Yes, ma'am. Just 1 second.
Okay.
You don't have to be sorry. The sorry has to be the organizers.
So I thought it's from my side. Extremely sorry.
No, Akanksha, go ahead.
Yes, ma'am. Yes, ma'am.
So kind of a.
Yeah.
Is my screen visible now?
Yeah,
Yes.
just zoom it a bit. That's it.
Yes, ma'am. So, good morning, everyone. This is Akanksha Bhatta, currently working as a project trainee from ICMR NIP. And my topic for presentation is genetic association of.
Excuse
You
me.
can go to results.
You can go to the results. We heard that.
Beginning. Go to the end. Go towards your conclusions now.
Okay. Yes, sir. So after obtaining this result and analyzing this results, we have come to a conclusion that there was an association of 607 IL-18 allele with the type 1 diabetic patients in North India, but till now we have not found any association of 137 with type 1 diabetic patients in North India. So according to this, our study has revealed an important association, and till now this is the first study to be analyzed IL-18 gene polymorphism in type 1 diabetic patients in North India. So this was it for my presentation. I would like to thank moderators and delegates for their kind attentions.
Can I ask a question?
Yes,
Thank
ma'am.
you, dear. Okay, go ahead.
I just wanted to ask you what was the rationale for selecting IL-18 and whether IL-18 807CA that you're showing, or 607CA, was it associated with the expression of IL-18?
Yes, ma'am.
And what about the rationale? Why did you select IL-18?
Ma'am, actually, a lot of work was going on the HLA reason, but I have not seen any of the work for like non-HLA reason in type 1 diabetic patients. So
There are quite a few papers on cytokine genes.
Yes, ma'am, they are, but I have like there are a lot of papers, but not on IL-18. They are on cytokines. Yes, ma'am, there are cytokines.
Just because there were no, so you did it, right? That is the only rationale?
Yes, ma'am. Also, and this is the first time, like in India, that we are analyzing results with IL-18.
No, I think she says it rightly in her introduction. If you look at the last line, She says IL-18 was originally named as an interferon gamma inducing factor with a correlation with genetic SNPs at a motor site. So that's the reason that you're doing.
That's the reason,
I
yeah.
mean, what they're saying is, it's not that if nobody has done it, I have done it, so it is justified.
No, sir, I'm not saying that. It's just that.
No,
sir.
You have to give a scientific justification. A scientific justification is already written by your mentors in the introduction.
Yes,
right?
sir. Yes, sir.
Okay.
Thank you, dear, for the posters. Any questions for her? So we will move to our next poster. Thank you very much.
Thank you.
Characterization of GAT65 epitopes possibly presented by type 1 diabetes associated DR3 or DQ2 molecules. Nueno Chuzu from New Delhi, India. Would you please present your poster?
Yes. Hello, everyone.
Thank you.
Yes, thank you. Thank you. I'm Aheno, and I thank the organizers for this opportunity to present my poster today entitled Characterization of GAT65 epitopes possibly presented by type 1 diabetes associated DR3 and DQ2 molecules. So GAT65 is a major beta cell antigen, and HLA-DR3-DQ2 haplotype is reported to be strongly associated with the prevalence of GAT65 antibodies in type 1 diabetes. But the precise mechanism of this association is not known, so we have carried out this study with two objectives. First, perform HLA genotyping using tag SNPs, and secondly, characterize GAT epitopes presented to the CD40 cells by DR3 and DQ2 molecules. So our study population consisted of 110 type 1 diabetes patients and 127 healthy controls. We used a set of 4 SNPs to type HLA for HLA genotyping as shown here. A few samples were validated using commercially available kits. For GAT assay, GAT peptides predicted to bind DR3 and DQ2 molecules were pooled into 4 groups: peptide pool 1, 2, 3, and 4, and used to stimulate the PBMCs. And the cytokines produced by the CD40 cells were analyzed using flow cytometry. So coming to the results, as we can see here, we found that 77% of type 1 diabetes patients carried DR3-DQ2 haplotype, while only around 17% of healthy controls carried this haplotype, and this difference was statistically significant. When the cells were stimulated with peptide pool 2, 3, and 4, we observed a significant increase in the production of interferon gamma by the CD40 cells in patients compared to control. Peptide pool 2 also resulted in significant increase in the production of IL-17 CD40 cells in patients compared to control. We found that the expression of TNF-alpha was comparable in patients and controlled, while IL-10 expression was significantly decreased in patients compared to control when the cells were stimulated with all the 4 peptide pools. Here, peptide pool 2 induced significant interferon gamma and IL-17 and a decrease in the production of IL-10 by the CD40 cells in DR3 positive patients compared to DR3 negative patients. Interpeptide pool comparison showed a very strong interpeptide pool comparison showed that peptide pool 2 was found to be the most immunogenic in type 1 diabetes. Coming to the discussion, we found a very strong association of HLA-DR3-DQ2 haplotype with type 1 diabetes patients in North India. Our SNP-based method showed 100% concordance with kit-based method, and it is convenient, simple, and cost-effective. Our study showed that 2 cytokines, namely interferon gamma and interleukin-17, might have an important role in GAT65-mediated immune response in type 1 diabetes. Our findings might be helpful in predicting disease risks and development of GAT65 peptide-based vaccines for type 1 diabetes. Thank you so much. I'd like to thank yes.
Go ahead, thank whoever you want to thank.
Yeah, I thank the organizers and my supervisors and my lab mates who has contributed to the study.
Any question? I think there is a question for you. Oh, go ahead.
Did you select the peptides based on their DR3 binding capacity, or just these are random peptides? Or
no? Yes, ma'am. Yes, ma'am. We have used an online epitope prediction tool, which is available online. And there we have selected the top 30 peptides which were predicted to bind to DR3 and DQ2 molecules.
Okay, that's good.
Yeah.
So is there a data in the Indian population stating that the GAD-A autoantibody is predominantly made by the DR3 DQ2 positive haplotype and not the DR4? Because as you know, type 1 diabetes is associated with both DR3, DR4, although in India, DR4 associated haplotypes are much less common than in the West.
Yes. Yes.
But in your introduction, there's a statement that this GAD-A autoantibody is predominantly made by DR3 DQ, which indicates to me that you have already done a study for the other haplotype as well.
Yes.
Has it been done?
Yes, sir. Actually, I've taken this statement and then I've cited Kishore et al. And Holston et al. This was done in Caucasian population and showed that actually there are 3 alleles were associated with prevalence of GAD65. But in our own study, we have also looked at HLA association and presence of GAD65, and we found that there was a significant relationship. And this paper is currently under review. And because it will make my poster very long, so I did not include it in this presentation.
No, it will be good on your part. Then you state in your introduction, this is in Caucasians.
Okay, sir. Yes.
In Caucasians, either DR3 or DR4 or both are associated with diabetes.
Okay,
not
sir.
In India, not in India.
Okay, sir.
Seen both in North as well as South India and various parts of India.
Yeah.
The strongest association or most predominant association is with DR3 DQ2.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir. Yeah.
Dr. Ohino, it was a very nice presentation. Just one
Thank you,
small
sir.
question that I have from you is, could you elaborate a little bit more on the translational aspect of your research?
Yes, actually, yes, sir. Actually, because this is a fundamental study, obviously there will still be a lot of time before it become applicable in clinical settings. But we think that if we can identify GAD peptides or diabetogenic peptides, which are presented by HLA-associated molecules, especially in population from North India, where DR3 DQ2 is very, very high, we can use these GAD peptides to design vaccines for this particular population. So this is a theoretical assumption, yes.
Just a point.
Yes.
I think the way you can use it, if you find the most immunogenic peptides, make altered peptide ligands, which would compete out the autoantigens rather than making a vaccine. And those altered peptides may be used, as a vaccine.
Yes, exactly. Usually, what people do with GAD peptides is that they use the whole protein. So instead of the whole protein, we can use the immunogenic GAD peptides also for therapy, for type 1 diabetes.
Good. Very nice.
Thank you, sir.
Very nice.
Thank you, sir. Thank you, everybody.
Thank you very much for this nice work. Dr. Monica?
Yeah, I'll continue. So continuing with the poster session, I would like to invite our next presenter, Ki-wook Jung from Seoul, South Korea, and he'll be presenting on allele and haplotype frequencies of 11 HLA loci in 339 Koreans using NGS. May I invite?
Thank you for introduction. Hello, my name is Ki-wook Jung from Seoul National University Hospital of Korea. Now I present allele and haplotype frequencies of 11 loci of HLA genes in Koreans using NGS. There was limited data on allele and haplotype frequencies of HLA gene, including DQA1 and DPA1 in the Korean population. In this study, we aim to investigate the genotype frequency of 11 loci of HLA genes from 300 Korean subjects using HLA-based, NGS-based HLA typing. A total of 339 samples from unrelated Hershey donor was genotyped for HLA A, B, C, DRB1, DRB345, DQB1, DQA1, DPB1, and DPA1 using 2 different NGS-based HLA genotyping kit. Among them, 173 patients was tested using the Ortai 1 lambda kit, and the remaining patient was tested using the NGS kit from GenDx. The 5-path software was used to estimate haplotype frequency and to analyze the population genetics. Coming to the result, a total of 221 HLA alleles, including 97 class 1 allele and 124 class 2 alleles, was assigned. The most frequent allele for each locus was A 2421, B 1511, C 121, DRB1 451, DRB3 221, DRB4 131, DRB5 111, DQA1 121, DQB1 311, DPA1 131, and DPB1 511, retrospectively. The most frequent haplotype was for class 1, A 3331, B 58113, C 322, and for class 2, DRB1 451, DQA1 331, DQB1 411, DPA1 222, DPB1 511. For genes encoding HLA DQ and DP heterodimers, DQA1 131 and DQB1 6111 haplotype, and DPA1 222 and DPB1 511 haplotype was the most frequently observed. In the Evans-Watterson homozygoity test, HLA B, C, DRB1, DQA1, and DQB1 showed a statistically significant negative deviation of F-value, indicating balancing selection. The frequent data of 11 loci of HLA gene in Korean from this study can provide a basis for expanding the range of HLA testing. Thank you for listening.
Any question?
Yeah, can I ask you, thank you very much, Dr. Jung, for a very nice study. I wanted to know, in the DRB104 group, is DRB10405 the most predominant, or do you also see other subtypes also?
Thank you for your question. Because of the narrow space of poster, I can show you the data for DRB1, but in non-shown data.
No, I mean, my point was that since DR4 is a very common allele in your population, so what subtypes do you see in addition to 4 or 5? What other subtypes have you encountered in your population?
You say for DRP4?
Yeah, DR4. So DRB104.
Okay.
DRB104 family in that
family
of
haplotype.
Yeah.
Okay. Wait a minute. Yeah.
You can show the data. I think I saw it 401 also a common one.
Okay. I will show our data. Sorry.
So my point was, I wanted to know, is the DRB10401 going with
We can come back later. So moving on to the next poster presentation by Vichya Rudjaman, and he'll be speaking on a longitudinal study of innate immune cell profiles of COVID-19 after recovery. Can we have Dr. Vichya?
Yeah. Good morning, everyone. Can you see my poster?
Right, we can see that.
No, please zoom it. Please zoom it and show.
Okay, okay.
That was better. You can scroll down. Yeah, that's good. Thank you.
Yeah, go ahead.
Okay, the title of my poster is a longitudinal study of innate immune cells profile of COVID-19 after recovery. For the introduction, many COVID-19 patients experience the long-term effect of COVID-19 or long COVID-19 on health post-recovery with various signs and symptoms. Most severe COVID-19 during acute phase are associated with hyperinflammatory condition driven mainly by the innate immune cells. Excessive inflammatory response during the acute phase in severe COVID-19 may potentially impact immune cells profile and function even after the recovery from the disease that may cause long COVID-19 in severe patients. Therefore, we investigate a longitudinal study of innate immune cell profile and function in Thai cohort COVID-19 patients. The objective of this study are to investigate the profile of innate immune cells at 1 and 3 months after recovery by comparing mild, moderate, severe patients and healthy control, and to examine IL-6 and TNF-alpha production by RPS-stimulated monocytes at 1 and 3 months after recovery. And for the material and method, we isolate PBMC from blood of recovery COVID-19 patients and healthy control, and then study the innate cell profile and function using flow cytometry. Next is the result. Sorry.
Go ahead, we can see that.
Okay. Figure 1, 2, and 3 show NK cell profile, monocyte profile, and MDSC profile in COVID-19 patient after recovery. The results show severe COVID patient in red bar increase the total NK cell as shown in figure 1A and the total monocyte as shown in figure 2A at 1 month after recovery when compared to the healthy control in white bar. And moreover, we found the increase of monocyte myeloid-derived suppressor cells or MMDSC as shown in figure 3C, this figure, in all COVID-19 group when compared to healthy control even 3 months after recovery. And moreover, to test the function of innate immune cell, we use LPS to stimulate PBMC, and figure 4 show IL-6 and TNF-alpha expressing monocyte in COVID-19 patient after recovery. The results show the increase of the percentage of IL-6 expressing monocyte and also TNF-alpha expressing monocyte in severe COVID patient at 1 month after recovery, as shown in figure 4A and B. The results suggest that SARS-CoV-2 infections had long-term influence on the frequency and function of innate immune cells at least for 3 months after recovery that may potentially lead to some of long-term health conditions in COVID-19 patients. Thank you.
Very good, Vijay.
Okay.
Any questions we have?
May I ask a question?
Yes, Dr. Anu.
That was a very nice presentation, especially the kind of cells that you have, really looked for with respect to the recovery. Now, I have a comment and a question. The question first, did you look at CD4 and CD8 counts as well? And did you have it, what was the kind of ratios that you found in patients, who had COVID versus those who were recovering? CD4 to 8 ratio.
I did not have the data on T cells. Yes.
And okay, fine. The other thing was a comment, and that's with respect to the recovery of the cells that you mentioned here. It is a normal phenomena whenever you will have a suppression of the marrow by any of the illnesses, which includes viral illnesses. Monocytes happen to be the first cells to recover in any case, and perhaps attributing it to just COVID may not be right. Would it be right on my part to say that for your study?
Again, please. Sorry, again, please.
So generally what happens is whenever there is a suppression of the marrow, the recovery of the marrow is generally seen as a monocytosis in the peripheral blood. So my question is, in your case also, would it be right on us to attribute it to COVID alone? Because in any other illness also, it will be similar kind of a concept when you have suppression of the marrow at any point. Would that be right on my part to say that?
Chair, do you agree with his comment? He wants to know, do you agree with his comment on the recovery of cell types?
Yes, yes.
But that's a natural phenomenon, he said.
Yeah, this is what. Have you seen this phenomenon in other viral illnesses as well?
इनके साथ जब बात करो स्लो बोलो स्लो.
I think Dr. Raja is also wanting to ask a question. Dr. Raja, if you could go ahead and ask the question, please.
My question is very similar to what you are asked. So during the COVID infection, all the cell type, the NK cells and monocytes goes down, right? So the severity of the COVID-19 is associated with the less NK cell population, as well as monocytes. But your data is when patient is recovered, like 2 months after, you see a lot of monocytes and a lot of NK cells. I think Dr. Handu's question is, we see it in the bone marrow transplant, probably the same effect. You lose the cells during the infection, then it comes up much higher than the healthy controls. So these cells may not even functional. So that's the question. Maybe you need to test the function. I think this is something new. So you see a lot of NK cells and monocytes after 2 months COVID infection. So whether it's functional, whether it's harmful, whether it's helping the patients, that's interesting question.
Thank you.
I think that will be very interesting if she can look into that aspect also.
Not only that, but I think what we need to see in the patient in the original state, what was their LN ratio between lymphocytes and neutrophils. Because in COVID, lymphocytes number go down, neutrophils go up. And the patients who are unable to recover their lymphocyte number, they go on to develop severe form of infection. So it is important to also correlate your data to LN ratio.
Okay, thank you.
Thank you.
But one more just quick question. I mean, so do you have the longitudinal data? So you collected the samples in different time points on particular patients, right? You may want to look at those data, whether these NK cells or the monocytes are stable or it is going down to up.
Yeah, at 3 months, it go down for NK cell and monocyte percentage. Yeah.
Oh, okay.
Thank you.
Okay.
We can move on to our next poster. And this is by Pimpyayo Sotsai from Bangkok, Thailand. And the poster is on unveiled digit mutation in a Thai severe COVID-19 patient.
Hi, everyone. Thank you for your introduce me. Today, I would like to share my study of the TIGIT mutation in Thai COVID-19 patient. We know that the severe COVID-19 patient risk to high mortality, so that we would like to observe the genetic variation in this patient group that may associate to disease severity. After we analyzed 55 of the severe COVID-19 patients by exome sequencing, we found a new heterozygous nonsense mutation. See here. In the TIGIT gene from one male severe patient. This mutation patient, truncation of the TIGIT protein at the residue 56. And this mutation is not identified in around 3,000 individuals in-house Thai exomes database, but can be observed in one in the around 30,000 allele in the genome AD database. And the TIGIT mRNA expression in PBMC of this mutated patient is lower than in non-mutated patient that I didn't show in the poster. Next, to observe the TIGIT expression on the T cell, we found the frequency of the TIGIT expressing CD3, CD4, and CD8 were less in mutated patient. Mutated is CD45, and this less than the non-mutated patient at 1 month after recovery. We do 2 patients for non-mutated patient. For the next. We know that TIGIT is an inhibitory receptor presenting on the T cell. It can suppress T cell activation and T cell function. So we investigate the effect of the TIGIT mutation on the T cell function in the mutated patient. It showed that the level of the TNF alpha, interferon gamma, and IL-2 producing T cell, both CD4 and CD8 in mutated patient were higher than non-mutated patient. See in the red bar. Moreover, the some cytokine and chemokine level in the serum of the mutated patient in the gray bar were higher in the mutated patient than the non-mutated patient, and also in the healthy control in the white bar. In the conclusion from this result, we hypothesize that nonsense TIGIT mutation in severe COVID-19 patient may cause low TIGIT expression and less control the immune responses. And thank you for my grant and also my advisor and all the collaborators. Thank you for your attention.
Thank you, Pimpayo. Can we have any question?
There seems to be a dichotomy in what you are saying. In your summary, on the one hand, you are saying that the numbers of TNF alpha, gamma interferon, IL-12 producing CD3. Can you go to the can you go towards your
summary
conclusions?
Summary, right?
No, little more. Yeah, here.
Yeah.
So on the one hand, you are saying that the frequency of gamma interferon and TNF alpha IL-12 producing cells, CD3, 4, 8, were higher in the non-mutated individuals, but lower in the mutated individual. But then on the other hand, you're saying the cytokine levels were higher. So does that indicate that there is a heterogeneity of these cells? Is there a subgroup of cells, the cytokine producing cells that have got enriched over there, and therefore there are more cytokines formed?
And normally in the severe patient, normally is the result is show the lymphopenia, right? But the cytokine production in some papers show that the severe patient show higher produce the cytokine of these 3 cytokines that show. And in the result, we found that in the all of this is a severe patient, and especially in the mutated patients, show a higher cytokine expression than the non-mutated patients.
Yeah, I understand that. What I'm saying is, on one hand, you're saying that the cell numbers are going down.
Yes.
But the cytokines are
But
going
the function is high, you're right.
So if the cell numbers are going down, you would
No, I didn't mean that the number of the cells. I mean the number of the TIGIT expressing CD3, TIGIT expressing CD4, and also CD8 is low, right? Yes, but the function is high, right?
So that might be due to a subset, a sort of.
Oh, you mean the subset of the,
assumed subset that might get enriched.
I think she's saying the number of CD3, CD4, CD8. Go to your summary, please. The number of CD4, CD8 T cells are lower, but the cytokine secreting cells are higher in mutated than in.
From the total CD3, CD4, and CD8, from the phenotyping T cell is the same, not different between mutated and non-mutated patient, but I didn't show in my slide.
Yeah. No, going back to Rajini, that is what I'm saying. So maybe this mutation is enriching a subset of CD3 or CD4 cells, and therefore these cytokines are more. So there's no evidence.
Okay.
And, on the face of it, it shows like a contradictory statement. Your cell numbers are going down, but your cytokines are going up. Unless one shows that there is a subset that has got enriched specifically due to
I agree.
mutation. Yeah. Okay.
Yeah, I agree.
Yeah.
Thank you.
We have one more question in the chat box from Dr. Gaurav. Do you update about any other TIGIT mutation linked to COVID-19 severity?
Sorry, please. Again,
any other mutation have you found with the COVID-19 severity?
No. I didn't see others. Yeah, only one mutation from the 55 BS skin. Yeah.
Thank you.
Thank you, Pimple. Nice.
Thank you.
So we move on to our next poster presentation, and it is by Supicha, is on the inhibitory effect of enoxaparin on the plasma-induced neutrophil extracellular traps from COVID-19 patients with severe pneumonia.
Hello, good morning to all the honest audience and welcome to my presentation. My name is Supicha, the PhD student from Chulalongkorn Hospital from Thailand. I'm here today to share my discovery in the training teachers about the COVID-19 and the treatment. As a recent report found that the neutrophils are the most predominant immune cell in blood and ability to. And during the COVID-19 infection rate, and the numbers of the neutrophil is very, very useful to prognostic true in severe case. And one of the most crucial functions of the neutrophil that is very dangerous is the neutrophil extracellular traps on it, which had been reported that the nets are associated with the pathogenesis of several disease, including with the acute respiratory distress syndrome or ARD, including with the COVID-19 infections too. Their web-like structures of nests can directly damage alveolar tissues and the endothelial cells, cause the breathing difficulty and thrombosis in the vascular lumen, leading to the respiratory failure and death. With this complication is always found in the severe case of the COVID-19 patients. Therefore, the early treatment with the anticoagulation could decrease the excessive thrombosis in tissue. Interestingly, the low molecular weight heparin or enoxaparin are commonly used as an anticoagulation, and it also shows the anti-inflammatory effect. So I hypothesize that the enoxaparin could potentially prevent the net formations and reduce the complications of the mortality of the COVID-19 patients. So The objective of my study is aimed to investigate whether the enoxaparin could reduce the net by induced by the plasma from the COVID patients and determine the molecular mechanisms of the enoxaparin on the net formations. In these experiments, the patients with the COVID-19 infection were classified into 3 groups based on their severity, which is the no symptom, mild to moderate, and the severe pneumonia. Plasma on the patient in each group and together with the healthy control were collected and incubated together with the neutrophil, with or without enoxaparin treatment. And after that, I measure the level of net products such as the double-strand DNA, neutrophil lysate, and histone complex. And some of the inflammatory cytokines were measured. The results show that this one, the net formation visualizes by the immunofluorescence microscope, and the net components was decreased by the dose-dependent manners. And the enoxaparin could reduce the gene expressions associated with the net and the inflammatory signal in the neutrophils. So, in conclusions, the level of the net were associated with the severity of the COVID-19 infections. And this could be like a predictive biomarker in the COVID-19 patients. And furthermore, the plasma from the COVID-19 patients enhance the net that could be inhibited by the enoxaparin pre-treatment. And our findings suggest that enoxaparin could be the potential use as the adjunctive therapy in the COVID-19 patients, particularly those with a severe pneumonia or ARD. Okay. Thank you for your attention.
Very nice presentation, Subisha. Can we have questions?
Chipa, can I just ask one question here? Can you let us know what was the number of patients that you worked on? What were the cohorts that you worked on? The numbers.
The number of the patient, right?
Yes.
I have the 20s in each group. The moderate, in the, and the no symptom is 20s, mild to moderate 20s, and the severe pneumonia is 20s. And half the control is 20 also.
And if I have understood you correctly, you also tried to do an in vitro exposure to enoxaparin. Am I right?
Yes, as I have a plan to collect the real sample from the patient who given the enoxaparin. It's on my process. But I didn't show the data on these presentations yet.
Is this enoxaparin only used in this severe pneumonia?
You mean as an anticoagulation, right?
No, I wanted to know, have you in Thailand done any clinical trials with this enoxaparin?
You mean we have a lot of drugs except
No,
from the
enoxaparin that you are talking about, and that it decreases the NETs, which is very good actually. Has anybody in Thailand done a clinical trial in patients of COVID?
I don't have any data from the others hospital, but in my hospital, they have some of the patients who give like the prophylaxis of the enoxaparin to the patients. It's like even the patients have the symptom of the thrombosis, but they give is like the prophylaxis to prevent the severity. Yes, but others hospital, I don't know. I don't have any information.
Yeah, I mean, well, it's a huge field. Then if the clinical trial has to be done, what will be the situation in a mild to moderate group, and in the severe group, what percentage, and does the reduction in NETs correlate to the disease improvement?
Yes.
Okay.
Thank you.
Thank you. Thank you, Supecha. Very nice presentation. So we move on to our next poster, and that is by Simi Mehra from Gurgaon. And the poster is on prospective study to analyze pre-transplant compatibility algorithm for life-related donor kidney transplant in Indian setting. It's a single center report.
Thank you so much, ma'am. Good morning, everyone. The topic of my presentation is prospective study to analyze pre-transplant compatibility algorithm for live-related donor kidney transplant in Indian setting. It's a single center report from Medanta, the Medicity Hospital in Gurgaon. So the main objective of our work was to establish an appropriate test algorithm that could cater to our settings, like developing countries like India, and which could include the HAI as the universal test. Because many of the test algorithms include anti-HLA identification, that is HAI-SA, as the upfront test, and obviously it is very expensive. So we have included almost 100 recipients which
Are you
underwent.
sharing your slides?
Yes, ma'am.
It's not being shared.
Can't see your slide or a poster. Can you share the screen of your poster?
Just a second. Sorry. Can you just see now?
Still waiting for it to come. It hasn't come yet. Yes, now it's come.
It's visible now. Sorry, ma'am. Sorry for the inconvenience. Should I start it from the starting or should I start it from the material and methods itself?
Just start from the objective.
Objective. Okay. So the objective of our study was to establish an appropriate test algorithm that could cater to our settings like developing countries, India, and which could exclude the universality of HAI because it's a very costly test and it is used. So we have included 100 samples for our study, and pre-transplant workup was done. And in that screening test, CDC crossmatch, flow crossmatch, anti-HLA antibody screen was done with that, along with typing. And as a gold standard, SAP testing was done, that is single antigen bead assay for all the recipients to identify the possible DSA. So if the DSA is not there, the patients were cleared up for the transplant. But alternatively, if the patient is having the DSA, we offered an exchange kidney program, or after desensitization, if the DSA goes down, the values goes down, we can proceed for the transplant. So in this case, we have we found out the sensitivity and specificity of each test, that is CDC, flow, and HAS, and individually and in combinations also, to find out which is the best sensitive test. Just a second. So in the results, out of 100 cases which we have analyzed, 81 were males and 19 were females. 88% of the recipients underwent the transplant. And broadly, sensitivity and specificity of CDC crossmatch was found to be very low, that is of 14.3%. And 100% sensitivity was found in the combination of flow crossmatch and anti-HLA antibody screen. 75.5% of specificity was observed in this combination. So our study suggests that this algorithm, which have flow crossmatch HAS combination test as a screening test, this can be used universally since it is 100% sensitive and it's inexpensive also. The specificity of 75.5% indicates that combining these two may detect up to 24.5% of false positive results, which could easily be resolved by using virtual crossmatch, that is again based on SAB results. And secondly, we would also like to suggest that CDC crossmatch assay may be discontinued in this workup because of its very low sensitivity of 14.3%. Thank you. Thank you so much.
Okay. Simi, have you not done the Luminix crossmatch for these patients?
No, ma'am. No, ma'am.
Okay. Any other question? Anybody?
Okay. I had to go somewhere, so I haven't followed this study, so it's okay.
I have a question.
Yeah. Who's this?
Yes,
Yes,
Dr.
Dr.
Monica,
Raja.
can I- yeah, sure.
Yes, yes, Dr. Raja, please.
Yeah, thank you. Yes, I think when you started, I was happy. I think someone has to think about the algorithm that suits for the Indian situation. You know, you cannot take everything from Western because we operate very different. So that's a good thought. But in the algorithm, I don't see any HLA matching, involved. Say, for example, out of 100 transplant, there may be some HLA matched sibling transplant. In that cases, you don't need to do any testing. There's all this cross-match antibodies. It has no value if the donor is completely matched. Okay, I think that should be brought up in the first.
I agree, sir. But the number of that patients are quite less. But in those patients where the 100% identical match is not there, so this algorithm would be helpful in that case also. And in families where they can't afford, this much of burden like of HAI, they can opt for, flow cross match and HAS. So just to lower down the financial burdens on the families, this algorithm, we get an idea to start this algorithm.
I didn't have the opportunity to listen to everything that you said, but I'm just commenting on your results, the last line. Finally, our results suggested, and you're saying that the sensitivity of the CDC in your center is only 14?
Yes, sir.
Oh my God. Does it not depend on the type of the patient you take? Is a highly sensitized or a retransplant or patient on a long waiting list and all?
No, 80% of the patients are male, so likely unsensitized. So only 19% are female, maybe a subset of 19% are probably sensitized. So kind of correlating.
Yes, sir.
Simi, you should mention the data on those female patients and what was the correlation there?
Yeah.
That will give you a better idea because otherwise it seems
yes, ma'am.
correlation.
Thank you so much, ma'am. Definitely, I'm going to write it down and I will look into that. Surely, ma'am.
Unless those male patients have some antibody or they are, sensitized by some other mode. But
yes, ma'am. So basically on sensitization, correlation of this. Sure, ma'am. I'll definitely look.
I mean, look, what I learned during our PhD days is some Nobel laureates came to our lab and they said, keep looking at your data again and again, That
Exactly.
really
Yeah, exactly, ma'am. Every
Before
time
making a statement.
Exactly. We are very new to this.
Okay.
I think Dr. Handu, you can take over, continue the
Sure, ma'am.
next.
Thank you. Thank you. Thanks, Simi. You can stop sharing your screen now.
Thank you so much. Thank you so much. Thank you so much.
So we move on to the next presenter, and the next presenter is Dr. Jyoti Rajak from Mumbai, and she will be talking about possibility of non-consanguineous parent being a full matched prospective donor for patients undergoing hematopoietic stem cell transplants. Yeah, if you could just, yes, exactly, and please zoom it again.
Jyoti, can you zoom the poster, please? Are we audible, Jyoti?
Yes, ma'am.
Okay.
I'm sorry.
It's okay if you can't then.
Can I start?
Just go ahead and present. It's all right.
Yeah. So hello and good morning, everyone. My topic for today's poster presentation is possibility of non-consanguineous parents being a full match prospective donor for patients undergoing HSCT. So as we all know, donor selection is an important aspect in HSCT, with identical sibling donor being the preferred donor. But the possibility of finding an identical sibling donor is between 25 to 30%. Matched unrelated donor and haploidentical donor do serve as an alternative,
but efficacy of such transplantation is still less than the related ones. We found that researchers like Rafa et al. And Balsi, they suggest that consanguineous marriage increases the availability of actually identical related donors within the family for HSCT. But my aim here was to present cases. It is a case study where we present the cases from northern region of India, wherein we found that non-consanguineous parents were complete match and could serve as a donor for HSCT. So the high resolution typing of patients and donors were done using Sangers and next generation sequence typing. Analysis was done using SPT and GS engine software from GenDx. Here we can see there are three cases, and the first and the third case are from Bihar and the second case is from UP. The typings are mentioned in different colors. The same color indicates that the typings are same. Here in the first case, we see that the parents, like the mother and the father, are sharing this haplotype, which is shown in blue, and that resulted in the offspring being a full match to his father. Same is the case. Same is the situation in the second and the third case. So here in the above cases, we can see that the parents are sharing haplotypes even when the marriage is non-consanguineous. The sharing of these haplotypes in parents could be due to the presence of common HLA haplotypes in Indian population. The common haplotype shared in case 2 and case 3 was A33, B4403, C0706, DR beta 0701, DQ beta 0201, and in the first case, the parents shared haplotype HLA A1101, B3501, C0401, DR beta 1202, DQ beta 0301. Frequency of occurrence of these haplotypes is 13% and 2% respectively in Indian population. This is an unpublished data from our lab. We may conclude that in cases where patients do not have sibling donor and carries a frequently occurring or a common HLA haplotype, parents should be integrated in donor search before or constantly with an unrelated donor search. Thank you so much.
Thank you, Jyoti. The poster is open for question. Yes, Dr. Rajni, you wanted to ask something?
Yes, saying that it is an Indian population is a very generalized statement. I would like to know which particular population or ethnic group or a caste group that you've studied, where you find this kind of, because it is very rare to find, the couples sharing a haplotype.
Yes, ma'am. Yes. So these three cases are from northern region. So the first and the third are from Bihar, and the second is from UP. And I have also studied the frequency, not the frequency, I would say, but
are these Muslims? Is there a caste? Is there some inbreeding or something in the population where you've studied? That's important to know.
No, I think that it may be because the marriages that happen in India are within a community. And as we also have seen that A33, B44 and C07 is a very common haplotype.
This is a common haplotype, no doubt.
It was also mentioned by Dr. Uma in her presentation from northern region that the Saharias and the Punjabi Khatris also shared. They had a good amount of this haplotype in their population as well. And when I checked in my North Indian population data, the first haplotype, the A33, 44 and C07 is in 11.89% of patient and donor. We see that this haplotype occurs very commonly.
That's true. We have published that. What I am trying to say is there a particular caste group that you see, the parents sharing the haplotype?
Rajini, this is from their day-to-day typing. They have found some people who share the haplotype. This reminds me of some studies done in the 80s. And we used to teach our students that HLA might also be involved in mate selection. That that study showed that the couples who love each other more than others tend to share, the number of alleles sharing in them was much more. But anyway, this is like separated at birth. I wanted to know what is the total pool that you have?
So I've had
these two haplotypes that you're talking about, which are common in the Marathi population, out of how many? What was the number?
So this is from North Indian population, and I have this total.
Again, Rajini will come back to you. What do you mean by North Indian population? North Indian population.
I'm so sorry. But so I have taken it from I have taken the individuals from
I
like
understand. My other question was, did you find A26B8? Because, I'm reminded of my study that I published first time in 1986. That was serology based. The A26B8 was the most common and with the highest delta value there. Is it that the NGS is teaching us something else? But my own data that we are generating in SRL laboratories indicates that that's the haplotype with the highest delta of the value in India.
No, sir. The A26B8 is not as frequent in when you look at population absolutely homogeneous ones. It is the A1135 that is more common that we also observe.
It still would be in the amongst the first three or so. It's a very common haplotype. It is disease associated and all. So it's good. What you're saying is this couple might be separated at birth, huh?
So, Dr. Jyoti, the only comment which I would like to make at the end is that as a practice in any BMT setup, the first activity starts from the sibling, then the parents, because haplo is catching up pretty significantly now. And obviously the take home from your poster is anyways being practiced for a large number of years already, So thank you very much for the poster. And I think we'll move on to the next one. We don't have so much of time to really wind up. We have 2 more posters to go. And I would request you to stop sharing.
Dr. Anil, can I just mention, if you want, the candidates are not able to Zoom, the technical team has the poster, they can do it for you.
Oh, wonderful. That's great.
So they will display in a bigger size if you want.
Yeah, thank you.
Thank you.
Thank you, ma'am. So we move on to the next poster by Dr. Alok from Mumbai, and he'll be talking about immunogenetic interactions of HLA and KIR genes along with scleroderma patients from Western India. If we could have his poster on the screen, please. Are you sharing, Alok?
Yes, sir. Just give me a second.
If you want, Alok, the technical team can do it for you. Manas, can we have it? Avinash can do it for us.
Is the poster visible?
Yeah.
Yes, absolutely. Go ahead, Alok.
Yeah,
I'm sharing the poster, so you can just instruct me up or down.
It's there already on the screen.
Good morning, everyone. The title for my poster is Immunogenetic Interactions of HLA-C1, C2 and KIR haplotypes among scleroderma patients from Western India. Scleroderma is an autoimmune rheumatic disease with certain complications and have a various genetic background, infectious and environmental factors involved. KIR genes are known as killer cell immunoglobulin receptor genes and are located on chromosome 19q13.4, and they comprise of inhibitory genes as well as activating genes. And they were discovered first on NK cells, but later on it was known that they are also present on gamma delta T cells and some population of CD4 and CD8 plus T cells. HLA-C are ligands for KIR, and especially HLA-C with epitopes of HLA-C group 1 and group 2 are recognized KIR epitopes. In this study, we assessed the KIR HLA interactions. The objectives were to determine KIR and HLA C1, C2 gene frequencies in scleroderma cases and healthy controls. And we also tried to find any association of KIR HLA interactions and in scleroderma. We enrolled 57 scleroderma cases and 100 healthy controls. Blood was withdrawn from them and DNA was isolated from the blood, and some serological assays were also done to know the severity of the disease. KIR genes were typed using duplex PCR SSP, and all 16 KIR genes were typed using this method. HLA-C group 1 and group 2 typing was also done by PCR. Our results showed that sclerodactyly was the most common clinical manifestation among scleroderma patients. Total 26 and 58 KIR genotypes were identified in scleroderma, and healthy controls respectively. And we used allelefrequencies.net database to identify these key genotypes. BX key genotype was more than AA genotype in our study population, which involved both scleroderma patients and healthy controls. The frequency of 2DL1 and 2DS1 was more in healthy controls than scleroderma. This was our first observation. And this is the graphical representation of key genotyping. And now if we look at the table, the combined frequency of 2DL1 plus and 2DS1 plus was quite high. And similarly for 2DL2 plus and 2DS2 plus was high in healthy controls compared to scleroderma patients. Now, if we look at the KIR and HLE combination, 2DL2 minus S2 plus and C1 plus was significantly more in scleroderma cases than healthy controls. And for 2DL2 plus, 2DS2 minus, and HLA-C1 plus was also significantly high in scleroderma individuals than healthy control. We conclude in our study, individuals with KIR HLA combination of 2DL2 negative, 2DS2 positive, and HLA-C1 positive may be susceptible to enhanced scleroderma manifestations. And KIR HLA combination of 2DL2 plus, 2DS2 minus, HLA-C1 plus may confer protection towards scleroderma manifestations. These are some of the references which we used for KIR genotyping and HLA genotyping. And we thank to ICMR for intramural supports, and there are no conflict of interest from all of the authors. It's a multicentric study, but there are no conflict of interest. Thank you.
Thank you, Alok. Thanks very much. It's open for questions. Any questions, anyone?
Just a comment, if I can.
Right.
I'm assuming that the controls are again the ethnically matched ones with the patient cohort. Is that right?
Sorry, ma'am, I didn't get you.
Are your healthy control population well matched ethnically with the patients that you're studying? Because the KIR gene frequencies and the HLA would differ from one to the other. So it's very important that you have.
No, they were not matched, ma'am.
Not matched?
No.
Then how do you compare the frequencies?
Actually, ma'am, this is a part of a study, and this is just a pilot study which we thought of doing it. And this was in a very initial stage. And later on, then we thought that if we can do this in the study which we were doing already.
How many centers have collaborated in this?
Ma'am, if we can see.
From which regions of
KEM Hospital from Mumbai and BYL Nair Hospital from Mumbai. There are two collaborating centers here.
Can you
primarily Mumbai based?
Can you keep your poster, showed up on the screen? Usko remove nahi karo.
Should I remove
Inbox se question lena.
Upload
Chat
again
box
in
mein
the
hai.
poster.
Avinash, can you just put it again, the poster?
Yes, sir. Just
Alok?
And there are questions from Rajalingam. Yes.
Yeah,
Yes.
sure. Alok, so your KIR 3DL1 frequency seems to be too low, so almost everyone should be carrying the 3DL1. But so only 40% of the scleroderma patients have 3DL1 and 80% of the controls. So I think there's some problem. That's one comment. Then the second one, I don't know why you want to see this combination of 2DS1 plus C2, but there's no one showed there's a binding of 2DS2 with the C1.
Okay. So previously we saw one paper, and after referring that paper by Momot, which he studied in scleroderma patients, we observed that frequency of 2DS2 plus was quite high. And in that paper, they didn't study HLA-C, but now we know that 2DL1 plus has, 2DL2 and 2DL3 has HLA-C1 as its ligand. So we thought of trying to combine this and try to find out if there is any KIR HLA association in this study. So as I told, it was a pilot study and it was not well planned, but still we tried to do it and tried to highlight the study in this Ishi Con.
I mean, so look at the frequency very quick, so all scleroderma, except this framework genes, everything's going down. That's something very unusual, correct? You
Yes.
look at the data later, yeah.
Yes, sir. Sure,
The
sure.
framework gene expression has to be there for all the cases that you're studying. So
Yeah.
methodology needs to be reviewed.
Yeah, ma'am. I think the framework genes are there 100%. 2DL4, 3DL2, 3DL3, and 3DP1.
Frequencies.
Yes, ma'am. If we can see it, 2DL4, 3DL2, 3DL3, and 3DP1. These are the framework genes, and they are 100% for both the healthy controls and scleroderma patients.
So Alok, I understand, and as you are saying, this is just a pilot study for you to get started, which is
Yes.
good. But then if your healthy controls are not belonging to the same ethnic group, then you cannot make a statement in the conclusions that so-and-so is associated. Right?
Sir, can I ask for a suggestion from you if you don't mind? So actually in Mumbai, we have a very heterogeneous population here. Like if we go to the OPDs while sample collection, people from different ethnicities have resided in Mumbai. So it becomes quite a difficult that one single ethnicity can be selected from the OPDs while doing sample collection and enrolling them in the study.
Yeah,
I'm sorry, Alok, I will have to interject here.
Yes,
We will
please.
have to stop you, and you can do it on the sidelines with Dr. Mehra.
Yes.
Meanwhile, we'll move to the next speaker. Thank you very much. Thanks a lot, Alok.
Thank you, everyone.
Avinash,
Thank you, Ishi.
Avinash, if we could have the final speaker of the day, Lekha Rani from Chandigarh. Her poster is effective presence of pre-transplant donor-specific HLA antibodies on kidney transplant outcome. So it seems like as though almost 30% of our posters are still dedicated to the antibodies. Yes, Madam Lekha, ma'am, can we go ahead?
Thanks for the introduction. Good morning, everyone. The title of my presentation is Effect of Presence of Pre-Transplant Donor-Specific HLA Antibody on Kidney Transplant Outcome. Coming to introduction, the detection of donor-specific antibody against HLA is a prerequisite for pre-transplant monitoring of patients, and it includes both complement fixing and non-complement fixing antibody. And the presence of cytotoxic antibody is responsible for acute rejection, whereas the presence of non-cytotoxic antibody or antibody against HLA class 2 has been associated with long-term graft loss. Therefore, the detection of both cytotoxic and non-cytotoxic antibody is of interest in both pre and post-transplant monitoring. And the cytotoxic antibodies are mainly detected by complement-dependent cytotoxicity assay, whereas the other methods of detecting DSA include flow cross match and single antigen bead array assay. But their impact on the renal transplant outcome, like the sensitive flow cross match and the low titer single antigen bead based assay on renal transplant outcome, is unclear. So in this study, we first compared the three methods of DSA detection for pre-transplant monitoring. And secondly, we analyzed the rejections in transplant patients with preformed DSA defined by positive on flow cross match and virtual cross match based on single antigen bead array assay, but negative on CDC. For that, we retrospectively analyzed our patient data of year 2019, which composed of 254 patients, and CDC and flow cross match were performed in all the patients, whereas a virtual cross match was available in 116 out of these 254 patients. And the CDC was detected by microscopically by detecting the damaged cells, whereas the flow cross match, we detected the anti-IgG binding on both T and B cells by looking at MFI. And for the virtual cross match, we defined DSA positivity against donor HLA antigens. Now coming to the results, first we compared all three methods for positivity, and the CDC was positive in 10% of our cases, whereas the flow cross match was positive in 33% of the cases. And when we looked at the dual positivity by both the methods, out of these positive cases, 14 cases were dual positive, which indicate approximately 13% of all the of these two, CDC and flow cross match. Similarly, for the virtual cross match, the 27 cases were positive by virtual cross match, whereas the 83 by flow cross match. And when we looked at the dual positivity, the 13 cases were positive by both virtual cross match and flow cross match. And then we looked at the graft outcome. Out of these 83 flow cross-match positive, the transplant data was available only in 40 cases. And among the virtual cross-match positive, the transplant data was available in 16 cases. The other cases were not performed, the data was not available for the transplant. And when we looked at the transplant rejection rate among these flow cross-match and virtual cross-match positive, the 20% of the flow cross-match positive cases showed rejection, whereas the VXM, the virtual cross-match, among the virtual cross-match positive cases, the 43% cases showed rejection. And we observed more of T-cell mediated rejection than the antibody mediated rejection. And when we compared the virtual cross-match and the flow cross-match along with the rejections, the total eight cases which were flow cross-match positive were rejected, and among virtual cross-match, the total 7 cases were rejected. And out of these 8 cases of flow cross-match, the 5 cases were virtual cross-match positive and showed rejection. So there was 33% concordance between both the methods on transplant outcome. So these results suggest that the presence of DSA by both single antigen and flow cross-match increases the risk of graft failure. So donor selection should consider the presence of antibody identified by both flow cross-match and even the weakly positive single antigen, even in the negative CDC cross-match. And the limitation that we showed graft outcome at 2 years of follow-up. So larger follow-up study with the stratification of group based on antibody strength will yield more knowledge. So finally, I would like to acknowledge all the consultants from our department, renal transplant surgery and nephrology, to our donor and recipient, and the organizer to giving me the opportunity to present my data. Thank you.
Thank you, Dr. Lekha. The poster is open for questions. So there is a question in the chat box, which I'm taking for you directly. What was the CDC cross-match results amongst the virtual cross-match positive patients? Were all virtual cross-match positive patients, cases positive for CDC also?
No, sir. Only among the 27 virtual cross-match positive, only 5 cases were CDC positive.
Okay. And, what the inference that I have been able to pick out from your poster is that what kind of a combination would you eventually, kind of propagate? Would you like to have people a virtual cross match with a flow cross match, or would you have only a virtual cross match and then go ahead with the, progress of the transplant? How do you project it now?
Sir, basically, I suggest that the virtual cross-match should be done, but in the cases where the MFI is very low, like less than 3,000, the weakly positive, what we consider the weakly positive, then the flow cross-match should be considered. So these cases which were showed concordance with the flow cross-match, they were weakly positive, because in the high titer, with the high titer MFI, we don't, the transplant is not performed, which is the usual practice.
Okay, so if I have understood you correctly, you are trying to say that whenever you have a weak positive flow cross match, that's the time you would go ahead to do a SAB. And in such a case, if the SAB shows you whatever positivity, you will then do a virtual cross match, and basis that the next course of action will be taken. Am I correct?
No, sir. Sir, actually, in our department, we first get the CDC and the flow cross match simultaneously for the same patient. And then the single antigen was also performed later on. But on the flow cross match, we used, because it's overly sensitive and we get positivity on B cells in some of the cases, like it was positive in 83 cases. But then we performed single antigen, it was only in 14 cases, which were also positive by flow cross match. So I mean to say that the virtual cross match should be considered when we are saying the flow cross match results.
Okay. Dr. Uma, you have a question.
I just wanted to ask, is the strategy the same for the primary transplants or repeat transplants? Would you consider the prior antibodies from the previous donor and then decide?
Ma'am, the retransplant patient,
low MFI antibodies even for the previous donor, and then how would you go about planning?
Yes, ma'am, that should be considered, because the flow cross match, if even if we get low positivity on the virtual cross match, then it should have high positivity on the flow cross match. So it should be considered for the retransplant patients, specifically for the retransplant patients.
Thank you for your answer.
Thank you. Thank you very much. I think with that, we'll conclude the session, so there are no further questions. I would like to thank
Thank
the
you.
organizers on behalf of my co-chairs, Dr. Monica and Dr. Sally. If you could just come onto the screen as well.
Last वाली लड़की नहीं आई?
नहीं sir, 14th one is
The
not.
last वाली नहीं है, हाँ.
ठीक है. चलो, all right.
So I would like to thank Dr. Sally as well as Dr. Monica and the organizers for giving us this opportunity, and we look forward to the rest of the sessions for the day. Thank you very much, everybody.
So
I would also like to thank the organizers for inviting me to moderate the session, and it has been a wonderful conference for the last 3 days. We have all enjoyed all the great speakers and all the great sessions. Thank you so much.
Anil, I was saying now you sit with these two ladies actually and send us the.
Yes, sir. Yes, sir. We're doing that parallelly, sir, on the WhatsApp.
Yes. I would really like to thank everyone for this wonderful conference. I really enjoyed it. Glad to be among you all, and I hope to welcome you in Egypt very soon in our conference.
Oh my God.
Yes.
So next Ishi Aphia conference will be in Cairo.
Yes. No, sir, not in Cairo. It will be in Sharm el-Sheikh. This is our better city, where we have all the fun.
Better. But anyway, Sally, we want to thank you because Uma told me that you were the brain behind getting us those CECs from ACHI.
It was my pleasure, sir.
Very good, very good. No, it actually is really nice and help our
Your
young
conference is highly appreciated. It's very scientific and well organized, and it really is an addition to the scientific community, really.
And we say namaste to you.
Thank you. Thank you.
Thank you.
Thank you. Akanksha, can you please thank everyone?
On the behalf of the organizing committee, I would like to thank all the participants and moderators for conducting this session. Now we'll break for 15 minutes since we are running behind the time, and we'll join after the break for our next scientific session. Thank you.
Thank you, everyone. Bye-bye. Thank you, Sally. Yeah.
15 मिनट तो हो गए भाई. Is Diane joined in?
Hello.
Hi, Diane.
Hi, how are you?
Can't see you. Your camera is off.
Hang on, it's coming. Can you see me now?
Yeah. Oh, there you are. Yeah.
How are you?
How are you doing?
I'm doing well. It's a nice sunny day here, so.
I wanted to talk to you for a couple of things, maybe a little later.
Yeah.
So, we will exchange emails, or maybe you can send your mobile, and then we can chat on WhatsApp.
Okay. Yeah.
So send me your WhatsApp number. We can chat on WhatsApp.
Okay.
Yeah.
I'll
Because
send
it
you
is
an
free.
email.
It is free. Send me your mobile number through email, so we can connect through the WhatsApp.
It's been a wonderful meeting so far. You've done a really wonderful job.
Thank you so much. And have you been attending it all?
Mostly in
Yeah.
between work, you know.
Quite a few from your lab were registered for the meeting.
We've had like people come in and out. So I think, well, we're 5, 6, 7, 8, maybe 8 or 9.
Yeah.
Yeah.
I saw the list from Aphia region yesterday. We have about 45 delegates from Australia, New Zealand, Korea, Hong Kong. So that's nice.
Yeah, we've tried to encourage people, some of the younger people, because if you know when we have to travel, it's always hard to get people to travel money to go.
Yeah.
So
And this is a good
take
opportunity.
advantage of the virtual meetings.
That's how we could get so many speakers also, because it's so nice that you can have all the international speakers come over. You don't have to pay any hotel cost and everything.
Yeah, well, you had some amazing speakers, so it was really good.
So one of the chairpersons we have switched, and we still have 2-3 minutes, I think, to start. Akanksha?
Yes, ma'am.
Any news about Campbell, Diane? Where is he nowadays?
Oh, I don't actually know. I know that he was traveling around Australia, but I don't know if he's back or where he is. We haven't seen him. I haven't anyway.
Is he now gone into another field or something?
No, he's retired completely.
Not tired. I'm also retired. I'm not tired.
No, I think Frank and Campbell just retired and that was it. Done.
And do you ever meet Frank?
Oh, I saw Frank probably, maybe middle of the year. Yeah, we had a coffee.
He's okay.
Yeah, he's all right. Yeah, he's enjoying his retirement too. So
yeah,
we haven't seen him for long.
Yeah.
His wife cooks.
Well, you've got, as you say, got the next generation.
That's true. It has to carry on.
Yeah.
Yeah, but I like your title. You're saying next generation.
Yeah.
This is Diane is all on next generation, even work. So Diane, what about the care? Are you doing also the care typing on this Oxford Nanopore or what?
Yeah, so John Downing, he's actually doing a PhD, and that's what his project is. So he developed a really nice method.
Okay.
So yeah, it's a shame he didn't get time to present some of his results, but next time.
I wish he had mentioned. We would have definitely included one. Yeah.
I'd love to do it next time. Hi there.
Oh, there you go, John.
John, we didn't know you were doing that work. It would have been so interesting.
Well, I'll keep it quiet until I've got something to show you, which is pretty much now. But
maybe we can have some of the future Ishi webinars somewhere we can include such topics.
Oh, yeah.
Interesting. Yeah.
Happy to do that. Sometime well into the next year, please.
Let us know when you have your results interesting to present.
Okay.
Yeah.
So, Akanksha, chalo.
Okay, so welcome back, everyone. This is the last session of this conference, and I hope everyone is having a great time. On the behalf of the organizing committee, I would like to welcome our moderators, Dr. Yogendra Pal Singh and Dr. Raja Rajalingam. Dr. Raja is the Program Director of UCSF Immunogenetics and Transplantation Laboratory. Here he is conducting more than 900 solid organ and hematopoietic stem cell transplants, whereas Dr. Yoginder Pal Singh is the head of the HLA and Molecular Biology, SR Diagnostic Private Limited, Gurugram, India. The session will be followed by a joint discussion. Participants are encouraged to post their queries in the chat box. I now request the moderators to kindly take over the stage.
Thank you, Akanksha, for wonderful introduction. So I will like to introduce our first speaker of this session. It has been a wonderful 3 days, very good sessions and excellent talks. So our first speaker of this session is Dr. Danny De Santis. Is a scientist, senior scientist in charge at the Department of Clinical Pulmonology, Pathwest, Pana, Stanley Hospital, Perth, Australia. She completed her PhD at the University of Western Australia, investigating the role of care in HST. She is currently in charge of HST donor search program in Perth, Australia, and responsible for developing new technologies to improve HLA typing methods used in solid organ transplantation and bone marrow transplantation algorithms. Dr. Dan and her team were the first to publish a novel high-resolution HLA typing methods utilizing single molecule sequencing on the Oxford Nanopore platform for deceased organ donors allocation. This method has been implemented and is Ishi accredited and can be performed in 4 hours during deceased organ donor workup. The technology is also currently being validated for high throughput sequencing for HST matching. She currently serves as a sub-editor for journal HLA and counselor on Aphia and International Histocompatibility Workshop Committees. She is co-chair on the HLA DP haplotype component on 18th upcoming International Histocompatibility Workshop. She will be speaking today on next generation rapid high resolution HLA typing on Oxford Nanopore technology platform. I feel happy to hand over it to Dr. Dan. Please, Dr. Dan.
Thank you. I can share my screen. You can see my slides?
Yes,
we can.
Okay, so I'd like to thank the Aphia and Ishi Organising Committee for inviting me to speak today, and I declare I have no conflict of interest. So I'm going to talk about next generation sequencing, and we started our journey in 2011, where we were the first laboratory in the world to be ASHI accredited, one of the first laboratories in the world to be ASHI accredited for next generation sequencing in 2015. And in 2016, we started evaluating the Oxford Nanopore sequencing technology for long single molecule sequencing for HLA. And so that's what I'm going to talk to you today about. So although we have implemented NGS sequencing in our laboratory, and we have been routinely typing since 2014, ion torrent sequencing, as we use in the laboratory, still, although provides mostly unambiguous HLA typing, there are challenges that we still face with this technology. So we still are unable to resolve phase ambiguities across regions that are greater than 500 to 600 base pairs. So therefore we still get genotyping ambiguity. We have the inability to sequence through highly repetitive regions. We're highly dependent on bioinformatics and instrumentation, large instrumentation, such as liquid handlers, quantitation equipment, large sequencing instruments. And we would like to sequence more genes and more transplant-related genes for the same cost and turnaround time. So some years ago, we asked the question, is MinION sequencing feasible for HLA genotyping? And we thought it might be suitable because of its ability to sequence long read single molecule full gene sequences. And that allows us to phase across exons and the entire gene, generating no genotype ambiguity. And it also allows us to sequence regions that we haven't been able to sequence, like introns or SNP regions in regulatory regions, which can affect expression. And we'd like to do all this so that we can better match between donor recipients and then hopefully improve patient outcomes. The MinION sequencing isn't heavily dependent on bioinformatics. There are no mapping algorithms required, just alignment. There's no need for expensive equipment. The actual device comes free when you buy the reagents, so it's fairly inexpensive. And the library preparation is simple, and the sequencing allows potentially reducing the turnaround times to HLA genotyping results. So just a bit about the technology. So the nanopore is a protein that is found in nature, and it enables the movement of molecules from one side of the membrane to another. Oxford Nanopore has engineered proprietary protein nanopores through mutating key residues in the barrel of the pore. And these pores are 1 nanometer in scale, so very small. So so the nanopore is embedded into a synthetic polymer membrane that allows measurement of current over time. So when electric current moves through the pore, when electric current is applied, ions move through the pore, and the current generated by the flow of the ions is measured. And this is known as baseline current. When an analyte moves through the pore, the disruption to the current is measured by sensors in the MinION device. And this disruption to the current by each of the nucleotides is unique and therefore allows each base to be recognized and recorded. So shown here is a nanopore embedded into the synthetic polymer membrane. Tethers on the flow cell help move the DNA to the opening of the pore. The double-stranded DNA passes through the motor protein, which unzips the double-stranded DNA and breaks down secondary and tertiary structures to form linear DNA. The adapter holds the DNA molecule in the pore, and the DNA moves through the pore. Sensors then identify the current disruption and records the nucleotide. Once the strand is finished sequencing, the pore is available for the next strand of DNA. So the MinION device is only 100 grams in weight and can be powered through a USB 3.0 cable. And the flow cell contains 512 channels, and each channel has 4 wells with a total of 2,048 membrane wells. And each well consists of 1 nanopore embedded into the polymer membrane. Sequencing can occur at 400 base pairs per second. And a typical run is between 10 to 20 gigabytes of data. The nanopore sequencing is scalable. So Oxford Nanopore have 4 devices or 4 ways of sequencing. So we have the Flongle, which is sort of a single-use flow cell, which I'll talk more about later in the presentation. The MinION R10 flow cells, which provide large capacity and enable you to sequence more samples. The GridION, which allows 5 flow cells to be sequenced simultaneously. And then the Promethean, which includes 48 flow cells. So all these are available, and depending on your throughput and your requirements from your lab, you can select from these different devices. So we started our investigations by evaluating the available sequencing chemistry at the time, and this was some years ago, so there were quite a few sequencing chemistries available. We looked at the 2D chemistry, and this chemistry is no longer available. The 1D108, the 1D109, and the 1D squared chemistry. All the workflows consists of an N-repair DA tailing step, followed by ligation of sequencing adapters and the motor protein. And finally, a bead-based purification step is performed to enrich the adapter ligated fragments. The library pools for bidirectional sequencing were further incubated with the hairpin adapter or the 1D squared adapter. We did try the rapid kit for HLA typing, which is a 10-minute library prep step. However, that was shown not to be applicable for HLA typing. Each of the sequencing chemistries were evaluated for quality, accuracy, the ability to sequence the length of the amplicon, and the ability to sequence through GC-rich or homopolymer regions. So the accuracy of data generated from each chemistry is shown here on the graph. Sequences with Q-score equal or higher than QC made up 99% of detected sequences in the 2D chemistry, shown in the pink or purple, 83% in the 1D squared, in the blue, 51% in the 108 chemistry, and 95% in the 109 chemistry, shown in the green here. The 2D chemistry had the highest Q score of Q15, followed by the 109 chemistry, averaging at a Q11, and then 1D squared chemistry. Sequencing reads generated by the different chemistries for 12 samples were compared for read counts, read lengths, and sequencing across the GC regions by mapping reads to the DPA0103 allele. So shown here in the top in figure A, read counts corresponding to their sequence identity. In B is the read density in log scale for better visualization. In C is the alignment, ability of the sequence to be aligned to the full length of the amplicon. And D shows the ability to sequence through the GC regions. Read counts with the 1D109 chemistry was higher than any of the other chemistries shown here in A and B. There was no difference in the ability to sequence the length of the amplicon or through GC-rich regions in any of the chemistries. In the table below, we show the accuracy of each sequencing chemistry, and the 1D squared, the bidirectional sequencing kit, showed a mean identity of 92%, and the 1D109 chemistry had an accuracy of about 90%. So based on these results, we decided that we would continue using the 1D109 kit, and that was on the basis that we were able to produce a large number of read counts or reads with the best accuracy or high accuracy. We also investigated the integrity of the nanopore, and we saw that after 24 hours of sequencing, the integrity of the pores was significantly reduced, which told us that sequencing for more of that time was really of no benefit, as most of the sequencing data was generated sort of around 16 to 18 hours of sequencing time. As discussed by Lin Trong yesterday, we needed to develop an automated cloud-based analysis pipeline to allow us to analyze the data in a turnaround time that was suitable for routine laboratory. So MinION sequencing requires a base calling, demultiplexing of samples if you're using barcoding, and filtering. We filter based on quality and size. And as described by Lin, the manual process used to take us around 3 days. And with this developed pipeline, we can now do it in the cloud with GPU power in 2 to 3 hours. And so this pipeline is not only used for analysis, but also the storage of our NGS and TGS data. And as we developed this with Microsoft, and they tell us that this is actually an Australian first, so we're quite proud of this work, and it allows us to do the analysis quite quickly. So in 2019, Oxford Nanopore released the Flongle flow cell, so a single-use flow cell, which allowed us to develop a rapid HLA genotyping method at 2-field resolution, and we call that the ONT rapid HR method. This method was accredited in 2020 by Ishi, and we have been using it since May 2020 for routine typing of our deceased donors. The Flongle flow cell is a disposable single-use flow cell containing 126 pores for sequencing, and this method allows us to achieve 2-field typing resolution within about 4 hours from DNA extraction to HLA genotyping. So we perform a DNA extraction. We then use the all-type method. So we use the all-type 11 loci single tube assay that amplifies the 11 loci of interest. And we chose this product because we used it routinely for our NGS. So we had experience with this assay, and it was available. But the normal thermocycling time was about 2.5 hours. So we wanted to reduce the time of PCR to make this method quick for deceased donor typing. So we modified the PCR conditions to reduce the amplification time down to about 1 hour and 40 minutes. And we extensively evaluated the PCR parameters to ensure that we weren't getting any allele dropout or allelic imbalance by reducing the thermocycline conditions. The benefit of the MinION sequencing is that you don't need a lot of DNA. So by reducing the thermocycline conditions, the time, we were able to still generate enough DNA to sequence on the fungal. We then performed the library preparation and sequencing as per the manufacturer's recommendations, and that takes about an hour and a half. And we generate about 8,000 reads in 40 minutes. And then we use NGS Engine to do the allele pooling software. So of the 252 class 1 alleles analysed, around 91% were resolved to 2-field resolution or higher. No alleles distinguished by polymorphism in exon 4 and rare alleles could not be excluded in about 9% of class 1 alleles. Of the 84 DRB1 alleles, 96% were resolved to 2-field resolution, and all DRB3 and 4 alleles were resolved to 2-field resolution. However, we did see some ambiguity in DRB4. We didn't find any discrepancies in DRB1, 3, 4, and 5 results between our method and the new fungal method, nor we actually didn't find any discrepancy across any of the loci. And of the 84 DQB1 alleles, 74% of alleles were resolved to 2-field typing. And the alleles that we couldn't resolve were mainly due to the ambiguities associated with DQB1 301 and 0302, but these were comparable with the low resolution SSO typing that we were using at the time. All DQA and DQB DPA1 alleles were resolved to 2-field typing resolution. And the ability to phase using long read chemistry provided the most benefit for the DPB1 locus. So of the DPB1 results, almost 98% were resolved to 2-field resolution. So just to show here, this is an example of a recent donor we typed and the level of resolution we able to obtain using this method. Shown here is the phasing ambiguity. So you can see for some loci, we're able to completely resolve the ambiguities, but in some, for some loci, we still get a small number of phasing ambiguities, but still the level resolution is much better than our previous SSO method. And just to show you some of the data here, showing DP, where we're able to phase exon 2 and 3, and therefore resolve ambiguities associated with the inability to phase across exons. That there is a higher background in MinION sequencing, and that's why it's so important to have good allele balance. And here we can see with the DP, we have the alleles represented equally, and therefore the high background really isn't an issue. There are some limitations, so we still have issue sequencing through homopolymer regions. And so here with DQB10603 at this position, we can see there's a string of 6 Cs, and at the 6th C, the software can't distinguish between the C and indels, and therefore we get an ambiguity because of that. So further limitations is the fungal flow cell stability. So the manufacturer indicates that there is a 4-week shelf life. And so that sometimes makes it difficult in the lab to ensure that we have enough to cover us for the 4 weeks for deceased donor typing, and we don't wastage. We don't have wastage of the flow cells because we're not using them within that time. And we have implemented this method for urgent family studies and other bone marrow related work, and therefore we're able to increase our order and ensure that we have sufficient flow cells available, but utilise them in different areas of the lab. And of course, as I mentioned, the homopolymer errors that we still see with the 9.4 flow cell. So in conclusion to that part of the talk, we have shown that rapid high resolution typing is possible during organ allocation without delay to the organ allocation. High resolution typing provides certainty when assessing donor-specific antibody, and therefore patients are not excluded on the presence of potential DSA. There is generally no need to retrospectively type donors, and that's a cost saving. We do retype patients to confirm homozygotes or any rare alleles that we may have typed on the night. Allele allows the identification of allelic antibodies and therefore increases the possibility of organ allocation to highly sensitized patient who currently wait in our transplant program 3 to 4 times longer than those who are not highly sensitized. High resolution will allow epitope analysis if that's what we do, if that's what we can do on the night. And we do know that there are some limitations. And in regards to those limitations, Oxford Nanopore have released some new nanopores, the R10, and we've been working with the R10.3, and there's just about, there's a new pore, R10.4, that's just been released that we will evaluate in our laboratory soon. But these new R10 nanopores have a longer pore structure and therefore allows more of the strand of DNA to move through the pore. And it's claimed that the accuracy is therefore improved when they're sequencing through homopolymers. So we have done a little bit of work with the R10.3 flow cells, and this is just data from our first 3 runs. Here shows data yields, the amount of data that comes off the 3 flow the flow cells, the number of reads that we generated for our first 3 runs, the mean length. And so for run 1, we used a full gene method. So the complete gene was typed for all the loci, whereas run 2 and 3, we were using long range PCR. The mean Q score, so across the 3 runs, so Q11, so good accuracy. And then the sequencing times varied between 4 to 16 hours. And that was mainly due to the number of samples that we were able to put on the flow cell. So we looked specifically at whether the R10.3 flow cell reduced the homopolymer errors, and we actually did see an improvement on the R10.3. So here shown is the R9.4, where we weren't able to distinguish the C at this position, but when we sequenced the same sample on the R10.3, we were able to, the software was able to call this position with higher accuracy, and therefore no typing ambiguity. So in conclusion, HLA typing is feasible with MinION sequencing with some limitations, and this is still very much a work in progress. Oxford Nanopore is releasing or just released a new 10.4 flow cell with new chemistry, which is claimed to be to further improve the homopolymer errors and also increase the accuracy. So we're about to start to evaluate that chemistry very soon. And we have an improved base calling software, which seems to improve accuracy, but again, that's still ongoing in our laboratory. But we are using the Flongle method to rapidly type to 2-field resolution deceased donors during the workup. And again, further work is underway at the moment to reduce the turnaround time to around 2 to 3 hours by modifying the library prep step. And that looks very promising. The workflow is very simple, and automation of the data analysis allows us to rapidly process the data and reduce that lengthy analysis time. And again, the limitations with the base calling errors, the shelf life of the flow cell, and obviously the high background requires your PCR to be robust. But again, with improved base calling software, we hope to see an increase in accuracy. So this all happens with the help of many people in the laboratory. So in our laboratory, Lynn Trong, who did a lot of the MinION work, and support from Dr. Lloyd DeSonia and Patricia Martinez, and Oxford Nanopore, who have supported us along the way, and with the data analysis pipeline, Microsoft Australia, Charles Paulson and Philippe Aurora from BizData in New Zealand. Yeah, thank you for listening, and take some questions.
Thank you, Diane. Dr. Diane, excellent talk. We will take all the questions at the end of the sessions. It was a very good insight for technology, which is reducing turnaround time. So we'll take all the questions at the end of the session. We are moving to the next next talk. Dr. N. Chandra Molin Servant, who will be speaking on who is manager at NGS Laboratory and Genomics Laboratory Private Limited, a Newburgh company group. And he has several 11 publications by his name, and he's also co-author for peer-reviewed journals. So over to you, Chandra Molin, please. Share your slide.
Yes, sir. Actually, thank you for this opportunity. I already give the video to conference team.
Yes, sir,
so, yeah. Yes, thank you. Thank you.
To Ananda. Thanks for giving this wonderful opportunity to make my presentation in Ishi. Once again, today my topic for discussion is towards continuous improvement in turnaround time, experience with Miafora Express software. Then we go for NGS. Before going to start the NGS HLA typing, earlier we do the Sanger sequencing data or HLA typing by sequence-based typing method. In that Sanger sequencing methodology, we do a lot of laborious process when we do the limited number of exons to cover the class 1 and class 2 alleles. Second thing is that we have lots of ambiguities. And the fourth thing is that we have the lot of reporting of patient to reporting to the clinician should be take too much of time to. That means the third is a little bit difficult for to report the samples. For this reason, we go for the next generation sequencing platform based on the criteria of the main criteria is that length of coverage of the genes, overall HLA genes. The second is that reduce the minimum number of ambiguities. Third thing is that we have to easily identify the novel alleles and also the easily identify the null alleles. So these criteria gives a major important role for reducing the turnaround time for the huge number of samples in a single batch. That means the high throughput of NGS platform may play a vital role in the HLA typing while going for a NGS platform. So for this reason, we are going to move into from Sanger sequencing into next generation sequencing. So the coming slide will describe the what we done in the genomics our laboratory. So let me briefly describe the what we done at genomics in 2017. So genomics started using Miafora NGS Plex kits based on HLA typing. Based HLA typing 2017 April. So genomics we are using the using the 2 mini-6 system using the mid-output reagent cartridge with having the 300 cycles run. So with the help of these two systems and the Miafora NGS kit, we done around 22,481 HLA typing samples as of now on 6th November 2021. So this is about little bit brief about our what we done on genomics laboratory for the past 4.5 years. So moving on to the next slide. So this image simply tell you the coverage of the Miafora kit. We are targeting HLA-A, HLA-B, HLA-C, and HLA DP beta 1, HLA DQ beta 1, and DR beta 1. So for HLA class 1, class 1 locus, we are targeting completely whole gene for the HLA typing. Whereas in DR beta 1, for targeting complete all the from exon 1 to up to exon 6. Whereas in DQ beta 1, they are covering all the genes except exon 6. So for exon 6, there it will be a development stage. So it will be in commercial for upcoming years. So then third one is the DPB1. Sorry, third one is the DPB1. We are covering exon 2 to 4 for exon 2, 3, and 4 for covering the DPB1 gene, DPB1 gene. So next slide, we are going to discuss the robustness of next generation sequencing based HLA typing using MIA4FLEX Limina Mini-seq platform. This data already present in European EFE conference in 2019. So this data clearly tells you that there are two operators done the HLA typing in NGS platform. So for this platform, we compare, we got the 95.9% of automatic calls from operator A and 96% of automatic calls from operator B. So roughly around 95% for overall automatic calls in this NGS platform. Second thing is for regarding the manual analysis, and that means the manual calls, it's 2.6% for operator A and 2.5% for operator B. The remaining 1.4%, from operator A and 1.5% from operator B, values are unassigned. Due to few factors. There are some insufficient data. There are some insufficient data are there, and very low coverage of the samples. So overall, for operator B and operator A and operator B, for overall, the accuracy of the NGS platform MIA4 for MIA4 and the MIA4 software, it's around 98.5%. This is the table simply clearly tell the robustness of the MIA4 software data analysis. So next thing is that this is a library protocol. Next thing is a library protocol. That is the older protocol we used few years back, 1 year back. It starts from long range PCR to data analysis. So it will take time. So the long range PCR will take 6 hours to complete. And quantification of PCR products and the gene balancing and coding. It will have a lot of hands-on and manual pipetting works still are needed. So whereas in MFlex platform, that is upgraded version of MFlex platform, they simply upgrade the protocol by reducing the time of long-range PCR from 6 hours to 4 hours. 6 hours to 4 hours. After that, they simply remove the quantification of PCR products and the gene balancing and coding. So this will help these both steps from long range PCR as well as the quantification and the gene balancing method. These two they simply cut down the time, and we done the remaining library construction, site selection, library amplification. After that, library quantification. While in site selection, we are using automatic site selection method in the Pippin Flip methodology, or else we are doing this simply by magnetic field based method. Both methods give the good quality of data as well as the good results. So this is a major thing. Because of this time reduction in the long range PCR as well as in the quantification and gene balancing, we reduced for overall total procedure time, it will reduce to 2 hours are reduced for overall protocol time. So this is the Flex protocol and the MFlex protocol. That is the multiplex protocol upgradation. Then moving into the software part. The previous slides it shows the complete the lab protocol slide. Lab protocol, it describes the lab protocols, and this slide completely describes the MIA4 software. There is the 3 proprietary algorithms. One is for dynamic phasing analysis algorithm. Other one is for consensus algorithm. These 2 algorithms helpful for the phasing the sequencing data. The second one is the competitive alignment algorithm. It will use clearly for the mapping of the genomic data. So next we will describe the what's the algorithm do in the software part. So these slides simply shows the version 5.1 software data and the version 1.10 of 1.0 data for data. Both images show the analysis window. It will simply clear the mapping of the data using the mapping algorithm. This data completely gives the exonic coverage and the genomic coverage of the data. This will give the complete mapping of the genomic data to the reference sequence. The second one is the phasing algorithm. This phasing algorithm is completely used for to identify the mismatch in the exonic position and intronic position. Also play a vital role in the identification of novel alleles. Especially this position includes the identification of nucleotides, whether this nucleotide is polymorphism or not. Based on the phasing algorithm, it will definitely helpful for the identify the novel alleles and the null alleles. So both the software have some kind of advantages as well as disadvantages. So some major disadvantages for 5.1 software is the timing. So for number of samples is 24 samples. It will take 12 hours to complete. For 32 samples, it will take 20 hours. It will take 20 hours to complete the analysis of HLA typing data. Whereas in 48 samples, it will take 24 hours to complete to analyze the data. It will be applicable to the 96 samples also. It will also give for completion of 96 samples, it will take 48 hours to complete. So it will be a very huge timing to analyze the data and reporting data to the clinician. So for this reason, we make an alternative for this software. Then we are moving into the MIA4A Express software. This MIA4A Express software version 1.0 released in September 2021 by MECOR. The first results of 24 samples will be in less than 2 hours. So all the 24 samples of the result will come within the 3 hours. So it will give you easy reporting for easy turnaround time for reporting this, analyzing and reporting the samples. Easy summary page for review of samples. Review of sample results while displaying critical warnings for more streamlined review. It's also helpful for the identification of novel and null alleles based on the mapping and phasing algorithm. So for what is the aim to proceed the comparison? What is our aim to comparison the both the software 5.1 the version 1. There are 4 criteria we have to focus mainly on that. What is the time from completion of sequencing to availability of results, accuracy, number of reads required to improve accuracy, percentage of automatic and manual. The main criteria we used for comparing the data. So these slides completely describes about what are the materials and methods used for comparison the data, software data. So around 538 samples typed with the MIA4A MPLEX kit. Targeting 6 genes in HLA. The sequencing kit is used for the Illumina mini-seq platform with the mid-output kit 300 cycles. IMG reference database version is 3.42. So the smallest batch size was 24 samples. The largest size is and largest size of the sample batch is 96 samples per batch. So based on these material methods, we first analyze the sample, all the samples in the MIA4A 5.1 version, and then analyze with the MIA4A Express software. So based on that, we finally got the results. In this page, we got the results from the above materials and methodology. So in this result, it will clearly tell you that the time taken for 32 samples to analyze the data in MIA4A Express software is only 3 hours. So 3 hours, it will get the 32 samples of analyzed data. When compared to 5.1, it will take 20 hours. Whereas in 48 samples, it will take time to complete is 4 hours only. Whereas in 5.1 software, it will take 24 hours to complete. Whereas in the same way, for 96 samples, the time taken for complete the analysis is only 8 hours. So 8 hours to complete. Whereas in 5.1 software, it will use 48 hours to complete all those 96 sample data. So the main criteria are the automatic calls. So the automatic call for MIA4A Express software is around 94.98% when compared to 5.1 version. it will give 93.40%. Whereas in manual calls, it will give 1.49% when compared to 5.1 software, it will give around 1.18%. The remaining, there are few unassigned alleles as 3.5% unassigned alleles in Express software. When compared to 5.1, it gives 4.71% of unassigned alleles. So both the software having the good data as well as the analysis of the data, but the turnaround time for the 5.1 software and the version 1.0 software, it will be drastically different. For especially analyzing the samples and the reports came out from the software. So it will be drastically made a drastic difference in the from the previous software. So the other minor queries or minor advantages and disadvantages in the MFR Express, there are no color codes. So that means the smart flag systems. Smart flag systems easily guide us to whether the sample is automatically called by the software or manually called by us, or it will be commonly well documented allele, or it will have the linkage disequilibrium table. So it will manually, these color codes are manually guide us to analyze the samples as an end user. So this is the major, this is the disadvantage of the MFR Express 1.0 software. So finally, I conclude my presentation as observations. The removal of the two steps, that means the gene quantification, gene balancing. It does not affect the quality of the results anyway. It is the laboratory part, lab typing protocol. This is a lab oriented procedure. The second thing is that informatics part, that is the analysis section. The TAT is considerably reduced using the Express version 1.0, and hence will be tremendous help to the clinician. So this is an observation we find out from the software MFR Express 1.0. So thank you. Thank you for once again giving this wonderful opportunity to present my presentation in Ishi. And on the same time, on this occasion, I would like to thank my team, my colleagues, and my IT team. Without them, this presentation is not possible to present in this conference. Thank you once again. Thank you.
Thank you, Chandramoulin, for your talk. We'll take questions at the end of this session. I will request Dr. Rajalingam to introduce our third and the final speaker.
Jaginder, can I have some slides? Yep, it's coming up, I guess. Perfect. So this is my pleasure to introduce Dr. Martin Penning. Is a Chief Technology Officer at GenDx. It's a long introduction, I see first time. So Martin graduated from the Washington University as a molecular biologist. I don't even see a lot of stuff. So I guess, he's going to talk about the latest best-in-class NGS-based solution for transplant diagnostic monitoring. So we'll hear Martin say, is the recorded talk? Martin, you're on? Oh, good.
Hello, everyone.
Yeah,
I'm happy to be able to present to you on your conference, although being virtually present. And in the next 20 minutes or so, I would like to give you an update on some of the cutting-edge technologies of NGS typing, HLA typing, as well as transplantation monitoring, also using NGS technologies. My name is Martin Penning, and I'm already for 11 years at GenDx, and I'm currently the Chief Technology Officer, and as such, responsible for all the new products that we develop. So today I would like to discuss three topics. I would like to end with showing some data and the new products that we developed for NGS chimerism monitoring. I would like to show you that we have a proof of concept method for rapid NGS-based HLA typing strategies using the MinION. But I'd like to start with a platform comparison study that we did applying our NGS-Go MX11 and 3 multiplex. So a similar study where we compared the different sequencing platforms we done at the times when NGS was still up and coming for HLA typing. Now, many years later, of course, there's also many developments in the sequencing technologies. So in this study, we used the state-of-the-art methods, of course, consisting of Illumina, Ion Torrent S5 was included, also the PacBio SQL2, Nanopore, the MinION was included, as well as the MGI DNB-seq method. So there were developments in the sequencing platforms, and of course there were developments in the reagents. So for this study, we used our new flagship product, NGS-Go MX11 and 3. It contains a 3-tube multiplex for all 11 HLA genes. And not only it's a multiplex, also what you can see in this picture is that, except for DRB41, all genes included are covering the whole gene. All exons are included in this analysis. Why did we choose for a 3-tube multiplex? Because in that way we can generate the highest data quality for a multiplex. Actually, the data quality is as high as with singleplex amplifications. So how did the study look like? As said, we started out with our NGS-Go MX11 and 3. It contains of 3 tubes of dried primers, and they all come with their own reconstitution buffer, as you can recognize it with the same tube, same cap color. So upon use, you dilute the primers in the reconstitution buffer. Of course, the kit contains the enzyme, polymerase, and water for the rest of the reactions. Then you set up 3 amplification reactions, identical protocols, and you also can run them on one cycler, as the amplification protocol is identical as well. After amplification, there's no need for quantification. You can just pool all the amplicons 1:1:1 ratio in one big tube. Subsequently, you can use part of that pool to continue with the library prep. So for this study, we applied 58 DNA samples from the GetRM panel as provided by the Coriell Institute. We used the amplification that I just showed, and then the pool of amplicons we separated out over the different library methods for the different methods. So we used the LibreX method and NGS-Go LibreX for Illumina and Ion Torrent, and for PacBio, MinION, and MGI, we used the respective library preparation methods. So to jump to some of the conclusions already, we show good data quality with all of the methods. So here I show an example of an HLA-A typing, and just to get familiar with this type of picture, here on the upper panel is for Illumina. What we show is the nucleotide position of the genes. In yellow, the exons are represented, and on the y-axis we see a logarithmic scale that depicts the noise and the allele balance. So here at the level of 1%, this is just below this just all the regular sequencing errors that you can find. They're nicely below 1%. And this line is important. Here all the heterozygous positions are presented. And as you can see here, they're nicely in 50-50 balance, meaning that both alleles are equally present. So I'm not going over all of the pictures here, but in the next few slides, I would like to give a bit more detail. Starting with Illumina, here's the data of one of the samples, the pre-typing result, the typing result that we found in our study. We looked at 3-field typing concordance, so in this case all were correct. And this is also an important quality parameter, the estimated second allele, which is a measure for the allele balance. So ideally it should have a value of 50, meaning that both alleles are there 50-50. So for Illumina, we found a 100% typing concordance if we look to the 3 fields pre-type. For the next platform we evaluated is PacBio. You also see 100% typing concordance. But I have to show that PacBio also offers another advantage, which is presented here. So this is an example of, I think it's DPA. We find a few heterozygous positions here in the intron and one heterozygous position here at the UTR, which is spaced by about 3,000 nucleotides. And in MySeq sequencing, you can nicely see the heterozygous, but you cannot phase them together. Whereas with PacBio, as it applied long-read sequencing, you can phase all of these heterozygous positions together. So PacBio may even result in fewer ambiguities. Next platform we looked at is Ion Torrent, the S5. Very high typing concordance, 96%. It's a little bit less than with the previous two platforms, and this has, of course, a very specific reason. We know that Ion Torrent, sometimes it's more challenging to sequence the homopolymers. So what you see here is the overview as presented in the previous slide. Nicely low noise, except here at this position, in the beginning of exon 4, you see an increase in the noise. I'll zoom in a bit, and then you see the increased position is here. You see that it's indeed part of a homopolymer, which makes it quite difficult to determine the exact number of Cs in this homopolymer. With Ion Torrent, this sometimes may result in difficult positions that really need attention in the data analysis. So here I show an example of HLA-A, but we found similar positions in, I think, DPB1 and some of the DRB genes. Lastly, the MGI platform. In this experiment, 2 samples failed, but overall there's still a 97% typing concordance. So also this platform results in reliable typing results. Remarkable was that at 3 positions in 3 different genes, we found similar issues as with the Ion Torrent, where just at single positions we saw an increase in the noise that sometimes might result in a position that's called heterozygous, where it should be homozygous. The last platform in this study was the nanopore platform, the MinION. So this work is still in progress. So far, I can show you the result of 8 samples, and the overall conclusion is that class 1 is fully concordant, 100%. We also see that class 2 and then especially DRB1, DQB1 are a little bit more challenging sometimes. As represented here, you see that the allele balance is 20%, meaning that the amplification is quite out of balance. Although in the other platforms, it was shown that the amplification actually was not out of balance. It was nicely in balance. But after sequencing, it seems that the second allele is present less frequently. So if I show you some more data, these are 2 samples, sample 1, sample 2, sequence on the MySeq. We look at the signal to noise ratio, the difference between the lowest heterozygous and the highest noise position. And with Illumina, you see that even though there is positions where they are slightly out of balance, both alleles, you can still nicely discriminate the 2. Same sample sequence on MinION, in this case, can also be nicely typed. But here in the second sample, you see that sometimes an increase in noise and a little bit out of balance of the 2 alleles can sometimes result in interference of noise and the signal. So this is a bit where typing whole genes with the MinION might be still challenging for some of the class 2 genes. So conclusion of this study is that Illumina, PacBio, Ion Torrent, and MGI seem to be suitable for routine high-resolution HLA typing. And with the latter 2 platforms, there are certain positions that might require some attention when doing the final data analysis. So I didn't call MinION here because we think there's a different way to use and to apply the MinION system. I think it's also earlier demonstrated by a presentation of Diane DeSantis, but in the next few slides, I'd like to show you a proof of concept for rapid HLA typing using the MinION sequence. So the goal is, like mentioned, to have a fast typing result, especially targeted to apply for disease donor typing. So we have to have a result within 4 hours and then 2 field typing results in by far the most samples. So we worked on optimization of all steps in the workflow, as mentioned here. And to give you a little bit of insight here, so for example, amplification, the original protocol took about 2.5 to 3 hours, but by changing the enzyme to the same enzyme that we also used for our other HLA typing products, and by doing protocol optimizations, by reducing the elongation time and by reducing the number of cycles, we now show that we can do this amplification in about 80 minutes. And we have some ideas for even further decreasing of this protocol. Library preparation, it can be also done very quickly, 30 minutes with the kit that is supplied by Oxford Nanopores themselves. And the nice thing also is that you don't need a cleanup, but can progress to sequencing directly after. Then sequencing time, 2 hours currently. And actually there's 2 options. We can either apply the Flongle, which is, let's say, lower throughput, but for single samples, this is results in way enough data for the analysis. As alternative, you can also use the normal Minion flow cell. The advantage would be that there's more pores, so you can reduce your sequencing time. Disadvantages is that on one hand, it's more expensive. On the other hand, you generate more data simultaneously, so then the subsequent data analysis might be a little bit more tricky. Overall, I show the result of a few samples here, only for ABDR in this case. So first glance, if we look at all the quality parameters, there seems to be a lot of red. But this is because we applied the settings that we also would apply for Illumina typing. Ultimately, I think this is more important, that you see that in most cases the typings are concordant, except in this case for 1DRB1 typing. What we learn here is that this number of reads is apparently too low to give it a reliable typing. So we will increase the number of reads in the analysis. So overall, very brief preview. So this is data from 14 runs where 5 samples were included. And then if we look at A, B, C, and the RB1 typing, you see that the concordancy is over 80% for all cases. So currently, we're also working on including the DQB1 and DPB1 amplifications in this workflow as well. So in conclusion, I hope I showed you that at least that we have a proof of concept for fast typing, rapid typing using the Minion sequencer. Of course, this is all HLA typing before the transplantation. After transplantation, you would also like to monitor the success of the transplant. Typically, we do this by chimerism monitoring, which consists of 2 steps. So step 1 is genotyping. So finding markers, positions in the genome where the donor and the patient are different. So we can use those markers for transplantation follow-up monitoring, which is the second step. So over time, you use the informative markers to determine the percentage of chimerism. So you may know that for years we already have qPCR-based assays for this, but today I would like to introduce the NGS-based technologies and the NGS-based products that we have developed for this purpose. So they are called NGS Track for the reagents and Track Engine for the software. So they are presented on the right. You see that the reagents consist of amplification mixes, but also index plates. So if you would apply the product, it's, I think, very fast from DNA to sequencing in less than 2.5 hours with under 90 minutes of hands-on time. Also, data analysis would take less than 5 minutes. With a decent sensitivity of at least 0.5%, maybe below that, but that's not what we have evaluated so far. And some advantages of next-gen sequencing methods over the qPCR-based methods is that, first of all, the method can be combined with the NGS-Go HLA typing workflow. Second of all, a single pre-sample measurement is sufficient. And last of all, post and pre samples, you can just combine them in the same workflow in the same experiment. So currently we have developed a method to include 32 indel markers, which are also in the allele seeker or chimer product lines. They are distributed over 18 chromosomes and also include at least one XY marker. So all the mixes or all the markers are combined in 7 mixes, and this gives high quality results, as I will show in the next few slides, but also allows for more flexibility in case you might want to test only one of the 7 mixes. So how do the markers look like? We chose to develop and to look at biallelic indel markers with a fairly high frequency in the general population. And it looks a little bit like this. So we have, in some cases, the deletion might be present. So with the amplification, it will result in a short fragment. And in the other part, the insertion will be present, which results still also in an amplicon, but of a longer sequence. So this is different than looking at SNPs, and I would like to explain why. So with these 32 markers, we basically cover over 300 unique positions. But what's main importance is that I think the detection of indels is superior over the detection of indices. And that's demonstrated in the noise plots in the right. So here we mixed the 2 variants, the long and the short, at a 95 to 5% ratio. And then you can nicely determine in your noise plot of a regular sequencing run, here the indel marker, and here the single SNP position. And if we do a 10-fold further dilution, you can still see the indel marker here, but the SNP starts to be disappearing in the noise. And even if we go 10-fold lower, you can still nicely recognize the indel motif. So this shows that it can be detected very sensitive and also more sensitive than individual SNPs. So the workflow, very similar to any other NGS-based workflow. So amplification takes about 60 minutes. Also, the protocol takes about 60 minutes. We do indexing directly in the amplification. So the library prep is basically just a single beat cleanup, which is identical to the HLA typing cleanup. And as of there, you can combine the libraries. Then you start doing the sequencing. We validate it on Illumina paired end. And then lastly, we do the data analysis in Track Engine, which takes less than 5 minutes. So a very convenient, reliable workflow. That is reliable, I think, is demonstrated in this study. Here I present data from the UK NEQUOS, which is a, let's say, an external quality assessment study. So the samples are distributed over multiple labs, and then the average and the boundaries are presented in the graph over here. So this is the average of the studies. And then in blue, the results of our NGS track are plotted over there. So you see that they're all very nicely within the boundaries and almost exactly, in most cases, on the average result. So NGS track and track engine seems to be a very nice method for NGS chimerism monitoring with all kinds of advantages which are presented in this slide. What I would like to also, I would also like to give you the message that these products will be available as of December 1st of this year. So overall, I hope that I showed you in this presentation that based on the whole gene amplification of NGS-Go MX11 and 3, we've done this nice platform comparison study that you can nicely do HLA typing on this, on all kinds of platforms. Secondly, I hope that I showed you that we can have demonstrated a proof of concept for fast HLA typing for disease donors using the MinION sequencing, and ultimately also that NGS Track and Track Engine, our new products for NGS chimerism monitoring, can be used very reliably and very elegant. So with that, I would like to end my presentation and thank you for your attention.
Thank you, Dr. Martin. Very nice talk. But now we are open for the question and answers session. There are few questions in the chat box. Dr. Raja, if you can ask your questions first, and then I can read the chat box.
Sure. So I'm unable to open my camera. Can the host make my camera open?
Yeah, I think there is a problem with the camera. Even my camera is not getting on.
Okay, perfect. That's, oh yeah, it's working now.
You're okay.
So I'll start with Diane. So all three speakers did a wonderful job. So Diane, I think that's the Nano 4. Good to see you here.
See you.
Nano 4 technology people are expecting for the deceased donor workflow. So I think the big concern is the dropouts because the design is multiplexing. You put all the amplification in single tube. So what kind of validation you did to make sure there is no dropouts? I see some ambiguities like a DQ7 and DQ8 ambiguities, which we cannot accept.
Yeah.
So can you just elaborate about your validation plan?
Yeah, so we use the all type from One Lambda, the 11 loci. So that's the kit that we use, and we had been using in our laboratory for a number of years before we started this work. So we're very familiar with that that kit. We did obviously modify it to the PCR amplification time. So we did extensively. We have a DNA panel which covers all the allele groups that we run with any new validation. And so we tested that panel with the modified conditions and to ensure that we weren't seeing any dropout. We did do parallel testing. So we did confirm when we started doing deceased donor typing, we did confirm all the typing results from the flongle the next day with our ion torrent method. And we haven't seen any allele dropout since we've started using the assay. There is allelic imbalance, and so we see that with DQ2 and some of the DRB104s. So yes, you do have to take some time to edit, and obviously the homopolymer errors are an issue, and we can't exclude those positions. So we aren't able to exclude some nulls and rare alleles, but again, we still get a much higher resolution than we did with the SSO method. So yes, PCR robustness is important with MinION sequencing, but if you have a good PCR assay, then really the background issues aren't a problem. But you do have to be aware of the limitations of your own assay. We have developed our own in-house assay, and we see different things, obviously with amplifying different alleles. But again, we have the DQ2 and the DR. So like Martin said, DR and DQ are the most challenging we find with MinION sequencing off the R10. So hopefully that answers your question.
Oh yeah, there's a lot of answers.
Yeah.
So again, Dr. Mehra.
Can I also ask Diane? I mean, you said that you don't need any expensive equipment for this. You don't need any bioinformatics, and the chemistry is very simple. So what is the cost? Is the cost dependent on the
usually I don't discuss cost because cost varies in different countries, but I will say with Oxford Nanopore, the cost is what is advertised on their system, well, as far as we're concerned. So the cost is quite low. Again, the Oxford Nanopore technology is about $100 US, I think, and then obviously it depends on what PCR reagents you use. But it's, yeah, I think it's fairly comparative to the current available technologies. And off the batch testing that we're hoping to do, you know, that's well below the $100 US, at least in our hands, using an in-house PCR method. So it is cheap.
I wanted to know whether the cost will be lower if you do more samples, or, you know, since it's a 4-hour test, I was also thinking in terms of using it for the diseased donor transplant, where you can do the typing right away rather than depend on the retrospective samples.
Yeah. Well, we're doing it for deceased donor typing. So it is roughly the same price as what our previous method was. And for the little bit extra, if it is a little bit extra, you get the higher resolution. So we definitely think it's cost effective in that way. And the clinicians love it because it makes donor assessment easy because you have the 2 field result. So
Okay, we take your word.
Okay.
Dr. Dian, Dr. Dian, may I ask a question? There are always the challenges that for typing the class 2, when you are typing class 2, you face a lot of challenges, and even in NGS, although it was known from the serologist point of view, so you're having that kind of challenges. Although, can you explain how you can improve the class 2 typing on this? Because you are taking the challenge of typing deceased donor, where the turnover time is very less, and you have to report in the diagnostics setups. It becomes a more problematic. So how you can elaborate on that? That how we can improve the class 2 typing over there?
Yeah, so we only, for deceased donor typing, we only analyze the exons. We don't consider the introns, where we find most of the problems, to be honest. So with 2 field, we report 2 field typing. We use Gendex NGS Engine, and that seems to handle the MinION data fairly well. So we don't really have to do any manual manipulation. The only issues we have is at those homopolymer, where there's a homopolymer error, we'll have to do a manual edit and not include that in the analysis. So that just increases the ambiguity, but we haven't seen any discrepant typings or anything like that.
So then how do you report, Diane, when you have the homopolymers?
We report the allele that's used on the night during the matching, and then we say we can't exclude the other alleles.
So maybe I'll ask second question, maybe to Sandra Mauli's friend. So you have 4% of the samples, you have either warnings or you cannot get the typing. So can you give some insights? Is it a sample problem, or what is some?
Thank you for your question, Doctor. Actually, we got the data. Some of the data are a little bit difficult because the sample may be lysed, or maybe the DNA will be fragmented. So the second thing, it will automatically affect the analysis also. So it will automatically gives us low coverage. So if beyond the coverage, below low coverage, it will automatically we don't have the analysis chances to analyze the sample.
Okay, that makes sense.
Thank you. Thank you.
Again, then you want to go for the next one? Unmute, please. You can then unmute. Okay, I'll go with Martin. So I think that the Nano4 that use the PCR-free based Nano4 methods, is called MinION. I think that's an excellent approach. Now, if everything worked out, it's the big challenge in disease donor typing is, in just looking at for future, we want to see the typing done bedside. Someone become a potential donor, can you do the typing at the donor hospital site? Because it takes a lot of time to ship the samples from donor hospital to the HLA lab. So if we get it done within the bedside, that will be the excellent approaches. How can you envision this approach?
Yeah, I think I think that first of all, Diane did an excellent job showing that if you can do the disease donors in 4 hours, I think that can already almost be done at the bedside. Standing for quite a while at the bedside. But that's actually our approach as well. So, and that's why we have split it into 2 projects. So of course we are all focused always for whole gene HLA typing. But it really comes with different challenges and different project specifications, so to say, as doing the fast typing for disease donors. So we made, that's why we split the project. So we for the disease donor project on Minayan, we really focused also on we decided to go with the allele seeker amplification, which is more targeted toward the important exons and not the whole gene. And so I think 4 hours is definitely a possibility. Diane showed this already. We are in a proof of concept that it's possible. We want to expand a little bit on the genes, of course, like the DQ-DP. I think it can be a bit faster, but I think that maybe 2 hours is the maximum which would be achievable.
Yeah.
So is that bedside enough? Well, I think that's up for the clinicians to say.
Then you want to add something?
I want to ask Martin, how close are you to a product that you can handhold and put a drop of blood over there and typing comes into us?
Oh, that would be ideal, eh? Yeah.
Yeah, I mean, that's what I mean. I see the three of you, the three big sort of wigs over here, the Imicor, the GenDx, and Nanopore. I think that's what we need in times to come in there.
Yeah. Can I just say, we really need to get Oxford Nanopore on board, and the pressure to really, they've been around a while in the research space, but in terms of a clinical laboratory, really tailoring their products and to the clinical lab and helping us achieve what we need for HLA typing and human genetics. They're very good in other spaces, but so some of this does rest on companies like Oxford Nanopore to help us progress the technology for a routine clinical for use. So I'd say get out there and put some pressure on.
That definitely helps. And if I can also comment on the, let's say, the finger prick approach or the drop of blood. So we are looking at all kinds of ways to enrich for HLA, but without enrichment, it's not possible at the moment. So you will always need some kind of target enrichment, which will take 1-2 hours at a very minimal. So that's I mean, the library prep was half an hour, sequencing. Well, I think it can be easily done in an hour, half an hour. So that's not the main issue. The target enrichment, I think that's the most time-consuming step for a while.
So Rajalingam, I have two more. I want to ask one to Martin about his chimerism monitoring.
Yes.
And of course, these are all these are marketing words, quick and easy and sensitive. But I mean, how about the cost? The question is the cost. The NGS-based chimerism is very expensive. How do you bring down the cost? Because you are the technical guy.
Yeah, exactly. So the cost is also a marketing word. So it's no, but I think so.
Before you answer, can I also add to the same thing? So if the labs have a high throughput HLA but not a high throughput chimerism, how would you kind of fit into that? It's something what he's asking, and you can add on to this.
Yeah, exactly. That was part of my answer, indeed, because I think the main driver for NGS-based technologies for chimerism testing is, I think there's twofold. Both are technological. One is that most chimerism testing is done with STR testing. And of course, the sequencer are getting a bit outdated and getting a bit in need of more maintenance. On the other hand, you see that more and more labs are using NGS for also other approaches. And then there's really a search to have as many applications to run on the NGS platform. And this is also where you can do the cost saver. So if you are running NGS anyway for HLA typing, adding chimerism monitoring brings the cost really down. And in that way, it won't be expensive. It will probably be more expensive than STR because that's really low price. But I think that's the main way to bring the costs down. The fixed sequencing cost you cannot avoid, but we can add as many applications as possible in one run.
So the indices are different, and we can pull them in one flow cell?
You can pull them in one flow cell, yeah. So we use now similar indices, but that's why we have this plate-based approach, so you know which index combination is there, and then you can combine it with the HLA typing.
Thank you.
So my second question is to all the three, but I will reserve it after everybody else have asked.
Yeah, sir, I had a question for Dr. Martin.
Yeah, sure. Go ahead.
Yeah, Dr. Martin, what are the technical advantages of NGS based platform for chimerism over the conventional STR based typing for chimerism?
There's an interesting question, and that's there I have let's say a lack of knowledge because I'm not an STR expert. So what I do know is that with NGS based typing, it's really easy for to come to a conclusion. What you basically do in the software is you just count the number of long and short reads, and that's the percentage. Also, because the way it's set up, you look at multiple markers simultaneously. And I think that's what I understand the big hurdle in STR testing, that data analysis is cumbersome and not always straight on. So I think that's one big advantage of NGS over STR.
Yeah, so just I want to add one thing, Martin. So you found this Intel very useful to go high sensitivity, but this SNPs are diluted. What about the the tandem repeats? Because that's the best. People use it for the STR methods. So why the STR is not included in your NGS based methods?
No. So we have not included any of the STRs. So I can't say how that would compare to the Intel result. What I do know is that like homopolymers in HLA typing, that's a little bit challenging, and that's I think caused by the initial amplification. You will have some slippery of the polymerase, which makes the accurate length of the homopolymers not always accurate. I think something similar may happen to STRs. A colleague of mine, Erik Rosemuller, you probably know, he did a study a few years ago where he looked at the STRs in homozygous DRB4, DRB5 typings. And then he showed just by doing the initial PCR, you already get a variation in STR length, even though you know that the input was really homozygous. So that's a disadvantage that well, you will not see with the indels because you really look at one indel of fixed length.
So we'll take one question from chat box, then Uma can go ahead. So this is from Dr. Mamidi Niraja. So how many samples can be pulled for one run in MinION sequencing for HLA typing? I guess it's for Martin.
So depending on the purpose, I think for the whole gene HLA typing, we use the regular MinION. Currently, we pull 8 to 12 samples. For let's say the bedside typing approach, we use the Flongle, which has less throughput, and we typically do one sample at a time at the moment. I'm not sure if that's the same experience as Diane, but
so on the Flongle, we're actually just working, looking at barcoding, and so we've been able to put 4 samples on the Flongle, and that's mainly to use that for urgent samples that might come in during the day, not for deceased donor typing. And on the R10, we find that if we pull 24 samples and then run that for 14 hours and then load another 24 hours of samples and then run that for a further 14 hours to get better results. So 48 samples on a flow cell for class 1 and 2. That's probably the limit.
Okay.
Yeah.
So one more quick question from chat box from Ramona Chopra. So what is the extra advantage of NGS chimerism? Again, to Martin.
Sorry, I missed the first bit of the question.
What is the advantage of NGS chimerism?
Advantage of NGS chimerism over over STR, I think we kind of discussed. STR is very cheap. I think NGS you can combine with all kind of other NGS applications. I think advantage over qPCR based assays is that you only need to do a pre-typing once. With qPCR, you have to have a pre-sample with every determination. Yeah, I think it's mainly, I think there's a lot of technological advantages over the, especially over the STR technology.
I would say it's everything in one flow. It's just one NGS workflow, typing, and the
Yeah.
chimerism.
Yeah.
Uma, please
Yes,
go ahead.
just
Yeah.
wanted to add to Yogendra's comment that he asked about the NGS-based chimerism. I think the informative markers are much more in this NGS platform than what we have on the STR. So that was something that we noticed. And I had a request to Diane and Martin that if any lab wants to set up the MinION, would you be able to handle also with the software and the web-based interface? Would you both be able to handle other labs and get it sorted?
Do the analysis for you, is that what you mean?
No, just help us establish the
Help us establish, yeah, absolutely.
Because that's something new, but we haven't started off, and you know, we can learn from your experience and Martin also. From that. Thank you.
No, I must say here, Martin, that we had developed the STR-based markers ourselves in our own laboratory. And at that time, we were struck that we created our own. I think they were population-specific informative markers. So, if you have a larger number of them, the cost will go up, but they may not be relevant for a particular ethnic group. So maybe I think there's a need for population-specific, because otherwise you have hundreds of them, and many of them are of no consequence. The markers that may be informative for the Caucasians may not be all of them useful for the Indian population, for example.
Yeah, no, that's certainly something, and I think regarding STRs, it's easier to include population-specific additional markers if needed. That's definitely one thing.
If my colleague Dr. Gurvinder is here, because she and we really worked very hard to sort of shortlist some of those, and we were then playing with about 6 or 7, and they were giving the kind of information that we wanted at the pre-transplant stage and the post-transplant. What is more is also relevant here is that she was trying to extract the DNA from the hair and from saliva. And the pre-transplant, the pre-HSCT DNA of the hair was of the original host. And because in the blood, of course, in the post-transplant, it will change. So I think the DNA of the hair is very, very important. And that paper is already in.
But I mean, so the NGS-based chimerism still just to be validated in clinical utility because it's too sensitive. Sometimes you may see an extra chimerism in women with the pregnancy, multiple pregnancies. They may have chimerism from the baby. So this also is too sensitive. So people need to validate. It's attractive feature is a simple workflow. So you put everything on NGS. Then also it is a possibility of more automations. You don't need to have a cumbersome STR analysis. The computer can throw the numbers. But I think the clinical still needs to be determined.
Yeah, I think there's a question from John Downing. You can read John Downing's question to Martin.
Yeah, there is a question. Yeah, he says that you don't discuss about KIR genotyping, but there are plans. If there are plans to adapt any NGS engine to detect more than 2 alleles per locus, as happens occasionally.
Yeah, it's an interesting question, but it's not that simple. Of course, NGS Engine was developed as HLA typing tool, assuming that you will have for each allele a maximum for each gene a maximum of 2 alleles. We know indeed that KIR is more challenging. It's not only duplications, but also inversions, everything. So it is indeed more challenging. So we are looking at that. It's not easy. The good thing is that you typically can recognize in the data that there is a third pattern. You're not yet able to solve what that third pattern is yet. So we're not there yet, but our team is developing tools and also using internally to determine multiple patterns in one sample. So it is work in progress, but it's also a fairly complicated project, I have to say. So it will take a bit for it's publicly available. Nevertheless, if you have a difficult sample, if you have a sample where that you suspect is multiple alleles, we can always help you with the tools that you have internally. So that's what I can offer at the moment.
Yeah, Martin, I am interested in knowing the sensitivity of your NGS-based chimerism analysis as compared to the STR in terms of fold, or maybe, you know, whatever way you can explain. Can you just please explain?
About sensitivity?
Yes.
Yeah, so I think so what the pictures I showed and the 0.5% that we determine, that is based on just analytical performance studies. So where we apply cell lines, dilutions of cell lines. Also, we did some very early studies to compare it with qPCR sensitivity. So it's all, let's say, in artificial systems. So I think what is important now is that there's real clinical studies starting to be performed, also to demonstrate that the sensitivity is true in clinical samples,
yeah,
but even more that it's also meaningful, like what was just discussed before, that it's a real chimerism and not from a pregnancy or whatever. So I think that would be the real proof of the pudding, to say that this is real in a clinical setting. But like said, from analytical performance perspective, so on cell line based material, it's real that we can reach this sensitivity.
Yes, and in terms of cost effectiveness, how you see NGS over STR?
Yeah, it's difficult. So for me to give a good answer, because like we said, I don't know too much about STRs. I just know it's cheap, and the cost is really driven also by the sequencing itself. So what do you do in one sequencing run? For the rest, it's basically an amplification and a beat cleanup. So it's really driven by the NGS run that you would need to perform.
Thank you.
Can I ask Martin one more question? Martin, as a lab that does registry typing, is Gendex doing anything in the ABO space? We've been typing on the MinION, or have you done anything with ABO?
No, not yet. Not yet. No.
Thank you so much. If no more questions, we would like to close this session. Thank you, organizers, and thank you, speakers, for excellent talks. Over to you, Akanksha. Thank you so much.
On behalf of the organizing committee, I would like to thank all the moderators and speakers for conducting the session so smoothly. Now, next we have a session on 18th International Histocompatibility and Immunogenetics Workshop, which will be conducted by Dr. Uma Kanga. Over to you, ma'am. Thank you.
Thank you, Akanksha. I think this workshop session will be led by Eric Sparings. He's joined us. And I would like Eric to please talk to the delegates about the workshop, which is really very relevant for all of us.
Yes. Can you all hear me now? So good afternoon, everybody, and thanks for joining me with this last session.
Hey, Eric, there's a lot of noise. A lot of background noise.
People all need to mute themselves except Eric.
Dr. Eric, can you please disconnect your audio and connect again, please? Dr. Eric?
Yeah.
Please disconnect your audio and connect again, please.
Audio. Manas, after that, we will play the video.
Is it better like this, or?
No, Eric, he wants you to log off and log in again.
Okay. Out of Zoom.
So we have the workshop video, which we will play after Eric's talk. and also a promotion video on the FE meeting next year. So these are being shared by Dr. Eric and Sebastian. So we'll wait for him to speak and then we'll put that on.
The video can wait. So let
Yes.
Eric come back and he has slides, right?
That's what we requested him to speak a few words about the workshop. So
Let him.
Yeah, we'll wait for him.
Eric, you have joined in. Could you unmute and go ahead?
Is it better?
Yes.
Yeah.
Yes, okay. I will log in because I'm now connected with my phone. I need to for the audio and I need to reconnect for the 1 second.
You want to share slides?
Yeah, I need to reconnect with the Zoom. So it is.
Eric, if you want to email slides, the technical team can display here. What's your convenience? Let us know.
Almost there.
Great.
Recording in progress.
Dr. Eric, please put one device in mute mode and another device unmute.
Okay, can you still hear me?
Yes.
Yes, okay. And you can see my screen?
Absolutely.
Yes, that's like this.
So we are seeing two Erics.
Yeah, that's right.
That's okay. Sound is coming from one, so it should be fine.
As long as you see my screen and you can hear my audio, then we're good to go. Okay, I think we are ready to go, right?
Yeah, go ahead.
So sorry for all the fuss and thanks for joining in with this session on the workshop. And I think it's also, I'm really grateful to the Ishi and Aphia organizers to offer this platform to update you on the 18th workshop. I will, in this session, briefly introduce you to the workshop for those that are not that familiar with it. And I will focus a little bit on one of the components that has our interest in the 18th workshop. And after that, Ben Matern has pre-recorded a small presentation in which he will elaborate on the data standards in the workshop and also beyond. And after that, I will get back to you with some information on the joint conference that the workshop will organize with FE. So first, the workshop introduction. In the short history of HLA that was written by Thorsby, he recognizes the importance of the contributions by outstanding pioneers in the field of HLA, but he also mentioned that the field has significantly advanced, particularly by the extensive collaboration that we have in our community. And he stresses the importance of the International Histocompatibility Workshop in these international collaboration. The workshops were initially organized to solve issues in histocompatibility and immunogenetics that required such a collaboration. And as such, workshops involved and still involve the exchange of reagents, the promotion of new technologies, dissemination of technical skills, and also very importantly, the definition of a common nomenclature. The first workshops were organized by people like Bernard Amos, John Van Rose, Rogeri Cipollini, and Paul Terrazaki in the early 60s. And they were leading these first workshops. And in these first workshops, the focus was on comparing and exchanging the assays and the reagents, but also cell panels, and on, as I mentioned before, categorizing antigens and constructing a nomenclature. And for instance, mind the presence of the W in the HLA-C nomenclature that is still a remnant of the workshop efforts. The 18th workshop will run until spring 2022 and is being chaired by Sebastian Heid from the Leiden University Medical Center and myself. And we are supported by the Dutch HLA community, the HLAWN, by the International Workshop Counselors, and by various project leaders. Basically, the 18th workshop has the same key components as the second workshop. And given Sebastian's and my own interest in epitopes, it won't surprise you that we want to give special attention to B cell and T cell epitopes, so the transplantation antigens. Various studies on the B and the T cell epitopes have shown that HLA is not a repertoire of antigens to be compared, but that all HLA antigens can be regarded as composed of epitopes. So a set of epitopes rather than just a nomenclature name. And that means that for matching and mismatching, it is more effective to compare these epitopes than these names that we assign to these antigens. And when we compare epitopes, it has become evident that the level of epitope mismatches is related to the risk to mount an immune response against these HLA mismatches. And that is indicated here by an example for B cell epitopes, where you see that the number of B cell epitopes is related to the chances to develop HLA antibody pregnancy. So while we are now all aware of these aspects, there are still some issues that block clinical implementation of the epitope concepts. And particularly the definition of epitopes and the identification of the truly immunogenic ones is an important aspect. These definitions and the identifications can only be done when we would use large data sets. And of course, we need to cover multiple populations to make it a universal concept. And this workshop is an ideal platform for getting these data through the collaboration and to analyze all these aspects in great detail. So we have two basic key projects, and one of them is the identification of the immunogenic epitopes. And this project focuses on non-immunogenic individuals that got immunized or remained free of HLA antibodies for at least 5 years. And the second one addresses the non-immunogenic epitopes in heavily immunized individuals. So in these individuals, we try to find out why certain mismatched epitopes did not result in an immune response. Data from these two projects will also be used in a project led by Lloyd Sonia and Anat Tambour to elucidate the HLA-DQ immunogenicity with a special focus on situations where we have two DQ mismatches, but where we only see a response towards one of them. The timeline for the epitope component is rather simple. We already have constructed a database for data collection, and Ben will tell a bit more about that. And we get data in and are preparing for the first analyses. The data collection and the analysis will go on until the workshop meeting next year in May. And you're all encouraged to join these projects, and feel free to get in touch with us if you have questions on how to do that. We got massive support from the various companies for reagents, and so you can get these reagents and do it yourself. And alternatively, samples can be sent to us or other laboratories to have the analysis done. So you can find more information on the workshop website, and it's important to mention that we recently opened the registration for the workshop meeting. please register, of course. As mentioned, the workshop meeting will be followed by the joint workshop Aphia conference in Amsterdam in the Netherlands. And more information will be shown after the presentation of Ben Matern, who will now present information on the workshop database and the HLA data standards.
Thank you, Eric. Does anyone have any query from Eric on the workshop related? Or we put on the video that Ben is going to talk to us on the database? If not,
Eric, I was just wanting to know your plans of organizing in May of next year, and hopefully COVID will go down. But at the moment, the situation in Europe is rather bad. And, we've seen that, particularly in Holland and Austria are the two countries in Europe where there's a steady rise in number of infections. In fact, what I see, and I follow COVID very closely, because of my new interest in COVID-19 immunity. The peak is rising very fast in Holland. I hope everything is all right, because most of us and all of us, and I would certainly like to attend it in person there rather than online.
Yes, yeah, that's a serious concern indeed. We see that COVID peaks are season related. I don't have I can't read the stars, but we certainly hope that we are getting down in March, maybe even earlier. If well, we will certainly have a meeting. There will be access after testing and vaccination screening in worst case. There are, of course, travel limitations that could hamper you coming to the Netherlands. So we are working on hybrid situations for the workshop meeting as well. If you subscribe to the meeting and there is a reason that you in the end cannot come, we will reimburse the registration fee. So there is no a problem with that. But of course, we all cross our fingers. We cannot read the stars, as I said, but we certainly are looking forward to really meet each other in real life again next year. Yeah, let's hope for the best.
Yeah, let's hope for the best, because I feel that maybe by the end of the year or early January it should go up. I mean, there's no solid data to indicate any relationship with the seasonal change, although a lot of discussion has taken place on that. I think it's just that everybody's opening up now, and there is a lot number of unvaccinated lot that is going around. And the vaccine hesitancy is all over the world, and that might shoot up the numbers of infection. But I think the death rate is not showing up, is it?
Well, yeah, we can discuss COVID right here. What I meant with seasonal peaks is that people being more indoor in winter time and contaminating each other more easily than in summertime. So that's what I meant with seasonal relations to that. But let's wait and see. And certainly, well, there's a need to get together, and we're working hard on that. Let's hope for the best. I'm optimistic.
Yeah, optimistic too. We wish to be there. Everyone would like to be there globally. Yeah. So can we go ahead and put on the video that Ben is going to tell us? Technical team, can I request you to please put the video that was shared by Eric?
Yes, ma'am. Just
we do have a few queries on the database, but we can email you, Eric.
वीडियो दिखा रहा है।
Ma'am, unmute, please.
I think he played the promotion video before the database. So let's have the database video first and complete our international workshop discussion. Sorry about that.
And thanks to the organizers of the Ishi Aphia Conference. My name is Ben Matern, and I've been working on collecting data for the 18th International Workshop. So we've been creating a database to collect information that can be used in many projects related to HLA and immunogenicity. And I'd just like to briefly show you some of the things we're thinking about and some of the ways that we can effectively collect data in a smart way. So the workshop goals are divided into 3 main components, and each component here has lots of projects with varying goals. There are projects focused on, for example, immunogenicity or defining haplotypes or family and inheritance studies. And because there's a variety of goals, means there's a variety of types of data to collect. For many of these projects, we need to collect HLA genotyping data. This might be from NGS sequencing or one of the lower resolution methods. We might also want to collect some HLA antibody data from single antigen bead assays. We might want to collect some raw sequencing data directly from the sequencing device. And we might also want to collect something that's project specific. Maybe we're looking at the outcomes of transplantations or looking how haplotypes are arranged or inherited in families. So with this variety of goals, it's necessary to define standards so we can collect this data in a smart way. Our main tool for collecting data is this workshop database. This is a website. It's hosted on Amazon Web Services. And this is a tool that workshop participants can use to upload data. And this data from around the world can be combined and analyzed. And we can use this data in a collaborative way to accomplish some of these workshop goals. So, like I said, there's many types of data, and they need to be handled in individual ways. So we came up with this step function architecture. This step function is a computational model that's run automatically on our uploaded files. And an overview is shown in this tree diagram. So what this is showing is when different files are uploaded, we can process them in different ways. And that's based on the format the data is in, or what information it contains, or what project it's intended for. So probably the most relevant type of data that we're working with is this HLA genotyping. And for this, we're using the histoimmunogenics markup language, the HML format. So I'm showing you here an example, a small piece of HML on the right side. So the HML format allows us to send and receive HLA genotype results, the actual allele calls. And we can also include all of the interesting metadata about how it was created, the sequencing platform or the reference sequence that your data was compared to, and any novel polymorphism that was found. The format for HML is well established and well defined. It's currently in use at NMDP Be the Match, and it is defined as our standard format for all workshop projects, and it's also being implemented for use in Eurotransplant as well. We've been in contact with many software vendors to implement this as an export format in the software, and I saw that several of these vendors are participating in this Ishi Aphia conference, so I would like to thank them for all the effort they've made in getting that implemented. HML is a very flexible format, so we need to define a few sets of standards and guidelines for how to use it in an effective and clear way. And the HLA community members have come together to define some sets of rules to help with this. One example is GL strings, which is a way we can represent HLA types in a text format, and that can include information about the phasing of HLA genes in a haplotype or any allele or genotype ambiguities. We're also thinking about miring, which is a checklist of information that should be included in a genotyping result so we can understand where these allele calls come from. We've also worked on defining a set of standard reference sequences because with NGS genotyping, it's important to compare your results against the well-defined, the full-length, and somewhat common allele in the IPD IMGT HLA database. And that can help us further understand the result. These data standards are the kind of things we're thinking about when we're gathering data in the workshop database. So when individual files are uploaded, we are checking the data for some of these standards. We try to provide some feedback on whether or not the data fits these standards and maybe give suggestions on how to improve the data. I would point out that this feedback is intended to be helpful. It's not necessarily saying that the data is accepted or rejected, because it's actually kind of rare that data completely passes all of the validators, and even if the data is imperfect, it's probably still usable. So we would encourage participants to submit even if there are some warnings shown. I want to re-emphasize that these are community standards. We didn't invent all these concepts on our own. Many of these standards were defined at these data standards hackathons, these DASH meetings, which have taken place a few times each year since 2014. At these DASH meetings, researchers, clinicians, and vendors are invited to come together to discuss these concepts and come to consensus. And also for the software and bioinformatics people like me, we try to create new tools and improve the software for handling these data. The next DASH meeting is planned. It's online for this coming February 17th and 18th, and the focus is on preparation for the 18th workshop. If anyone in the audience is interested in joining these discussions, you're certainly welcome, and I'd be happy to help put you in touch with the organizers of the event, or you're free to contact Martin Myers directly. So I'm just finishing here with a few links. You're certainly encouraged to visit the workshop website to learn a little bit more. If you want, you can use the IHIW database when you're ready to upload some data. And of course, you can follow this GitHub link to learn a little bit more about the DASH hackathon. And if you wish, you're free to send me an email in case you want to get in touch. So thank you for your time.
Okay, so Eric, if anybody has questions, would you like to take that on the database or?
I will do my best, yes.
So if anyone has any queries and are submitting data to the workshop and would like to know something from Eric, please feel free to.
Yeah, feel free to contact us anyway outside this meeting if you have questions or specific comments. What Ben mentioned that's important to stress is that the next DASH is quite an important meeting. Also for the vendors, we will actively involve them. We had a great help from them in the past months, and we're really moving forward with the HML standard being implemented in many software packages right now, which is, I think, a great start to synchronize data exchange formats and being able to exchange data between labs at a higher detail than we did before.
So Eric, this Ben Mattern, is he an expert in bioinformatics?
Yes, he has a connection in the very past with NMDP. Has been working with Marcel Tilanes during his PhD studies, and with a special focus on also the bioinformatics. So he's a good guy. Yeah.
So he has been working in the HLA lab earlier with
Yes, yeah.
Therefore, he understands the whole genomics of the HLA system.
The complexity of HLA. Yes, he's very well introduced into that. Yeah.
Okay.
Thank you. And I think Eric also wants us to play the FE promotion video. So we will.
Yes, please. Try again.
Let everyone know about the FE conference also that's going to be there. So can I request the technical team please put the video? Thank you. Thank you. That's really exciting. Eric, you want to say something more?
No other than to invite you all to the FE meeting. Abstract submission is open, and submit your abstract, please. Subscribe, join. Let's meet again next year. Thank you.
How far is the workshop venue from the conference venue?
We're a pretty small country, so it's something like 30 kilometers away from it. We will provide transportation for all individuals that join the workshop from the airport to the venue and from the venue to the conference. So that's all included in the fee.
Okay.
Thank you, Eric, for joining us for the session. Thanks a lot.
You're welcome.
So with your permission, sir, we go ahead with the rest of the closing ceremony.
Yes, I think now we come to the most important part of the conference, closing ceremony. We have had 4 days of very extensive scientific conference. Very important. I'm so glad that it happened. And how many people are there in the platform on today, Manas? I see 37 over here, and how many?
Sir, 15 on YouTube and 86 on platform.
How many?
86.
86 on platform and 15 on YouTube.
Okay.
And 37 here. Yeah.
Good. All right. So we go ahead with our closing ceremony. The two secretaries will announce the awards that we had said that we will give for the oral. There were 2 oral sessions and 1 poster session. So go ahead and announce the awards. There are all those youngsters are looking forward to it.
I hope that all the youngsters who presented the 3 days are available here, and we will soon announce. On behalf of the organizing committee, first I thank all the moderators who chaired the oral presentation and the poster presentation sessions. Thank you so much for helping us run the sessions. And I'm very pleased on behalf of organizing committee to announce the award. We had 2 oral awards for the day 1 oral presentations and 2 for the day 2. So I will go with the day 1 first, not in any series. There are 2 awards and they will be contacted by the organizing committee. A certificate will be sent to them and a cash prize of ₹5,000. They will be contacted and their bank details will be taken and the funds would be transferred by our treasurer accordingly. So I'm now going to announce the names. The day 1 oral presentations, we have the abstract number 59, presented by Vidisha Payal, a rare case of anti-U antibody alloimmunization in antenatal patient. So congratulations, Vidisha. And if you can put on your video, and we will also call the second, the other awardee is Dr. Selma De Silva, and she presented abstract 38, validation of allele level high resolution KIR genotyping. So both of you.
This is Vidisha Payal. Oh, good.
Vidisha. And Selma is also here.
Yes, ma'am. Thank you.
Selma is also here. Okay.
So, sir, can you just bless them both? Your comments, sir.
Weekend for you. Have a good day.
Thank you so much, sir. Thank you, sir.
Congratulations for your hard work. Yeah.
Congratulations.
Thank you, Doctor.
Congratulations. So we move on to the day 2, and we have the day 2 oral presentations award. And again, we have 2 awardees. Number 1, the abstract 39, was presented by Akanksha Sharma, long non-coding RNAs as molecular signatures for acute renal allograft rejection. Akanksha, are you here? Akanksha has also been anchoring the sessions for us. And the other abstract is abstract 28 by Dr. Bisu Singh, which was on HLA profile of Indian Kami population, shows a close genetic affinity with the North Indian Indo-Aryan population. So Akanksha and Dr. Bisu.
Thank you, ma'am. Thank you. Thank you, ma'am. Thank you, sir.
Thank you very much. And
Thank you, sir.
Thank you.
Congratulations and keep up the good work. Both of you are still very young, and set up your goals in life. and continue the good work.
Thank you, sir. Your words means a lot to me, sir. Thank you, sir.
We are really proud that our Indian delegates are presenting well, and the moderators appreciated your work.
Thank you,
ma'am.
ma'am.
Thank you, sir, for the appreciation. Thank you so much, ma'am.
So I now request my other organizing secretary, Dr. Varsha, to please announce the poster awards. You have the details.
Thank you, Dr. Huma. The poster awards presentation number 5. 3 awards we are going to give, and the Nehi Nuo Chung Ho from New Delhi, India. Characterization of GAD65 epitopes possibly presented by type 1 diabetes associated with HLA-DR3 and or DQ2 molecules. The second way,
1 minute.
third one is
how do you speak your name?
It's Ni He Nyo Chizho. Ni He Nyo
Ni
Chizho.
He No.
Yes.
I don't
Yeah.
think Ni Hao.
What part
Ni
of
He
India?
Nyo.
Ni He. Okay.
Do you come
Yes. Yes. It's in Naga language, so it's a little difficult. Yes.
You're from Nagaland?
Okay.
I'm from Nagaland, sir.
Very
Yes.
Very nice.
Congratulations.
Congratulations. Okay.
Thank you so much, sir. Thank you, organizers. Yeah.
Poster presentation number 7. Vichaya Runenjaya Ime. I hope I have pronounced names. She's from Nonthaburi, Thailand.
Nonthaburi. Nonthaburi is a very famous place in Thailand.
Oh,
Okay. Yeah.
Is she around?
is she on the platform here on Zoom? Vishaya?
No, sir, no.
We had emailed all the presenters to be available for this session. I hope, anyway, we will send the email and communicate to her if she's not around.
And the third one is Pompayo Sorsai from the Bangkok, Thailand. Is she around?
I think she's also not there.
Okay. Congratulations
Yes,
to all.
she's here. She's
She's there. Why don't you put on your camera and come on? Yeah, she's there.
Yeah, I'm here. Thank you.
Okay, congratulations.
Thank you so
Pimpayo,
much.
Pimpayo, what hospital in Bangkok are you from?
Sorry, my internet is.
Which hospital or which institution of Bangkok are you from?
Oh, Chulalongkorn University.
Chulalongkorn. Oh
My PI is Natya Hirangan.
Natya student.
Yes.
Yeah, I have been there earlier. What was her name? The earlier person from Thailand. I'm forgetting her name.
Chanvit.
Huh?
Chanvit?
Chanvit? No, the other girl. Oh my God.
From
was
the Faculty of
Dasanayani-
Medicine?
One was Dasanayani Chandana Ying Yong,
Yeah.
and the other one,
Pimpun.
very famous,
Pimpun?
Professor Kiet? No?
Oh. Anyway, doesn't matter.
Hope to see you here.
Congratulations. Yeah.
Congratulations.
Thank you so
Once
much.
Congratulations.
I-
Once
I-
again, thank you very much for making this conference.
I have one
Success.
request. I have one request to the President Ishi. Sir, these were
Yeah.
scores given by the moderators.
Yeah, we have given
Can
4
I ask-
awards for oral, and I wanted to match up 4, although we told the judges for 3.
Yeah, this is what I was going to ask you.
You might call it an additional, you might call it consolation, you might call it presidential award, but I was impressed by the work done by this young little girl, Akanksha Pathak. So so we announced one more award for Akanksha.
Akanksha Pathak, are you on the platform?
Yes, ma'am.
Okay, why don't you put on your video and also join other awardees?
Akanksha ji, aap kahan hain? Oh, there you are.
Okay,
How old are you? How old are you?
I'm 20.
26. Very nice. So you work
So
with
23.
23. Oh,
Yes, sir.
then you deserve even more consolation. Thank you very much. And we are very happy to also include you in the list.
Thank you so much, sir.
This is just to encourage another youngster and to be in the field of histocompatibility and immunogenetics, because
sir.
the future lies on your shoulders.
Yes, sir.
Thank you.
Thank you so much, sir. Thank you so much, ma'am.
So I would like to inform all the awardees that the organizing committee will contact you for further distribution of certificates and your award money. So please respond and send the details accordingly. I now request Dr. Gaurav Sharma and Arishma Lata.
No. Closing
Sorry.
remarks by me.
I'm sorry. I'm really sorry.
Heather has joined. Heather
is here.
9:30 p.m. for her, but it's nice of her to join. Heather, are you around?
Heather?
Yes, I'm here.
Yeah.
So Heather,
I invite you both, so the presidents of the societies, Ishi and Aphia, to please convey your closing remarks for the meeting.
No, it's not the presidential remarks, it's just the remarks that we are very happy that this conference has gone through. We went through some very difficult time during the organization phase, whether we should do it or not do it. And as you know, that India was experienced a very aggressive second wave in April, May, and it went up to June. And then after that, we thought that we must do it because we were not able to do our joint conference in 2020. And it was very important that we do it in 2021. The challenge for us was the huge time difference in the Aphia region there. And we wanted to use this opportunity to invite as many speakers of eminence from around the world as we could. And I think all of that was very successful. We are very happy that we could have speakers from North America, Europe. Europe, of course, when we were starting at 8 a.m., it was too early for them. So we had another challenge, how to accommodate them. So, but I think at the end of the day, I'm so glad that the feedback that I am hearing from a lot of people is that it was an academically a very high standard conference and academic feat. Academic feast for you guys. And I think our goal was to make sure that all aspects of MSc are actually covered. I may also announce, and as we discussed in our GBMs, some of you were not there, that we are planning to propose holding the 19th International Histocompatibility Workshop in India. So we will send our bid, put our best foot forwards, and hopefully there will be support from Ishi, there will be support from Hong Kong, there will be support from Aphia to our bid. And I'm sure if this bid gets approved, because it has never happened in Asia, the workshops have mostly been held in Europe or North America, only a couple of times outside of that. And it is, I think, time in 2025 that we should come to India. And I'm particularly very happy to see that there are a lot of youngsters all over India. There was a time in the 70s when there was only one lab all over India, or maybe 2 or 3. But now I see that the field of histocompatibility and immunogenetics has spread all over India and all over Asia. So I think Asian continent does have a claim to have the next workshop here in India. So with this, I want to really thank each one of you. I want to thank all the speakers. I want to thank all the moderators who really came about, and whenever we asked them to come over, they were available to us. And I think that was wonderful from our point of view that they helped us. I also want to thank the executive of Aphia. I call it Aphia because that's the way we sort of speak in India, and the Australians call it Aphia. But I mean, Aphia is what we speak in India. And I want to thank Heather and her team for joining hands. And I think this joining hands proved out to be very, very useful. Thank you very much. Over to you, Heather.
Thanks, Narendra.
Yes, on behalf of Aphia, I'd like to say a very big thank you to you and to Uma and Manus and all your organising team for organising this conference and running it all. I think it's been a great success. I thank you for all that you've done behind the scenes, which people don't see. They just see the front of it and get to hear the talks, but it's been a lot of work behind the scenes. So thank you for all of that. I think it's been great. And definitely want to thank all the invited speakers. We've heard so many great talks over the last few days. So informative, so educational, and I'm sure it's challenged a lot of us to go back to our labs now and think about what we've learned and if we want to change things or implement new things or do research, because we've been challenged by what we've heard. So it's been really good. And also I've been very impressed with all the local presenters, the abstract presenters and poster presenters. It's great to see so many young people coming through in both the Ishi and the Aphia community and presenting their work. So that's really great too. And also, as Narendra mentioned, to thank the moderators for their time. And we know that these sessions have been going on at different times across the world, depending where we live. So I appreciate all the work that the time that the moderators gave for that. And of course, our sponsors, you know, we can't run any meetings these days without sponsorship, and we appreciate all of that. I think we have ImmuCore, CareDX, GenDX, BAG Diagnostics, SRL Diagnostics, and Premas. So thank you for all of that. So thank you. This has been a great virtual meeting, and hopefully in the future we'll actually be able to get back to some in-person meetings and see people face to face again. But this has been a great virtual meeting. So thank you, everyone.
Thank you. Sorry.
Yeah, no, go ahead.
Thank you, Dr. Mehra and Dr. Dunkley for the kind remarks. I now invite our joint organizing secretary, Dr. Gaurav Sharma and Arishma Lata to propose the vote of thanks.
Yeah. Namaskar, everyone. On behalf of the organizing committee and team, it's my privilege to deliver this vote of thanks along with Dr. Arishma. First and foremost, I would like to express my gratitude towards my mentor and Ishi President, Professor Narendra Mehra, for giving me this opportunity and the responsibility of Joint Organizing Secretary. As the chairman of Ishi Aphia Conference 2021, he has been instrumental in supervising organizing team for formulating the theme-based program and diverse sessions. We have been fortunate to bring together a galaxy of high-impact, internationally reputed speakers covering diverse topics in theme-based manner. This could only become possible through persistent efforts of Professor Narendra Mehra, along with Dr. Uma Kang and other team members. Let me acknowledge Ishi Secretary Dr. Uma Kanga, who coordinated with Aphia as Aphia Vice President for finalizing the program, and also got the American College of Histocompatibility and Immunogenetics approval for credit points for this academic activity. We sincerely appreciate the concern of Aphia President Dr. Heather Dunkley and her associates, Dr. Arishma, for coordinating promptly in real-time manner and their valuable suggestions. Dr. Heather has always been supportive with her ideas. A special thanks to our organizing secretary, Dr. Varsha Trivedi, for being very helpful with her original ideas and valuable suggestions. I would like to acknowledge Conference Treasurer, Dr. Yoginder Pal Singh, who played a key role in promoting the conference and financial management. We thank all the conference speakers for their esteemed talks and most advanced and high impact details on diverse topics related to the field of histocompatibility and immunogenetics. Our thanks and congratulations to Professor Narendra Mehra Oration Awardee, Professor Effie Peterdorf, for delivering an excellent thought-provoking oration this time. Further, our experienced moderators played key role in not only regulating the sessions, but also encouraging vibrant discussions. I am running short of words for my humble thanks to all the judges for evaluating oral and poster presentations. Indeed, it was a challenging task to opt the bestest among the best lots. So we take this opportunity to thank all the delegates for actively participating, their great scientific inputs, submitting abstracts and posters for their recent work. And they encouraged vibrant and thought-provoking discussions during last 4 days. We acknowledge the kind voluntary support from Dr. Selma, Ms. Akanksha, and Ms. Tanvi for introducing the moderators. I must acknowledge the sponsorship from all the companies, Immucor, KRDX, GenDX, BAG, Primas, and SRM for supporting this high impact academic activity. Last but not the least, I must mention that the conference secretariat was handled very efficiently and professionally by the meetings and more, including Mr. Gaurav Quatra, Mr. Manas, Mr. Jia, Mr. Prateek, and others. The team played a key role in registering all the delegates, speakers, and moderators. They interacted with them and shared updates in real time. They collected their information also. Mr. Manas has been phenomenal in overseeing PowerPoint presentations and helping with logistics. I am delighted to conclude that this conference was a great success. And if I missed somebody, I apologize for that. With these words, I stop here and invite Dr. Arishma to express her thoughts.
Okay, thank you, everyone. I think Gaurav has covered quite a bit, so I'm going to keep mine short and simple. On behalf of my fellow Aphia Council, I would like to express my gratitude for providing this joint meeting with Ishi and Aphia. I think this was a great success with a very high level of education. All the sessions were fulfilled with the requirement of Aphia, which was education in histocompatibility and immunogenetics field. I extend my gratitude to the speakers, moderators, delegates for variety of exhibitions, e-posters, oral presenters, and finally to the organizing committee for such a successful virtual conference. Thank you, everyone.
Thank you very much. Two participants had called me that they want to say something. Ajay and Anil.
Thank you, sir. I think you all will agree that we need to give our appreciation to the organizing team for putting up an excellent show, which included extraordinary presentations covering all aspects of MSc and gripping discussions that it generated. Cheers to organizing team, and let us all clap together. And also let us vow to work together to bring the international workshop to India. Thank you, sir.
Thank you, Ajay. Thank you so much. You are a part of the team, Ajay.
Thank you.
So, let me also take this opportunity to echo which almost everybody has already said. It is not easy, to sit in front of a computer for a long time, but what I must admit is I have hardly left my chair for just going to the washroom. That was the level of conference which we had. It was an immense conference, done very, very well. And I think the credit goes to the entire organizing team led by Dr. Mehra, Dr. Kanga, and the rest of the team from both Aphia and Ishi. And really a wonderful, wonderful experience. We really, really enjoyed every bit of it. And we are really looking forward to hosting the workshop in our country as well. Thank you very, very much, sir. And thank you, everybody in the team. Thank you.
Thank you, Dr. Handu, for those kind words.
Namaste, namaste, and namaskar. And we in India say fir milenge, see you again, fir milenge. I want people from Aphia to also echo with the words that we speak of namaskar, namaste. This is the way we greet from India, and we want to greet everybody. We will love you all from the core of our heart. And I think I have done huge number of conferences all my life, but this actually stands right on top. And because absolute academic feast. Absolutely, Ajay and Anil. I had to really wait for hours to go to the loo. We didn't want to leave the chair. Absolutely, yeah.
So just the last one information that all of those who are interested to get the ACHI points, a survey will be circulated to you, and if you fill in the information of the hours and the program that you have attended, now you have got a participation certificate, but the other one that you will receive will be with the ACHI points. So I request all of you to do fill that survey and give your details of the sessions that you have attended. And I thank everyone for attending this meeting.
Okay. Until we meet, in Dutch we say tot morgen, in India we say fir milenge, and in English we say see you again. Thank you very much.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you. Bye.
Thank you, Dr. Lata.
Bye. Bye.
Bye.